My name is Andreas, and in this video we'll deploy an AI model to the cloud and build a web app around it. The app lets us test how big of an impact specific genetic mutations will have in causing diseases such as cancer. The model we will deploy is Evo2, which is a newly released state-of-the-art language model that can be used to generate DNA and predict the impacts of genetic variations. I have made sure that everything in this video is free for you, including the GPUs and cloud compute required to host and run the model. You will also not need a biological background, since I'll teach all the biology prerequisites you'll need for this video. The user first selects a human genome assembly, which is a computational representation of the human genome sequence. Next, the user can search for specific genes. These could be cancer-related genes such as tumor suppressors, but they can also browse by chromosomes, where the user can pick a gene that appears in a certain chromosome. After selecting a gene, we will load the genetic variants that are already known within the specific genes from a public database. The already known classification, such as pathogenic or benign, is then shown. The user then has the option to analyze the mutation with the AI model and compare the AI's classification with the database classification. The entire nucleotide sequence of the gene is also shown, and it is possible to select a single nucleotide and run an analysis on that one to analyze its clinical significance. Under each variation, it is also possible to navigate to the public databases to see even more information about it. Likewise, information is also pulled from public databases about the selected gene. The AI model we will use was released from Stanford in February 2025 and is a large language model for biology. But instead of inputs and outputs being words like with ChatGPT, Evo2's input and output is a sequence of A, T, Gs, and Cs, which is what a DNA string consists of. It has been trained on many different organisms and has learned the syntax of how DNA should look. It is incredibly promising, and I'll break down many of the use cases and concepts behind it from the report. We will start by covering some basic biology before moving on to the dataset. Here we will dive into the dataset and go in depth with features the model learns in biology terms. In the report, a large variety of different techniques are used to determine if the model has learned certain things. I'll break some of these down since we can learn a lot about AI from these. Then we'll talk about how the model was trained, so which models were trained and how does the loss function work. We will check out the pre-training and mid-training phases. And I'll also break down how the researchers adopted the model from an 8K nucleotide context window and all the way up to a 1 million nucleotide context window. There lies some pretty cool techniques behind this which we can learn from. We will cover the trained model's use cases, and I'll break down how the model can be used for generation. We will then cover how the model can be guided with other AI models to generate DNA sequences with specific properties. Then we will cover scoring, so Evo2's prediction capabilities. Lastly, we will finish off with how Evo2's embeddings can power the training of new AI models. We'll take a look at how that might be done using a practical example. We will use serverless GPUs for developing and deploying the AI model, and we'll check out the pros and cons of serverless versus using EC2. We will go in depth with an example from the researchers where Evo2 is used to predict whether a particular single nucleotide variant of the BRCA1 cancer-related gene is likely to be harmful for the protein's function and thus potentially increase the risk of cancer for the patient with the genetic variant. We'll then adopt these techniques from the example for our web app. We'll talk about how to calculate thresholds for pathogenic versus benign, as well as the confidence in the score. Lastly, we'll go through the user flow in the frontend and where we get our data from. Our AI model and backend will be deployed completely for free with serverless GPUs on Modal, and we'll use NVIDIA H100 graphics cards. On the platform, we can check the status and requests to our backend. We'll use Next.js, React, Tailwind, Python, and other modern technologies. Now, let's get started. Let's start out with the basic prerequisites in biology that we'll need to understand why we use Evo2 and then also what it does. First of all, if you search for Evo2 and then find its GitHub repository, you should be able to go down and find the link to the paper. Just let this verify and then press download PDF. When this has been downloaded, you should be able to see the paper written by the scientists who made Evo2, this one right here. So much of the content from my SkeleDraw slides is based off of the content written in this report right here. So if you're interested, you can go ahead and just read the report. Maybe you understand some of it, maybe you understand a lot, but at least my slides will go in depth with why it works the way it does. So in this report right here, you can see all of the same facts that I'll be presenting, just more in depth. I'll of course also add some more facts about other topics, but this is just so you know where I have all my information from. Great. Let's start with the DNA we see over here. So over here we see a double-stranded DNA string, and a DNA string basically is just a molecule, And this molecule is like a recipe book or an instruction manual for building out a full organism. So all organisms have DNA strings, and these determine what your eye color is, for example, and then how all or every part of your body works. So the recipe is written in a pretty simple 4-letter chemical alphabet, so it consists of A's, T's, G's, and C's, and that is what you see over here, A, T's, G's, and C's. And since it is double-stranded, we'll need to keep the strands together, and this is done with base pairs. So A's always bond with T's, and G's always bond with C's, and this way the two strands are kept together. So a DNA string contains genes, and these are specific sequences that typically provide instructions for building proteins that can then do work within the body. Now we have two different type of organisms here. We have prokaryotes, so these are simple organisms like bacteria, for example, and these typically just have a single circular chromosome, so not a lot of DNA is needed to encode organisms that are simpler. We then also have eukaryotes right here. So eukaryotes are like plants or animals, including us humans, fungi or yeast. So the cells right here are more complex, so this means we'll also need more DNA to actually encode the organisms. So humans, for example, have 46 chromosomes, which is what we use to arrange the DNA in. So just remember that prokaryotes are simple organisms and eukaryotes are more complex organisms. And then I also wrote here that by DNA string, we mean physical DNA molecule, because this DNA string is actually stored in a physical DNA molecule that is within every single cell. Now imagine how many A, T's, G's, and C's we'll need to encode a human, for example. And also imagine that we'll need to store this entire recipe within every single cell, which you have tons of in your body. So these DNA strings are obviously going to be very long, so we'll need to pack them down into a way that makes them pretty compact. So if you folded out the DNA of a single human, it would be meters long, and that obviously doesn't just fit into a cell if it's just folded out. So we use chromosomes the body uses chromosomes to efficiently package this DNA into something that fits within the cells. So the DNA strings aren't just loosely lying around within the cells, it is actually tightly wound around proteins, which we call histones, and this is then further compacted around structures like chromosomes. So for humans, there are 46 chromosomes or distinct chromosomes, and each of these contains a segment of the overall DNA string. So it's like splitting this massive DNA string into 46 different volumes, basically. So when you hear chromosomes mentioned further into the video, this is what you have to remember back to. Great, so let's move on to genomes. Well, genomes is just another word for the full DNA sequence across all the 46 chromosomes for humans. So basically just the entire recipe book for building a human or for building another organism for that sake. So there are genomes that have been measured for multiple different organisms, and you can find these reference genomes in databases on the internet and then see all the individual A, T, G's, and C's and how they are ordered to build out a human. So genomes just contains all the information that an organism needs to function and then also reproduce. Okay, hopping back to the DNA string here, we can see the nucleotides. So these were the A, T, G's, and C's, and these are just the basic building blocks of DNA strands. So they're just fundamental units for carrying information about how to build the organism. So these will of course also be different from organism to organism, or even within the same organism if there are mutations. So as you see over here in the DNA string, we have the ATGCs divided into pairs. These are called base pairs. So going over here, if the DNA is double-stranded, then the strands, so this line right here and this line right here, will be held together by these different nucleotides that will pair. So A always pairs to T, and then G always pairs to C. So if you know the entire row or the entire sequence of ATGCs for one strand, you will always also know the entire sequence of the other strand because they'll always pair in this way. So there won't really be any ambiguity at all. So this is what is called a double helix, and this just consists of two strands winding around each other like you see down here. So when working with Evo2 as well, what we'll work with are single sequences of A, T, G's, and C's, and therefore not pairs, because as I said, these pairs will always be complementary to whatever they are supposed to be. So A will always pair to T, etc., and G always to C. So if we just have a single sequence, we'll always be able to find out the other strand over here. So we always just work with a single sequence instead of pairs, because we can always derive the pairs from the single sequence. So to just summarize the basic prerequisites, DNA strings are molecules of ATGCs, so nucleotides that encode how life is produced. It can be simple life forms such as bacteria called prokaryotes, or more complex organisms like eukaryotes such as humans or animals. And since these DNA strings can be extremely long because of all the information needed to encode a human, they are divided into chromosomes. So each chromosome contains a part of the DNA. So for humans we have 46 chromosomes, and for other organisms they can contain another amount of chromosomes. When we talk about genomes, this is just every genetic material in an organism. So a human genome, for example, would just be a sequence of all the nucleotides to create a human. And nucleotides are just the basic building blocks of DNA strands, so the A, T's, G's, and C's. And in double-stranded DNA over here, they are put into pairs called base pairs. So if you know one pair, you'll also always know the other one, because T or A always pairs to T and G always pairs to C. So these are the basic units we work with before you can start to understand Evo2 and how that works as well. Before moving on to the dataset and how Evo2 has been trained, let's talk about what Evo2 actually is, so we'll understand why we need the dataset that we do. Good. So what is Evo2? So Evo2 it is a large language model, first of all. So it's a large language model, just like ChatGPT is. So what ChatGPT and other large language models do is that if you give it some text, so for example, this is some text, then it'll be able to predict the next token. So this is a token, for example, this is a token, and this is another token. So if you give it and then just end it here and just give this as the prompt to ChatGPT, it'll be able to predict the next token and then continue with something like, and this is the second part. So this is something that ChatGPT will predict from this over here. So just like normal text is given to ChatGPT, for Evo2, we'll instead give it ATGCs and just give it a long sequence of that. And then what it'll do is just predict the next sequence of that. So it'll just predict more ATGs and Cs in a sequence that it finds fits well with the old sequence or with the previous sequence. So by taking all these genomes from different organisms, so this could be humans or animals, yeast for example, and all kinds of bacteria, and then giving all of these to a large language model as training data, they then trained a large language model to then predict the next A, T, G's, and C's in a sequence that it has been given as an input. So Evo2 specifically has been trained on over 9 trillion DNA letters, and it has also been trained on DNA letters from all kinds of life, so both from single bacteria to complex humans. So hopping into the repository here, for example, it has been trained across all domains of life, so all kinds of organisms it knows about. So it isn't just applicable to humans, but also a bunch of different organisms that it has been trained on. Great. So what the researchers has done with Evo2 is that because they have trained the model on this many DNA sequences or ATGCs, Evo2, the large language model, has then learned the syntactical correctness of the ATGs and Cs. So if you give it an ATG or C, it won't just randomly predict the next ATG and C, it'll then actually predict something that would fit into the gene. So to build a large language model like Evo2, the researchers behind this model needed a bunch of training data. So we'll just take a look at the dataset and then also how it was trained. Good. So hopping over here to the dataset, the researchers created a dataset called OpenGenome2. So this was about 8.8 trillion tokens of nucleotides, so A, T, G's, and C's, in just a long sequence of strings. And this dataset was created and released by the authors of the model, so not some external researchers. Both the dataset and the model was created by these researchers. So when training this Evo2 model, the researchers behind the model used a 2-part training strategy. The first part is called pre-training and the second part is called mid-training. So for this pre-training section right here, the training data was limited to much smaller sequences containing bacterial genes, for example, so prokaryotes, instead of huge organisms like yeast chromosomes or human chromosomes. The difference between the pre-training and the mid-training can be compared kind of to reading one book at a time versus reading one page at a time. So for the pre-training, the model was given one page at a time, so learning the shorter stories. And then when the model had learned a bunch from those 1 pages at a time, they then gave the model an entire book at a time to also teach it larger organism sequences such as yeast chromosomes. So with pre-training, they gave the model 8K context, an 8K context window. So what this means is that they gave it up to 8,000 nucleotides at a time when learning to predict the next nucleotide. So when working with a context window of 8,000 nucleotides, the model isn't big enough to, or the model's context window isn't big enough to learn about these organisms over here, but it is big enough to learn about bacterial genes, tRNA, and these smaller sequences that you might find within this range. So we basically just teach the model the fundamentals, and then when the model has learned the fundamentals, the researchers then teach the model the larger organisms. So this scale, just for reference, is small organisms to the left and larger organisms to the right, based on how many nucleotides they contain. The specifics about what is learned during pre-training versus what is learned during mid-training, I'll come back to a little bit later in these slides from in Excalidraw, just so you understand fully why the researchers divided it into two parts like this. So let's first look at the pre-training part of this training. So for the pre-training part, the model has been limited to 8,000 nucleotides context window, and then it has to predict the next ATGSE within that sequence. So with this pretty small context window, the basics of the DNA grammar can be learned. So for example, if we relate this to a ChatGPT clone, if you were to train a ChatGPT large language model where it predicts letters, for example, in the English language, if you gave it smaller context windows like this, the pre-training might teach the model how to spell with the English language. So the model might be able to spell basics of DNA grammar like this, just like the basics and fundamentals of the English language. And then when expanding to mid-training, the model might be taught specific knowledge that can then express with the fundamental knowledge that it learned within the pre-training section. So that was kind of the idea with pre-training and mid-training. Also, some of these patterns of A to G's and C's, because these can be arranged into different patterns, can then also be picked up by the model in pre-training. So with this bacterial gene, for example, it might be small enough to be fully contained within an 8K context window, and the patterns that might be contained within that sequence might also be picked up and learned by the model already in pre-training. But for the patterns for these larger organisms, they might have to be taught during mid-training, because the context window is too small for the model to pick up the patterns already. In these sections down here, I would like to talk about some of the tests that have been done in the report post-training to validate the effectiveness of the pre-training up here. So while these tests are done post-training, the knowledge being tested is precisely the kind of information that the model would have picked up during the pre-training phase. So these tests down here can therefore serve as an illustration that the pre-training stage was successfully done, and that the model actually did learn the crucial basics of the DNA grammar. So as I mentioned earlier over here, if you have a sequence of A, T's, G's, and C's, these can code different things. So one part of the sequence might code for a specific protein, while another part of the sequence will code for another protein. So with that in mind, let's hop over here and talk about codons. So codons are a fundamental unit of genetic code, and codons consists of 3 nucleotides that encode an amino acid. So proteins consists of amino acids. So you might have a sequence of codons that will then code the entire protein by coding for each specific amino acid in an order. So most of these codons specify which amino acids should be added next when building a protein. And remember, proteins are the molecules that perform the tasks in the cell, as we talked about earlier over here. So let's say we have a sequence of A to G's and C's, like this. Then this could be a specific codon for a specific amino acid. This could be, let me just mark this. This could then be the next amino acid in the protein. This could be the next amino acid in the protein. But how do we know when to start coding the protein? So for this, there's something called start and stop codons. So these are the signals that are used for marking the beginning and end of the protein coding. So this is one of the patterns that I talked about up here. So this is only 3 different nucleotides. But imagine you have a sequence that is maybe 1 million in context window or 1 million nucleotides long. Well, then in that case, 3 nucleotides in that sequence is a very small pattern, and it might be hard for the model to pick up if it were given 1 million nucleotides. So with the pre-training, by giving it only an 8K context window, it might be more efficient for learning these smaller patterns consisting of only these 3 nucleotides for the start and stop codons, for example, than giving it 1 million nucleotides and then hoping it picks up these small patterns of only 3 nucleotides. So after training the entire model, both with pre-training and mid-training, checking if the model has learned the patterns of start and stop codons, we can actually verify, or the researchers could actually verify, whether the pre-training was done successfully, because if it was done successfully, then the model should be able to know these start and stop codons because it has been taught the patterns. So the researchers wrote a little bit in the report about how they can check whether it has learned about the start and stop codons. So to figure this out, we have to take a look at what the capabilities of the model is. So going into the GitHub right here, we can see what it is able to do. One thing it can do is forward, so this is called scoring. Another one is generation, so this is what we basically do with ChatGPT. We give it a sequence of letters, so this one for example, for ChatGPT, if we use that example, and then it'll be able to generate the next letters in the sentence. So that is what we do with generation. We just predict the next A, T, G, and C from a previous sequence of A, T, G, Cs. That is generation. The other one is scoring. So with scoring, you can give it a sequence, and then you can get the model to predict how likely it is that the T is actually the correct letter that is supposed to be after A, C, and G. So we can give it a letter like this and a sequence, and then the model will score how syntactically correct that it is that the T comes after the A, C, and G. So that is basically scoring. So if we go back to the example with ChatGPT, if we give ChatGPT Evo2 can generate, and then we also give it the token DNA, then ChatGPT would be able to score how likely it is that DNA comes after Evo2 can generate, versus if we, for example, put in a totally unrelated word like lamp, so Evo2 can generate lamp, well then the word lamp would get a lower score than the word DNA, because DNA is more syntactically correct in this context. So that is basically what scoring means up here. Good. So hopping back, how can we use these two features to check whether the model has learned about these codons or not? Well, we can use the scoring, and just to read what I've written here, since another one is scoring, so the feature scoring, since we are able to generate A, T, Gs, and Cs, Evo2 has learned the DNA syntax rules. I really want to hammer that down. Therefore, we can give Evo2 an input and have it score how well each nucleotide conforms to the patterns learned. So to solve this problem, we can use these steps. The first step is give it a sequence where we put in the correct codons and then get the score out. So maybe we put in a sequence where the codons are actually correct and correspond to how it would look in real life. This will then give us a score for that sequence. Step 2 is we do the same, but we create a little mutation in the codon. So we take the same sequence, then we change one of the A, G, T's, and C's to another one. So maybe we change the G to an A, so now it's AAC instead of AGC, and this is obviously incorrect, so the model should have learned this and then score that sequence lower. So now that we have a score for the correct sequence and for the incorrect sequence, we can then take a look at the average score, the difference between those two scores, and see if there is a difference. And this can be done with a heat map plot, for example, where the colors are more red whenever the difference is larger. So the model should assign lower scores now because there is a bigger difference with this mutation. So this basically just means that it is more syntactically incorrect because the model has learned the patterns and the syntax of how ATGCs is supposed to be structured. So if we see lower scores where these codons are, this would then confirm that Evo2 has picked up about what a codon actually is, because the protein is spelled incorrectly and the model knows that the protein is spelled incorrectly. So let's take a look at one of the diagrams from the report. So this is basically just a mutation analysis that was done, like I explained up here. So what we see in this diagram, first of all, is a heat map. So on the x-axis right here, we have a sequence of nucleotides. So this is part of a protein coding sequence. And then we have different nucleotides for each one of these ticks. So this could be an ATGC, this could be an ATGC, etc. And then because codons consist of 3 nucleotides, then there is a codon 1, 2, 3, 4, blah, blah, all the way up to 7. So the first codon would be 1, 2, 3 nucleotides, which matches up with what we explained up here. So over here, they explain delta likelihood. So this symbol right here means delta likelihood, which means the change in the scores from when they introduced the mutation compared to the reference. So if the mutation is really bad, then the likelihood will be pretty large. The delta likelihood would be larger and the color would be more bright because the model knows that the mutation is bad. And again, the model knows that this mutation isn't correct because it has learned the syntax rules of where codons are supposed to be from the pre-training. So first of all, we have different species right here. So we have simple species up here like E. coli, and then we have Homo sapiens down here, which are the humans. And since the x-axis is nucleotides, so just a sequence on the x-axis right here, what this 5 UTR means is just a region before the coding. So there is a region right here, and then there is another region in the sequence right here, which is the coding region. So this region I have marked right here is the region that starts coding for that specific protein. So we therefore see a start codon right here, so start of amino coding. So that is what we see right here. So when introducing mutations into the start codon, or the part of the sequence that marks the start codon, so this part of the sequence right here, and then comparing the sequence that has been mutated compared to the sequence that is a reference sequence, so a sequence that is actually correct, we see that the model picks up that the difference in scores is pretty big. So this means that the model can actually detect whether the syntax is correct or not, because if we introduce a mutation into a start codon, the model knows that, oh, that probably isn't correct right in that place where the start codon is supposed to be, so we'll give it a worse score. And because we know that this place right here is the start codon, we can then verify that the model has picked up what a start codon is, and that this pattern actually does exist. So this region over here, the 5 UTR region, doesn't really contain the codon, the information about the codon encoding, like this region over here does, but it does have some information about how the gene is turned into protein, and as you see, the mutations can also be learned and detected over here. So it probably contains some pattern that the Evo2 model has picked up on, and this can then also be detected that the model has learned these patterns from using this same mutation analysis where we compare a reference sequence with a mutation sequence and then check whether the model picked up on a difference in the scores. So the key observations we just want to make here is that the model is extremely sensitive to almost all species, so both the E. coli and all the way up to Homo sapiens, so humans, so across multiple species, because as I said before, the model has been trained on multiple species as they describe. It'll be able to pick up these mutations within all of these species. And the model also clearly picks up the importance of the start signal, since that is the lightest part of this sequence right here. And then we also see this periodic pattern every 3 nucleotides, so right here, right here, right here, right here, and so on and so forth, corresponding to each codon. As we also see the 2 first nucleotides in a codon, so this one right here, for example, causes larger changes in the likelihood over here than in the 3rd nucleotide right here, of a codon. And as far as my research goes, changes in the 3rd position often also don't change the amino acid, meaning that the model has probably picked up that changes in the third nucleotide in the codon right here makes less of a difference to the end results of the protein produced compared to changing the 2 first nucleotides in the codon. So it has learned these very small and fundamental patterns in how DNA is encoded. So just to summarize, by systematically mutating sequences, like it has been done right here, then observing how the score changes for the scoring feature with the Evo2 model, it can then be confirmed that Evo2 has learned this fundamental structure of codons, so start codons, and then also codons and which parts of the codons are most important. And this is then done for both the small organisms all the way up to Homo sapiens. So it has learned this across a large variety of species. So since this is what it should be able to pick up in the pre-training up here, because it's so small, such small changes, it helps indicate whether the pre-training was successful or not. And if you're interested in how this scoring works, this is what we'll work with later on in the video when we actually build out the tool. But for now, this is just a snippet from the GitHub of the scoring feature. So how it basically works is that you create a new instance of the model right here, which will then load the model file into memory. This is Evo2 7 billion parameters. Then you give it a sequence, so a sequence could be A, C, G, and T. Then you can run scoring by running evo2_model. So what we instantiated up here, along with the input IDs, so the sequence that has been tokenized. And the output down here would then be the likelihood across the DNA sequence, so A, C, G, and T. So what they did was they just took the sequence, or what they did up here, just to make myself clear, was they took this sequence of A, C, G, and T. So let's say A, C, T, and G was correct. Then they ran the score, they ran this code once, then they got a score out, then they changed the C maybe to a G, so now it's a GGT, so we introduced the mutation that is wrong. Then you run this entire code again, get out another score, then just compare how big the difference is between those 2 scores, and then if the difference is big, then there is a large likelihood, so the color is light, or large difference in the likelihood, so the color is very light, Otherwise, the color is dark because the model didn't really pick up any differences. So the score would be kind of the same. So that is kind of how they did this test right here with codons. Going down further, we can also talk about introns and exons. So just to give you an idea about what introns and exons are, if I just create a random sequence like this might be a DNA sequence. So a gene is one long sequence, but it might not be continuous. So maybe we have a specific gene that codes for something, and then that gene is contained right here, and then it is also continued right here. So these three parts right here aren't actually what codes for the gene. So it is differentiated between what parts of a sequence codes for the gene and what part of the sequence does not code for the gene. So exons is called express regions, and these are the segments of a gene, so maybe this part right here, that actually does code for the gene. So they hold the instructions necessary to build the protein, so the codons. And these are the essential and important steps to be able to construct this protein. And then there are also introns over here. This means intervening regions. So if this was the exon right here, then these three nucleotides that does not code for that protein, it can be called an intron. So the introns are located between the exons, but still within a gene sequence, and these introns do not code for the final protein. So while they are part of the initial gene transcript, they are removed later when the actual protein should be synthesized. So just to try and rephrase that as a simple example, if you're watching a movie, then you're only obviously interested in the movie and the storyline. So the introns would then be the ads in the movie. So they're part of the movie, but they aren't really what matters within the movie. They aren't part of the storyline. It's a completely different thing. So just to rephrase that in a more scientific way as well, DNA is a long sequence. So exons are part of the gene that contains the valid coding sequence. So this is what we actually need to build the protein. And then introns are the parts in the gene between exons that does not code for the protein, but are just there. So an illustration down here shows the exons, so the red parts does code for the actual protein we would like to produce, and the introns are still part of the sequence and the gene right here, but they don't really matter in the RNA later on. So as you see, what happens, the introns are just removed at one point, and then all that is left is the exons right here, which is the actual coding sequence. So because Evo2 needs to learn the syntax of how DNA works, it obviously also needs to be able to recognize the difference between exons and introns. So these introns between the exons that actually does matter can be highly variable in length. So they can vary between maybe tens or hundreds of nucleotides all the way up to maybe hundreds of thousands of nucleotides. So taking this back to the pre-training, if the pre-training only has, let me just make this a little bit smaller and just delete this again. Right there. So if the pre-training only has a context window that allows the model to accept a sequence in this length, then obviously we won't be able to see all the exons that is required to code the gene. But the model might fundamentally be able to learn that there is a border between an exon and an intron right here. So the model might be able to learn the borders between exons and introns, but not actually which exons correspond to each other and are related. So whenever we have a training that is mid-training, so the mid-training might have this entire context right here, then the mid-training might be able to see, oh, this exon relates to this exon, and this exon relates to this exon. So those are the two different things you might expect to see, or might expect the model to pick up between the pre-training right here and then the mid-training. And ultimately, the model should then be able to distinguish between whether we are in an exon or an intron given a position in a sequence, so just a single nucleotide position. So one way that we can differentiate between introns and exons given a single position, so if you just give a single nucleotide position like this, the model should then be able to classify whether we are in an intron or an exon. So one way you can be able to do this is also explained in the report, and that is by using something called embeddings, and that is another feature in Evo2. So going down here, all the way down to embeddings, that is another feature apart from forward and then generation. So what is embeddings? Well, here it says Evo2 embeddings can be saved for use downstream. So just with forward down here, you instantiate the model, tokenize the sequence of ACGCs, and then you score a sequence. But instead of just getting the outputs like you do up here in the scoring, you also get out the embeddings. And the embeddings are the model's internal representation of the sequence within its hidden layers. So we can just go ahead and hop over to a later section, which I'll come back to later as well, just so you know that, and just talk about embeddings. So this is one of the features that Evo 2 has. So right down here we have a neural network, we have some input nodes right here, some output nodes, and then we have the hidden layers in here that contains the model's hidden state of the input layers between the inputs and the outputs. So the model will get some input numbers, do some transformation to those input numbers with its weights and biases, and then take those input numbers and output them in some way. So an embedding is just taking one of the representation of the inputs within one of the layers of the model. So if you go back to the GitHub, that is what they do right here. They pick that the layer blocks 28 MLP13. So that is just a random layer from the architecture of the model. Although they haven't picked that layer randomly, they have probably thought well about which one to pick out. And the reason you might want to use these embeddings is that these embeddings, although they're just numbers, might express the input sequence better than the input sequence does itself. So let's say the input sequence is just A, T, G, and C. Well, then inside the embeddings, it might be a bunch of numbers, just random numbers, which are the internal representation of the A, T, Gs, and Cs after being run through these layers. So even though we aren't able to decrypt what these numbers actually mean as a human, the model might know that this contains information about what introns are there, what exons are there, and all of this syntactical knowledge that the Evo 2 model has learned through its training. So going up here, Evo 2 might be able to capture more accurate features in its embeddings than other models are. Therefore, Evo 2's embeddings would be more precise. So if you want to train a model to classify whether you are in an intron or an exon given a specific position, it might be more accurate or might give you better results if you give it an embedding of a model like Evo 2's, because it contains information about introns, exon, and everything that the model has learned, instead of just giving it the input sequence A, T, G, and C. So Evo 2 embeddings, for example, reflect knowledge about codons or introns and exons more than the raw data itself would do, just because it has learned all of these informations. Well, what can we then use this for, you might ask? Well, if we want to build out another model that can classify whether we're inside or outside an exon given a position, we can then use Evo 2's embeddings as inputs for the model, instead of just the raw data, so the position and sequence maybe. And they actually explain in the paper how they do this. So going down to page 5, we can take a look at these diagrams. So what they did in the report was that they build an exon classification model. So they built a much simpler model than just the Evo 2 model. And the input for this model is an input sequence, so a sequence of nucleotides, and the output is a likelihood, so a number between 0 and 1, about how likely it is that we are inside an exon. So it would then use the extracted embeddings from the Evo 2 model. And then right here, the blue parts are an exon, and then an exon over here as well. And then the white part between the blue parts is an intron. So this means that if you give it the position right here, then it should predict 1 because we're inside an exon. If you give it a position inside or between the exons, so in the intron, then it should predict 0. And then again, if you give it another position inside another exon, it should then predict 1. So we can then also see over here how the model they trained using the embeddings did. So looking at the labels, an exon, the dark gray part right here, this is an exon, this is an exon, and then over here on the y-axis, we see the scores. So this is 0 for an intron, and this is 1 for an exon. So we can see that it predicted high scores close to 1 if we're inside exons, and it then predicted low scores closer to 0 if we're inside introns. So that is how it can be verified that Evo 2 actually did learn the difference between introns and exons. And the reason we know this is because it is obviously embedded within the embeddings or the hidden states of the Evo 2 model, which can then in itself be used to train other simpler AI models to classify whether we are inside or outside an exon. So that is how it can be verified that the pre-training was successfully done for learning introns and exons like this. So again, these AI models are great at learning patterns. So just like this up here with the codons is a pattern, this down here with the introns and exons is just a pattern. So if the model is able to be taught patterns such as what is an intron and what is an exon, it might be useful for working with or training other models in the future that also deals with introns and exons, because we can utilize or leverage the knowledge that Evo 2 has from reading all of these different nucleotides in its training data. Another thing that is probably also part of the pre-training phase and learning the fundamental DNA grammar are promoter motifs down here. So promoter motifs marks the start of a gene in a DNA sequence. So let's say we have a DNA sequence like this. Well, the gene might start right here, and then the promoter motif will be some part of the sequence right before that. So just how codons up here mark the start of a protein with a start codon, the promoter motifs down here mark the start of the entire gene. So it's like a start reading here sign right before the genes begin. And this 8K context window of nucleotides might be enough to actually see both the promoter motif and also the gene that starts right after. So being able to teach the model that a promoter motif actually marks the start of a gene because it fits within the context window. So hopefully the model can learn what is usually written before a gene begins. So it'll learn the concept of promoter motifs. Good, so down here there is a pretty small diagram from Wikipedia. And the only thing I just wanted to show you is the promoter motif right here, the orange part, and then there is a gene right after. So a promoter motif is just a part of the sequence right here that comes right before the gene. The rest you can just ignore, but I just wanted to show you that it starts reading from the promoter motif and forward. So in the report, they actually tested whether the model has learned these promoter motifs, and they used a more advanced technique called sparse autoencoders to actually test this. So this isn't what we have seen before and talked about before with mutation analysis, for example. This is a more advanced technique which you can learn about both here in the report and also online by searching for it. But here in figure 4 and then figure F, you can see that it actually did learn how to detect these promoter motifs. So the x-axis down here marks the distance from the transcription site, so where it should start reading from. So going back here, this is the transcription site where it should start reading. The promoter motifs then marks the area right before the transcription site. So over here, that is as close to the transcription site as possible, as you see with the gray color right here. And then the promoter motifs are what is being detected right here before as these lit up color bars right before the transcription sites. And they also found that where the model lights up, so where the model detects these promoter motifs, the sequences also correspond to the motifs that are actually known. So specific sequences of A, T, Gs, and Cs that corresponds to how motifs actually look in real life. So this was just another piece of knowledge that the model probably learned during pre-training, just so you know what the model likely learns during both pre-training and also mid-training down here. So in mid-training, the model is trained on longer sequences, so it's given maybe multiple genes at a time. So during pre-training, the context window is only at 8,000 nucleotides. So using only these 8,000 nucleotides might be fine to teach it the basic grammar of DNA. And it's also way less expensive computationally since there's much less data to compute, but it also doesn't teach the model the large picture about massive genomes. So let's say you want to feed it an entire genome containing multiple genes. Well, maybe there is a relation between two genes within that genome, so only giving it part of one gene obviously won't teach the model the relation between those genes within the genome. So by using this larger mid-training phase as well, these more full picture relations can be taught to the model. So this of course is about handling complex sequences first of all. So remember eukaryotic cells, well these are the complexer cells such as those within humans for example. Well, there might be patterns between different genes within these eukaryotic cells. So by just looking at these small fragments that would be looked at with only the pre-training window, well those larger patterns can't really be understood. So their intricacies aren't well understood by just looking at small fragments of these DNA strings. So I have a little analogy over here. So it's like understanding a whole book by reading only one page of the book. So one page might be the 8K context window, whereas the whole book might be the million nucleotide context window. So that is kind of the difference between the pre-training and the mid-training. So during pre-training, if introns are super long, then the model won't really learn much about the exons, which actually encode the genes, so we need more context. So let's say you feed the model a sequence of 8,000 nucleotides for the training data, for example. Well, if those 8,000 nucleotides happen to be part of an intron, well then it doesn't really learn about the nucleotides that actually does code for the gene, so the nucleotides within the exon. So by giving it a larger context window, it can have both the nucleotides from the intron and the exon, and then learn which actually code for the gene. And again, the larger context window also allows the model to learn the relationship between different genes because they might be related across large distances. distances. Also, what a model can be used for after training largely depends on what the model was actually trained on. So to be able to generate or score sequences that are longer than just the 8,000 nucleotides, you would like to train the model on sequences that are longer than these 8,000 nucleotides, just so we're able to use the model for predicting or scoring sequences even longer than just these 8,000 nucleotides when actually using inference with the model. Another thing that can be taught during mid-training is the knowledge of regulators. So different cells need different proteins and also different amounts of proteins. So even though each cell contains the entire recipe or the entire DNA and all chromosomes for building the entire human, specific cells only need a part of this entire genome. So therefore specific genes can be silenced or enhanced to control what proteins are produced within that specific cells and how many of these proteins are actually produced. So this means that genes aren't always turned on, and there are therefore enhancers that can boost genes' protein production. There are also silencers that can decrease gene activity. So these are kind of like volume controls for genes. So when looking at a DNA string like this, or just a sequence of A, T's, and C's, you might expect the gene to be right here, for example, and then the silencer or enhancer part of these sequences to be right after the gene, such that it is known the gene to be interpreted along with how much it should be silenced or enhanced. So you would expect all these information to be right next to each other, right? Well, in reality, this information, so the gene and then the silencers, can be hundreds of thousands of base pairs away, so this will take up a lot of context. So because these genes and silencers and enhancers can be that far away from each other in this long sequence of A's, G's, and C's, this can really be taught to the model that there is a relationship between these parts of the sequence in this pre-training window. It'll have to be taught to the model in the mid-training window that allows for much more context. So physically, a DNA molecule like this can loop around, such that if this is just the long sequence of A's, G's, and C's, this is just like a long linear sequence, well, this can physically loop around such that the gene can be brought close together to the regulator, such as the silencer or the enhancer. But because we just work with the long linear sequence of A's, G's, and C's, when working with nucleotides and AI, for example, we don't really know that the gene is looped around. We just see the entire sequence as one linear string instead of this looping around motion. So the DNA molecule can loop, bringing these two close to each other, but we can't really see that in the nucleotide string though, because we just see the long linear string. So in real life, even though the gene and regulators are super far apart on the DNA strand, they are brought close to each other by folding of the strand. So to actually teach the model that there is a relationship between the gene and the regulator, even though we just see this long linear string, a large context window is needed to teach the model the relationship between, or teach the model of the relationship between the genes and the regulators, even though they are far apart. So this is another obvious reason for having this mid-training phase as well. So let's hop down and take a look at the distribution between the pre-training and the mid-training phase during the training. So as we can see from this diagram in the report, far more tokens are consumed during pre-training than during mid-training. So the reason is that during pre-training, the model needs to learn everything from scratch, so the fundamentals of the grammar itself. And then in mid-training, it has already learned this fundamental language, so the DNA syntax, and it just needs to adapt to patterns that it then sees in the training data on a larger scale. So down here on the diagram, we can see that the difference is pretty big. Pre-training takes up most of the nucleotides of the training data, and mid-training then takes up a much smaller part, as you see on both models right here. So just to summarize, the pre-training uses massive amounts of data with a much shorter context window to build a pretty robust understanding of the DNA rules and just the DNA grammar from scratch basically. And the mid-training phase then uses a smaller amount of the total training data set, but with a much longer and much more expensive context window. And this is then to adapt the model and teach the model long-range patterns across entire genes or between genes even. And these are things that wasn't possible to be trained within the pre-training phase. Another consideration that can be made for why there is both a pre-training and mid-training phase is that training in the mid-training phase is most likely much more expensive than training in the pre-training phase because you have to handle that much more data. So it might be unnecessary to use this massive context window that was used in mid-training for learning just the fundamentals of the DNA syntax that can be learned with a much shorter context window that is also much cheaper, as was done right here in the pre-training phase. So that likely also contributed to the decision about why there is both a pre-training and a mid-training phase. So let's just take a look at why not just train on human DNA instead of just training on all these different organisms that is shown right here in the paper as well. So the only human genome that was trained on in the training data set for the EVO2 model is the human reference genome. So this human reference genome is one long sequence based on measurements about how the human genome should look to not contain variants basically. So measurements have been made on actual humans to see what the human genome looks like, and then one long sequence has been made from that, and that is then the human reference genome. And then the rest of the genomes comes from other organisms, so these are then also part of the dataset. So in the paper, they tried the model EVO2 on the BRCA2 cancer dataset, and this is a dataset that contains variants and mutations that the model can then predict whether is bad or good. So let's say you have a DNA sequence like this, then a dataset such as this one would then say, oh, what if this is changed to an A, for example, and then it would have a label that says, okay, this is harmful or this is not harmful. So this way, mutations can be labeled, just for pre-reference to what this means. So when given this BRCA2 cancer dataset with these variants, the model could predict that the variants found in the dataset was harmful without having been fine-tuned to the BRCA dataset before. And it performs even better than the existing specialized model to predict how harmful these variants are. So since this EVO2 model hasn't been fine-tuned to the BRCA2 dataset variants, it obviously must have learned that these variants are harmful from some other source. So this is obviously the multi-species or all the different species that has been a part of the training data that it has then learned this from. So having been trained on so many other species, the broad knowledge of this training data has then helped the EVO2 model perform better for cases like this, because it has so many different species to look at when actually doing this assessment. And this brings us to multi-species variation. So EVO2 has learned what parts of DNA change a lot across species and what parts stay very similar. I'll just say stay right here. So the parts that often stay very similar should therefore be more functionally important, since they often are the same across different organisms. So variations within these different sequences or parts of sequences are then also more likely to be harmful. So variations in these parts of the BRCA2 dataset are detected with help from having been trained on many species. And this is pretty cool because having a model like EVO2 that has been trained on maybe a mouse and also a mammoth, it is able to use this information from these other species to actually predict whether a variation in a BRCA2 gene is harmful or not. So this is a pretty cool thing that they found out in this EVO2 research paper. And in the paper they write themselves, notably the only human genome in the Open Genome 2, so the dataset, is the reference genome, and EVO2 achieves this zero-shot performance without any human variant training, but instead leveraging multi-species variation as a proxy for evolutionary constraints. And a constraint, or what they mean by constraint right here, is for example that if you change a specific or create a specific mutation in this BRCA2 gene, then that would be a constraint because it might be harmful. So hopefully that cleared up a little bit about the dataset that was used for training EVO2, and then also what EVO2 is supposed to learn from this dataset. But to go more in depth with the actual training and how to carry out the training itself, let's take a look at this section over here. So for the model itself, EVO2 was released in several different sizes, so primarily a large 40 billion parameter model, and then also a smaller one that is 7 billion parameters, and then the super small one with only 1 billion parameters. So more parameters generally mean a more powerful model, but it also contains more computational resources, so more GPU memory to actually run these models. And it also takes much more or requires much more training data to actually train these models, so they will probably be even more smarter if they have more parameters. So this table right here from the report shows how many parameters each one of these models have, and then also how many tokens was used for training data down here. So for this 40 billion parameter model, they had 9.3 trillion nucleotides, and for the smaller one it was 2.4. And then you can also see how many layers were in this neural network for each of these models. So we'll primarily work with this one for this video right here, but you can also work with this one if you do require even more processing power or even more accurate results from the tool which we will build. So all the code for actually building the model, training it, and running inference with it is actually public on GitHub. So over here you can see it, it's entirely open source, and it was built from or with a repository called Vortex or tool called Vortex, and then trained on top of that. And then the actual architecture of the model is based on something called Striped Hyena 2, which is also a modern architecture that was built partly to train models such as EVO2. So I won't go much into depth with that. You can look into both striped hyena and also vortex if you'd like to, but let's hop back here. So with Vortex, what you do is that for each model you would like to train, you do go in and create a configuration file, so a .yml file, where you can define the different layers you would like in the model. And then Vortex can help take care of some of the hard work for you. So let's just first take a look at how inference is ran with the model, and then we can take a look at how the actual training is done below. So inference is done with the forward method right here. So a forward method in a neural network is just the most basic building block of all model operations. So a forward method receives the tokenized sequences as a tensor, then it output logits. So this is just the raw output of the model. So a logit is the following. So there is one logit for each possible token. So A, T, G, and C, and also short combinations of these. And then each logit has a score for how likely that token is to be the next one in the sequence. So let's say you have a sequence of A, T, G's, and C's. Well, then there might be a logit of G, for example, and then this one will have a score of how likely this is to be the next one in the sequence after A, T, G, and C. So that is logits, and that is basically just the raw output of the model. So this makes sense for how to use the scoring function, since the scoring function outputs how likely it is that that sequence is syntactically correct. So that score up here that it outputs is just the score of how syntactically correct that sequence is. So a simple call like this is then used for scoring, but if you want to do actual generation, so what you might see with ChatGPT where it generates the next tokens, as you see right here, then you can call a generate function. And how that works is that it calls vortex generate, where you pass in the input sequence, and then this vortex function tokenizes the input sequence, makes multiple autoregressive calls. So this means that this function right here keeps calling the forward function where it scores what token is most likely to be the next one, and then appends that most likely token to the sequence. So when looking at an existing sequence and then predicting the next one, and you keep doing that, you call that autoregressive because you look at the previous sequence. And after having done this a couple of times, or as many times as you want, it will then just return the predicted sequence, so the fully generated sequence, and not just the logits, so the scores for the different tokens. And I just want to clear one thing up. Tokenization, as it says right here and also here, means that it takes a sequence such as ATGC right here, and then it turns it into a tensor, which is just a matrix that the computer is able to understand. So the computer isn't inherently able to understand ATGs and Cs and doesn't have much meaning, but a tensor where this has been turned into numbers that the computer can work with makes more sense for the computer. So that is just the process of tokenization. And then also a note I made over here of autoregressive, so a more formal definition maybe is that autoregressive means predicting the next part of a sequence based on the previous sequence. So if you have ATGCs, then it can predict that after this comes an A, for example. So this is basically how the inference step works for both the forward function right here and then for the generation function. For the embedding specifically, what you do is just run this scoring function, and then instead of using the logit output, you just take out the embeddings. So this really isn't something for itself. This is just a part of the scoring function up here. Good. So hopping down, we can take a look at how the loss function has been described in the report, and then how it works and how they define it to be as optimal as possible for the training phase. So during training, when a model makes a prediction, a loss function determines how wrong that model's prediction were, and then an optimizer changes the weights and biases to hopefully make it a little bit more correct next time. So this is the fundamental cycle of supervised machine learning training, and the specific loss function can then be defined to punish the model more or less for a certain behavior. So first of all, you need a way to measure how wrong your model's predictions are when training it. So the measurement of wrongness is called the loss, and that is what the loss function calculates. So Evo2 uses cross entropy, and that is just the standard for tasks predicting probabilities of different options. So the probability of either the next token being an A, T, G, or C. But they do modify this cross entropy loss function a little bit though. And the reason is that the DNA sequence can be divided into two different parts. The first part is the region that contains the unique parts that code for that gene. And then there's also repetitive regions such as introns, as we talked about earlier, that are non-coding for genes. So these parts of the DNA sequence doesn't matter as much as these parts that actually does code for the genes, such as exons. So what this means is that if the model messes up and predicts a wrong A, T, G, and C, so a token, in a part that doesn't code for a gene, so within an intron for example, it doesn't matter as much as if the model predicts a wrong token in the part that is important for coding of the gene. So if the model predicts a wrong token in an exon, it is pretty bad because that actually codes for the gene. If it predicts a wrong token in the intron, it doesn't really matter because that doesn't code for the gene anyways. So these two cases can be taken account for by changing the loss function a little bit. So in the report, they defined the loss function as the following. So first there is the loss score. That is the variable you see over here to the left. And the loss score is just a number that defines how wrong the model was in this prediction. The loss score is calculated from the sum of all losses, and a sum is taken with the large sigma symbol, so that is what you see right here. And the large sigma summarizes this statement right here across all different losses ran through this loss function. And each of these evaluate to the loss of the single position or single prediction, and then we multiply a weight onto that. So this is just the loss. So if we just remove the weight right here, for example, then the loss would just be the same regardless of whether you're in a repetitive region or not. But then by multiplying this weight onto it, you can punish the model less hard if you are in a repetitive region than if you are not in a repetitive region. So the weight is determined if you are in a repetitive region or not. So if you're in a repetitive region, it'll be 0.1. So this means you will punish the model 10 times less hard if you are in a repetitive region. Otherwise, you'll just punish the model as normal. And then this is being summarized, and then the average of that is being taken. So over here, each loss, so the loss of CE, is the loss with the weight multiplied onto it. And then the weight is then determined by whether you are in a repetitive region or not. So as I write right here, it comes down to if the loss is reweight, the loss is reweight such that a wrong prediction in a repetitive region is only 10% as wrong as a wrong prediction in a coding region. So let's take a look at what made the researchers actually do this, and they describe a little bit about their thought process behind this in the report. So the reason they wanted to do this was to encourage the model to learn unique parts of genomes instead of just simple repetitive parts, such as seen in the introns, for example. So they write here, this loss has been found in other DNA models to improve performance on downstream tasks and better calibrate likelihoods between repetitive and non-repetitive DNA. So it made the model perform better, which is great. But now you might question, how do you determine whether you're inside a repetitive region or not? So how does determine whether a region is repetitive or not right here? So this method for determining this wasn't actually described in the research reports, but it seems that it was annotated in a pre-processing step. And this I get from the following lines. So this affects the genomic window and whole genome portions of the data, which contains these annotations. So they describe how they reweigh this cross-entropy loss, and then they describe the annotations right after. So it seems like they have annotated whether you're in a repetitive region or not in the training data. And they also describe down here, for the first 3 trillion tokens of pre-training, lowercase tokens are input to the model to add information on which portions of DNA are repetitive. So this is just their way of annotating whether you're in a repetitive region or not. So this was just a little bit about the loss function that was used, which is a pretty cool insight into how the model works and some of the thoughts the researchers have done while building out the Evo2 model. The researchers also described the hyperparameters used for both the 7 billion parameter model and also the 40 billion parameter model. And then they describe a pretty interesting fact, which is how they scaled the model up from the 8,000 nucleotide context window and all the way up to the 1 million nucleotide context window. Because you don't just take the model that is 8,000 nucleotides, trained for that, and then suddenly pass in 1 million nucleotides. You scale it up slowly and gradually until you have the desired context window you need. So down here, pre-training allowed the model to understand the basic DNA grammar, and since real genomes are much longer than the 8,000 nucleotide context, they gradually adapted the model to be able to process the amount of DNA that should be able to be processed at once. So each one of these rows is a different phase in the gradual scaling from the 8,000 nucleotide window and all the way up to 1 million nucleotide window up here. So you can see how they divide the different tokens, so 50 billion tokens per phase, into these different phases, and then scaled it all the way up to the 1 million context length window. And apart from this, they also mention a base frequency along with a scale factor, and these we'll cover in just a second down here. So down here in the report they mention, we explore 2 different rotary embedding based methods to adapt to longer sequences. Positional interpolation by scaling down the positional index of tokens and increasing the base frequency of the rope embedding. They then decide on a combined approach using both together with a 10x increase to the base frequency for every doubling of context length. And this table 4 up here then describes the parameters for these methods that they used. So this might sound a little bit like gibberish. It at least did to me first time I read it. So I looked into what rope is, so I can try and explain it to you as well. So first, what is even rope? So rope is an abbreviation for rotary positional embedding, and it all boils down to the model needing to be able to understand order. So the sequence ATGC is different from the sequence TCGA. So if you give the model a sequence, it needs to be able to understand the position of each DNA base and thereby keep the order, so it has to know that this order is different from this order, for example. So the first solution would to be just number the positions right here. So for example, if you have 1, 2, 3, 4, and 5, and 6, these would then just be different numbers for the different positions. So this would be 1, 2, 3, 4, etc. So these symbols over here, the square, circle, X, star, etc., represent the actual data tokens, so the specific nucleotides ATGC that exist at each numbered position in the sequence. So position 1 contains a square, and this would then be an A. So why use these symbols instead of ATGC? Well, the problem is that a model can't really learn what an A is, but it uses abstract symbols such as these, this is just an analogy, to represent what an A actually is. So these are just codes, just like the model would use codes within its internal states to represent these different ATGs and Cs. So in this example, the model would learn a code for position 1, it would learn another code for position 2, etc. And it doesn't automatically learn from these position codes that, for example, 5 and 6 are neighbors, or that 4 comes before 5. It just has to figure this out by seeing a lot of sequences, which isn't very efficient or effective, especially not for long sequences. So the first problem is that the model doesn't know that position 5 and position 6 are closer to each other than position 5 and position 1000, because there isn't any inbuilt relationship between these different positions. So the model learns a unique code for each position, number that it sees, but there's no built-in mathematical code that says that a star is related to a triangle. So the model also doesn't know that the star comes before the triangle, and therefore has to learn this from scratch. So let's say you train a model with maybe a context window of 10 tokens. This means that the model would learn 10 different positions inside its internal states. But then if you run inference with the model afterwards and you give it 6 tokens input, for example, then it hasn't really learned that a 6th position even exists, so it won't be able to handle this properly. So that is the problem with solution 1, which is numbering positions. And this brings us to the second solution, which was what was used in the report, and that is with rope, so rotational embeddings. So instead of these absolute positions up here, you can then encode the positions and also how they relate to each other with rotations. So up here, we don't encode how they relate to each other, just the positions. But with these rotations down here, which is another representation, what they try to do is just also encode how they do relate to each other. So with this rope method, each position gets a rotation by a specific angle m. So each one of these arrows is a vector. And this A would be a vector, for example, and then the T right after would also be a vector, the G would also be a vector, and the Z would also be a vector. And their position in the sequence is marked by their specific angle. So this one would be a specific position, this one is another position, and this one is another position. So the angle of this vector can be calculated from the position, and then you multiply that by an angle, so a specific angle like M. And this is an angle you define yourself. So let's say you have position 1 and you have position 2. Well, because position 2 is twice as large as position 1, then the angle would be twice as much because of this equation right here. And the relationship between the different arrows will then just be the difference between the angles right here. So going down, how does it store positional data? Well, the position is stored as the direction of the arrow, so the direction of the angle. So this just means where it points. And the relative distance between the different positions are then stored as the angle difference between the rotations of each arrow. So this is just the angle difference you see with red right here. So to give an example, if the first base has a 10 degrees rotation and the second base has a 20 degrees rotation, the difference is going to be actually 10 degrees. If you do rotate both arrows by 90 degrees, their relative difference will still be 10 degrees. Because there will still be an angle difference between these, even if you do rotate them around the axis. And because there are only 360 degrees around in a circle, they'll just keep wrapping around the axis and you'll never run out of positions. So unlike the simple numbering up here, Using this method inherently encodes relative distances. The model doesn't have to learn from scratch that 5 and 6 up here, for example, are neighbors. The small angle difference between their rotations tell us that directly. So a relationship between position 5 and 6 would be encoded the same way as the relationship between a position 1005 and 1006. So the reason that they wanted to use this method is that this is very useful for extending the context length, as we saw up here. So these base frequencies are actually the angles down here that we can multiply with. So the model can apply the same logic of comparing the angle differences even to positions far beyond what it saw in the initial training. So this makes it way better at handling longer sequences. So with that in mind, let's just talk about extrapolation. So the rope method handles position outside its original training data somewhat naturally. So because the position is encoded into, so the position right here is encoded into the rotation angle, as the position gets very large, so above 360 degrees, it just wraps around the circle like you see right here. So you see position 1, 2, 3, but then position 10 is obviously way larger, so that might wrap around a full 360 degrees and then even further all the way up until here. So it just wraps around this axis. So the model can still just calculate the relative angle differences. So the problem the researchers were trying to solve was that they wanted to be able to extend the context length of the model after already having trained it for pre-training. So again, if you go back, they explored 2 different rotary embedding based methods to adapt to longer sequences. The first one was positional interpolation by scaling down the positional index of tokens. The second one was increasing the base frequency. So let's talk about increasing the base frequency first. So there is a little problem when extending the context length. So when extending the context length from 8,000 nucleotides to 1 million, that is a context length growth of 125x. This means that the same position line has to fit in 125x as many positions, so the difference between the positions will also be 125x smaller. And this makes it harder for the model to recognize the difference between the positions. And therefore, you can adjust the base frequency up, so the model again knows where the base is again. So going all the way up here, we can see this is exactly what they did. They scale the base frequency up by a factor of 10 every time they up the context length as well. So you might think, why wouldn't they just take the base frequency of this massive context length right here and just use that both for pre-training and then all the way up through all the different phases? So the reason for that is that the 1 million token base angle isn't also used during the 8K pre-training because the difference between positions would be massive. So remember, this base frequency is what we multiply onto, let me just find the formula, is what we multiply onto the position. So let's say we have a very small context window, but a massive base frequency. Well, in that case, it would wrap around a lot of times before even reaching some of these first positions. So to prevent this wrapping around going crazy because you have that big of a base angle, you should scale up the base frequency as well when you scale up the context window. So let's say the base frequency is super large because you just used a massive base frequency for the 8K context window. Well, in that case, it might wrap around a couple of thousand degrees even for position 1. So this would make the relative distance quite confusing for the model because it is simply such large numbers all of a sudden. So that is why they increased the base frequency. They also talked about they talked about positional interpolation by scaling down the positional index of tokens. So let's just talk about what they did down here. Well, if the model has a context window of 1024, so if it's trained on these positions 0 to 1024, but it now needs to adapt to a double as or twice as large of a context window, so 2048. Well, now the model has to be able to handle twice as many positions. So within this range right here, there will be twice as many positions if you use the full context window that is now twice as large. So because the model was trained for 1024, these new 2048 positions can be scaled down by 0.5. So because you double the amount of positions, so you scale down the positions by a half to what it has been trained on. And then the unseen positions can then be interpolated. So let's say the model has seen these 4 positions, and that is what it was initially trained on, but now we want to increase it to more positions. Well, then we can interpolate the new positions by putting in new arrows between these arrows. So the model basically just needs to interpolate to find out what is between these arrows. So that is pretty much possible because we have the formula. We'll just put in the position and then also the base angle. And this way the model can interpolate the new positions that is between the old positions and thereby extend to a longer context window. So to summarize down here, the positional interpolation maps the new position range to the old one that the model has learned. So that was the first approach they used up here. And then the base frequency scaling adjusts the rate at which the angles change within that map range. So by preventing the angles from wrapping around 360 degrees too quickly, it maintains a unique rotational value for positions that are much further apart. So these are the two approaches the researchers used, and they use a mix of both. Great. So this gave you an insight into how the training was done with the model. So with that out the way, now we can go ahead and take a look at how we can actually use the model over here. So Evo2 is a model for genome modeling and design across all domains of life, as they wrote right here in the GitHub repository. What features does it actually have, and how can we use the model? Well, first of all, it was trained on 9.3 trillion base pairs, so a lot of data, and it has a context window of 1 million tokens. So we have a lot to work with here. And it also knows DNA from many species and not just human species, as we covered. So the first use case we're going to talk about is going to be generative tasks. So fundamentally, Evo2 is a generative model, and this means that we can use it to generate DNA sequences. So if you give it an input, which is a sequence of H, Gs, and Cs, so nucleotides, it will be able to predict the next nucleotides in the sequence, append them to the sequence, and then return that. That is also what we saw over here in the documentation under generation, where it is inputted a sequence of nucleotides, and then it returns the finished sequence with more appended nucleotides to the sequence. So the model is able to generate sequences matching what is found in nature. So up here, if you give it an input sequence and have it generate an output sequence, there isn't really any purpose to this generated sequence though. So down here, we can take a look at how the researchers tested its generated capabilities, and then use something called guided generation to help the researchers generate a sequence that they actually wanted. So let's just take a look at this excerpt from the report. So they say, we found that Evo2 can generate mitochondrial genomes with the correct number of coding sequences, tRNA, RNA genes when annotated with mitoset. So why mitochondria? Well, they are pretty small organisms and they can also easily be analyzed. So a BLAST-P analysis revealed that Evo2 created diverse mitochondrial genes with varying degrees of sequence identity to natural mitochondrial proteins. So they found that Evo2 can create mitochondrial genes that looks like how they do in real life. And then they say, notably, individual genes showed the highest homology to different organisms ranging from fish to mammals. So I wrote here, the model doesn't only generate genomes most similar to humans, but because it was trained on all these different organisms, it is able to generate diverse outputs while still sticking to biological characteristics. The researchers then say, the generated sequences also maintain proper synchrony. So what does synchrony even mean? Well, in the mitochondria generated, different genes appeared in the same relative order as in human mitochondrial genomes, so this shows that the model knows how to generate content of these sequences in the correct order too, and that is what they mean by proper synchrony. They additionally also write, while exhibiting considerable sequence diversification when compared to native sequences. So what they mean by this is that the model is able to generate sequences in the correct order, but still sequences that are diverse enough that it isn't just copying the native sequences. Lastly, they say, using AlphaFold 3 for protein multimer prediction, we found that the generated sequences form structures matching expected mitochondrial protein complex folds and interactions. So when taking amino acids from generated DNA and folding it, it will match the existing protein structures from naturally occurring mitochondrial protein in real life. And they actually put in a diagram down here of how much they look and match. So in real life, protein folds. So a protein, as we talked about earlier, consists of amino acids. And the sequence of these amino acids primarily determine how the protein actually folds. So if the order of amino acids is the same in the generated sequence, kind of as it is in the real life sequences, the protein should fold in the same way. So for this diagram in the report, we first of all see the annotation right here. So this is the name of the mitochondrial protein gene. The sequence ID percentage down here is how similar the Evo2 generated protein sequence is to the closest real protein found in a database. So obviously the higher this is, the more accurate it is when you compare it to existing proteins in the database. And what they want to show with this diagram right here is that Evo2 generated proteins are clearly super related to the real mitochondrial proteins since they are this alike with these high percentages. So the overall meaning of this diagram is that the EVO2 generated DNA sequences doesn't actually just look like genes, but they actually code for these proteins that, when folded with AI, for example, is predicted to have the correct 3D structures that also assemble correctly together in these larger complexes, as seen in real life as well. So it kind of suggests that EVO2 has learned some deeper functional patterns and constraints just beyond the sequence patterns that actually allows us to create these larger protein complex when folded. So hopping down here, there is another diagram from the report as well. So in this diagram, we see two different plots, one for each model. There are the 7 billion parameter model and the 40 billion parameter model. So in here, each dot represents one protein sequence generated by EVO2. They then looked through a database online or something to find the most similar matches to those proteins. So they used the generation feature up here to generate sequences that would produce proteins, and then they would match those proteins generated with actual real proteins from databases. So on the y-axis over here, this means how much of the generated protein's length matched up with the real protein, so that is the query cover over here, between 0 and 100%. So how this works is that you take the sequence that has been generated by EVO2 that produces this protein, and then you do what is called a blast search. So you use the protein sequence as the search query for the database, and the database then returns the best hit for what matches that DNA sequence the most. So the y-axis over here, so the query cover, means what percentage of the total length of the EVO2 generated protein, so the query, that was included in the alignment region with the best real protein match found. So for example, if an EVO2 protein was 100 amino acids long, the best alignment covered 80% of these amino acids. So this would mean that the query cover would then be 80%. And then for the y-axis down, also the x-axis down here, the sequence identity. This is how well or how similar the generated protein is to the closest real match. So where the y-axis says that maybe 80% of the generated protein is able to be aligned with the best match from the database, well, those 80% over here, down on the x-axis, well, how similar are they actually to the real database sequence? Because just because they align doesn't mean that they're 100% equal. So if there are some mutations between the generated sequence with EVO2 and then the reference sequence from the database, well, that is then what this sequence identity x-axis label means. And one thing to consider as well is that it isn't a bad thing that the dots aren't at 100% match, because that could just indicate model overfitting, so that the model has just been memorizing the sequences that is from the training data, and then it's just replicating those existing sequences, because the model can instead generate biological plausible sequences. So the goal isn't exactly to copy the genomes one-to-one, but to understand the rules of the genomes and then generate new, so novel sequences, but still plausibly. So instead of just remembering the sequences from the training data, it is able to learn the syntax of the sequences and then generate new sequences from that. So that might also be the reason that they aren't at exactly 100% for all of these proteins generated. So let's hop down to the next part. This is called guided generation. So this is about designing genomes for specific goals. So up here, as I talked about just a few minutes ago, you give it an input and then it predicts the next outputs that are most likely to be syntactically correct. But if you want to design sequences that have specific purposes, that is what you can do with guided generation. So you want to design genomes for specific goals. One of these goals could be chromatin accessibility. So to give you a little background about what this chromatin accessibility is, before actually designing for it down here, we can take a look at this explanation. So first of all, the DNA strings are really long, but they still need to be fit within a single cell. So since the DNA for the entire genome has to be stored within all the different cells, they need to be packed in a very tight and compact way to fit within those cells. So the cells need to pack the DNA very efficiently, so instead of just stuffing it randomly, they have to pack it very efficiently and nicely. So the DNA is wound around small proteins called histones, spools, and these spools are then wound and then packed tightly into complexes called chromatins. So over here in this image from Wikipedia, you see the DNA being wound around histones. So the histones are the big green balls, and these are then packed even more compact into these complexes down here called chromatins. But when you pack it this tightly, the problem is that if the genes within the DNA is packed so tightly, it might be inaccessible for being read. And if you can't even read the DNA, why even have it there to begin with? So the solution for this problem is that the cell unpacks only the chromatin structure around the needed gene, so this will then make that gene accessible. So to hop back to the chromatin accessibility definition, well, this means that how physically open and reachable this gene is within its packaged chromatin. So why is this chromatin accessibility important? Well, every cell stores the full genome, but cells in the brain won't need the same genes as cells in the skin, for example. So for example, a cell in the brain keeps brain genes easily accessible while packing the skin genes tightly away. So DNA can be designed so certain chromatins are more or less accessible depending on where you are in the body. So what the researchers then tried to do here is to guide this chromatin accessibility to regulate how genes are expressed in the DNA. So they call this generative epigenomics with inference time search. So epigenomics is the study of how chemical changes to the DNA and the associated proteins affect how the genes are turned on and off without altering the DNA sequence itself. So expressing certain genes more or less, for example, without actually removing the genes. So in the report they write, eukaryotic genomes regulate gene expression through a complex system of chemical and protein-mediated modifications, referred to as the epigenome. And then they say an important component of the epigenomic regulation involves modifying the openness and compactness of chromatin, which in turn controls which DNA regions can be accessed via transcriptional machinery. So in this diagram from the report, on the y-axis they have a metric for how open the chromatin is, so how accessible it is. And on the x-axis they have some metric for the location in the sequence, for example. So the black boxes is where they wanted the chromatins to be open, and the blue peaks are how open the DNA sequences turned out to be in those locations. And as you see from the diagram, it turns out that they were able to make the chromatin very accessible in the specific places that they wanted it to be, while keeping it inaccessible in the places they didn't want it to be accessible. So they could design for chromatin accessibility within a specific pattern, which is basically the idea behind the guided generation. And in the report they write, we therefore aim to develop an approach that uses Evo2 to generate DNA sequences for which we can specify both the location and the length of chromatin accessible regions. So that is what they did up here by specifying the locations and also the width of how these regions of chromatin accessibility should look. So when hopping up here to the generation example, where you just give it an input sequence and then you receive some output where it has appended some tokens to the input sequence, it might not be very obvious how they were able to actually generate the guidance, because it doesn't look like from the GitHub as well that they can actually give it a prompt that it has to generate certain chromatin accessibility features in certain locations, because that isn't really a feature of the model. So how they fixed this problem was by using other AI models as well and then coupling these. So there is two different components to being able to do this feature. One is the Evo2 model, and another component is these two AI models which they used in addition to the Evo2 model. So we have two AI models, Informer and Borzoi, and these are AI models that can predict chromatin accessibility profiles from DNA sequences, but they can't generate the sequences themselves. The Evo2 model is the second component to this process. This Evo2 model can generate sequences, but it can't apply conditions to these generations, like I just told you. So they won't be able to predict chromatin accessibility profiles, for example, and apply these constraints, but it will only be able to generate the sequences. So you can combine these two components and thereby generate a sequence and then check whether the generated sequence matches the desired output, and then reject or accept these generated sequences based on whether they fit into the constraints set by the Informer and Borzoi AI models. So how do you do this in real life then? So there are two components to this process, as I said before. First you have the Evo2 model, and then they have 4 different examples right here giving it the same input sequence. And because you can change the seed, for example, or have it generate different output sequences based on the same input sequence because it's a large language model, you can then take these output sequences, run it through the Informer and Borzoi AI models, and then these two AI models will then predict whether the chromatin is accessible within the desired region, and then accept whether to use this output sequence or not based on the output of these two models. So as you see in the first example right here, the first row, the researchers gave the model an input sequence, it then generated an output sequence, and then this output sequence then just happened to have a chromatin accessibility feature that matched with what the researchers desired. So therefore they accept this output sequence right here. If you then take the next output sequence that was generated from the same input sequence, it then didn't predict a chromatin accessibility feature matching what the researchers desired, so they rejected this output sequence. And they can then do this for the different output sequences generated by the Evo2 model. And this way you can guide the generation by rejecting or accepting different output sequences that matches the certain constraints that you have set before the generation process even begins. So this diagram right here, or at least the same process, is also described in the report. So the researchers say we use autoaggressive generation with evo2 to sample multiple 120 base pair chunks of DNA given the same genetic prompt. So they give the model the same genetic prompt, but you get different output sequences, and these output sequences will then be 128 nucleotides long. After doing this, they say we then use the informer and borzoi to predict how these chunks affect the chromatin accessibility profile of the generated sequence and score these profiles based on how well they match the desired peak patterns. So again, the black boxes right here is the desired patterns, and the blue patterns and the blue boxes you see right under is how the informer and borzoi algorithms or AI models actually predict that the generated sequence match these desired patterns. So this is basically the score. So this score would be pretty good because it matches the black box very well, and this score would be very poor because it doesn't match the black box very well. Then the researchers say chunks that have the most desirable accessibility profiles are retained by appending to the prompt, and generation proceeds to the next step, using a beam search algorithm to keep track of the best generated sequences. So to break it down even more, to just follow a step by step, they give the model a sequence. So this sequence right here, TCAGTA, they then generate 128 base pairs. Then they score the 128 base pairs through another model to see if they fit the criteria set up from the start. If they fit the criteria and the constraints, they then add this output sequence to the existing sequence and generate 128 base pairs more. If not, they regenerate the sequence and see if that fits even better. So that is how you can do guided generation with the evo2 model by using other AI models that can predict whether constraints are followed or not. The next feature apart from generation is prediction tasks. So hopping over here, we can take a look at scoring. So just using the normal forward, where you can score likelihoods across the DNA sequence. We have already talked about this pretty extensively. So this is just for spotting mutations in DNA, for example. So a small example right here is that just like some AI models can spot spam mails in your inbox, this can spot harmful mutations. And we have actually already talked pretty extensively about this up here with the mutation analysis, where we talked about how this mutation analysis was used to validate that the model has actually learned about start codons. So with these prediction tasks already pretty extensively covered, we can go ahead and just hop down to embeddings, which is the next feature. We covered embeddings pretty briefly, but I would like to go more in depth with what it is and what it can be used for. So embeddings is this third feature. So vectors that evo2 generates as part of processing DNA are what we refer to when we say embeddings. So hidden layers compress information down to numbers/vectors, and these can then be used to train other models. So in the example we used before as well is that evo2 might be able to capture more accurate features in its embeddings than other models are. Therefore, models using evo2's embeddings will be more precise because these models can then leverage evo2's internal representation of these inputs and then use these representation as inputs for these new models. So evo2 embeddings, for example, would reflect knowledge about codons more than raw data would in itself. So just to summarize what an embedding is quickly, if we have a neural network here, we have some input neurons, we have some output neurons, and we have the hidden layers. So we know what the input neurons or what the input data will look like, and we'll also know what the output data will look like. But inside these hidden layers right here, this will just be numbers that won't really make much sense to us as humans, because they'll just be numbers generated from weights and biases. So the embeddings in this case would then just be an entire layer's output. So if you just take the output of numbers from this entire hidden layer, which doesn't really make much sense to us as a human, we can then take all of these numbers and then use these numbers or these neurons' outputs as an input for a new neural network, which would then train. So that is the basic idea behind embeddings. So as explained earlier, you define your sequence and tokenize that. You then define the layer you would like to extract the embeddings from. So this layer would have a name, for example, and that is what they've done down here. They have described the layer to grab the output from, so blocks.28.mlp, etc. Then they just run a forward with the evo2 model and then grab the embeddings like this. So let's just go through an example of how this is used. So let's say we would like to train a model to predict if a genetic variant increases cancer risk. So for the training data, we'll need two different things. First, we'll need a non-mutated version of the gene, so we'll need the base pairs for a gene related to that cancer in a reference genome, and this reference genome should then therefore also be non-mutated. And then for a training sample, we can then take the blood sample from a person with a specific type of cancer related to this gene. So we sequence the DNA from that sample, find the base pairs for the cancer related to the gene, and then this will then contain the variant compared to the reference gene. So this up here is a sequence of how is this gene actually supposed to look with these nucleotides, and this down here is a person with cancer, where you measure the actual nucleotide sequence and then find out how those nucleotides differ from how it is supposed to look. And this way you can see maybe which nucleotide mutations cause this cancer up here. And with enough of these samples, you might be able to train a model to detect whether a person has cancer or not. So with this data right here, we can go ahead and extract the features from the Evo2 model. So step one is you run the reference gene through Evo2 and extract the embeddings at layer and then blocks 28, MLP, L3, or whatever they wrote right here, or just the layer that you find works the best. Secondly, you do the same just for the sample gene. So you extract the embeddings from the reference and then also for the sample of the person with cancer. So in this case, now the embeddings has been extracted from Evo2, and Evo2's knowledge can now be leveraged. With this training data now pre-processed as well, we can go ahead and train the model. So the features for this AI model, or the inputs, would be the reference embedding and also the sample embeddings, so these two right here. And the label or the output of the model would then be a true or false flag about whether the variant is pathogenic or not. So in real life, this would be some kind of score between 0 and 1, so between 0 and 100%. And if it's 1, then it's pathogenic. If it's 0, then it's non-pathogenic. So when training the model, this would obviously be 1 for the training data because the person has cancer. And if you give it a sample embedding where the person does not have cancer, this would then just be 0. So the alternative to using these embeddings would to be put in the actual sequences itself as raw inputs, so right here, and then you would give the reference sequence and then the sequence with the person who has from the person who has cancer. So maybe there's a mutation right here, and the model itself would then have to learn that this is a mutation, what a mutation is, and whether this is syntactically correct or not correct, and also how dangerous this even is. So this is all something that Evo2 has most likely learned and can then be leveraged by extracting these embeddings right here. So the benefit of using these embeddings is that since Evo2 has been trained on DNA, its embeddings will be richer and more informative, those are the keywords, than just using the raw base pairs, as I explained right here, as the model inputs. So there is really so many use cases for using Evo2 to build other AI models as well, for example. So it's really powerful to have models out and open source like this. So there is a lot of opportunity for building cool stuff with Evo2 and also the things that derive from Evo2. Now with the basic theory about Evo2, what it can do and how it works down, we can go ahead and take a look at how we can actually work with this on the code side, so on the tech side. Now running these large language models over here requires a lot of GPU memory, so you have to use some pretty hefty graphics cards. So we'll use serverless GPUs for this video. And for the previous SaaS products I've built, I've almost gone with serverless solutions just because they are partly cheaper, and also the developer experience is much nicer versus something like cloud platforms, so just normal Azure or AWS. So I reached out to Modal actually to see if they would sponsor this video. And the reason is that Modal has a great developer experience and also a very generous free tier. So if you hop into Modal and then AI, you should be able to find Modal right here and then just see their pricing. So you have about $30 worth of free credits every month. And then their most expensive graphics card right here is about $4 per hour. So you have a full hour of runtime, and remember this is serverless, so you really probably shouldn't run out of this from building this video. So this means you can do the entire video without paying anything basically, and still running some of the most powerful graphics cards in the world. So that is what we're going with for this video. So you usually have two approaches when working with these GPUs. The traditional approach is using AWS, for example, where you use something like an EC2 instance. This is a great approach if you want it to be always warm, but you'll also then have to pay for all the uptime that you need for your serverless or for your GPU. And this can get quite expensive if you don't use it constantly. So for a traditional approach like EC2, you'll need to pay per hour of instance uptime until terminated. You'll need to build the Docker image locally as well, push to ECR and then pull down to EC2. You'll need to start the instance, install packages when started, and also shut them down. And then there's also no GPU instance free tier. So if you hop into AWS, for example, and then I think it's called P5, their instances, these are the instances that have 8 H100 graphics cards. So they have H100, and the smallest one you can get has 8 H100s, and it's about $96 per hour as far as I know, which is crazily expensive. If you go into Modal instead, you can get one H100 for only $4 an hour, and you get it for free because you won't really use all that within a month anyways. So it's really a no-brainer to use Modal compared to AWS in this video, because we'll need a very huge graphics card to be able to run this model. So AWS does have Lambda, but that is only for serverless CPUs. But for AI, we actually are going to need the GPUs, so we can't really use AWS Lambda. So the usual workflow you also use with AWS is that you write some code. So you code the program, then you build a Docker image locally, and then you push that Docker image to ECR that stores the Docker images. Then you can take an EC2 instance, pull that Docker image down to the EC2 instance, and then from your computer you can then say, okay, now we start up this EC2 instance and then configure it and then run the Docker image. So there is significant overhead with the development process in itself. So when I say that the developer experience is very nice on some of these serverless GPU platforms like Modal, this is what I mean. So going down to a platform like Modal that is serverless, you get access to GPU serverless, so you don't have to pay for every hour of uptime, so you only pay per use. So this also means that everyone can follow along for free in the video, which is huge for me. And then a Docker container is automatically built by Modal when you push. You can then just pick different GPUs by switching out a string in the code, instead of having to start up a new EC2 instance. If you want to switch out the GPU, then you have to configure all this again. So start up and configure the instances, and this is just a huge mess, and it takes a lot of time and it's a lot of overhead. So you can then pick between different GPUs by switching the string in the code, as I said. So the local development experience is very nice, because you can just run a single command, do Modal run, and then it'll run in the cloud without you having to push it to a Docker container and then do it in the cloud. So deployment is also a single command. So up here, instead of having to code the program, build the Docker image, push it to ECR, and then download it to EC2, you code the program, then you say Modal run. The code is then pushed to Modal automatically, instead of you having to push a Docker image. Modal then automatically builds the Docker image and then runs your program with GPUs in the cloud. So all of these steps, which are massive overhead, are then completely avoided with Modal, which makes the developer experience much better in my opinion. And then when the program has been run, the output is just returned back to your local machine and also stored on the Modal website. So as I wrote down here, having to not upload the Docker container every time is a massive time saver and makes the developer experience so much nicer. And you'll see how much nicer it's going to be in this video when we actually do go ahead and run with Modal. So we can start with the repository evo2 by going ahead and cloning it down, and then they have a notebook about BRCA, so a specific gene for cancer, and then we can try and work with their notebook and just see how it works. After looking at their notebook, we'll just try and run this example on Modal as well, so in the cloud, and then we'll go ahead and build out our program from the knowledge we have learned from this notebook. So I'll just start by opening up Visual Studio Code right here, and then hopping under File, and then Open Folder. Then I'll hop into my Documents, Repositories, and then in here I'll create a new folder and call this Variant Analysis, and then Evo2. I'll then say Create. In here I'll just create a new folder called Backend, and just say Create, and then I'll open that up. Great. In here, I'll just go ahead and first of all create a new terminal, and then we can hop all the way back to evo2, the repository. If you haven't had it open, it's under Arc Institute evo2. What you can then do is scroll down and then find the following command: git clone -- recurse submodules, and paste it into your terminal. You'll then just put in a single space, and then we'll hop up to code and then just copy to clipboard, paste it in, and then press enter. This will clone the repository down along with all the repositories inside of it. So you see how it references to another repository right here called Vortex, which we talked about earlier as well. This is another repo, and that will need to be cloned down as well. And as far as I know, Vortex also contains other repositories in itself. So Cutlass, for example, and Transformer Engine. So all of these will be recursively cloned down to your computer, so you can actually go ahead and see what is inside of it. So right now it cloned down evo2, so you can see the evo2 folder, and you can also see all the code, and the Vortex folder is also populated, so it cloned it successfully. Now I'll just go ahead and say Shift Command and P, create a new Python environment, v. I'll use Python 3.12.3, and then just take all of these and just say OK. This will then create a new Python environment and install all the packages from the requirements files like this in all the recursive module as well. Now, while this is installing, I can just go ahead and go and search for Python. So if you wasn't able to see Python under the create Python environment menu right here, you should be able to just go ahead and install Python. So under Evo 2, they do say that you need Python 3.11 or more. So I'll just roll with Python 3.12. You can go ahead and test other Python versions if you want, but I'll just use 3.12 because it has worked for me. So go ahead and download that if you wasn't able to see your Python version. And then if you wasn't able to replicate this create environment menu right here, you might need to install some of the extensions I have as well. So I use AutoPip8, I use iSorts, Pylance, Python, Python Debugger, and then TensorBoard. These are the ones I have installed for Python specifically. Great. So now our environment should have been created along with the requirements installed from requirements.txt right here. So under the venv, you should be able to see all the libraries that was installed. So those should be successfully installed now at least. That is great. Good. So one thing you need to notice is that we won't really be able to run this entire model on our own computer. We'll need to run it in the cloud. So that is also why we'll use Modal specifically, because it lets us run this in the cloud so easily. So the reason we can see this is because on the requirements it says Evo 2 is based on striped Hyena 2, which requires Python 3.11, and it also uses the transformer engine Floating Point 8 for some layers, which requires an H100. So this is this massive graphics card, which is hugely expensive as well. So we won't be able to run this locally unless you have an H100, which you probably do not. So we'll actually need to run this with Modal anytime we want to test some of the code in here, just because it has such high requirements for our hardware. Great. So before actually running the code, let's just go ahead and check out one of their notebooks, which is an example of how they actually use Evo 2 in real life. So in this notebook right here, BRCA1, what they do is the example I talked to you about right here. So using this BRCA cancer dataset. Great. So in here, they run through with text and explanations how you can actually use the Evo 2 model. So I'll just close down this, and then we can go through how the model works before we actually try and run it ourselves. Cool. So what they say is that the human BRCA1 gene, so this stands for breast cancer 1, encodes for a protein that repairs damaged DNA. Certain variants of this gene has been associated with an increased risk of breast and ovarian cancers. Using Evo 2, we can predict whether a particular single nucleotide variant, so a mutation in a single ATGNC or C of the BRCA gene, is likely to be harmful to the protein's function, and thus potentially increase the risk of cancer for the patient with a genetic variant. So this gene right here repairs DNA that has damage, and damaged DNA is what causes cancer. So if this gene isn't able to repair the DNA because of a mutation, then you'll be more likely to have cancer. So the idea is to look for mutations in this DNA, in this specific gene, and then see if those cause cancer. So first they say we start by loading a dataset from Findlay, which contains experimentally measured function scores of 3,800 single nucleotide variants. A single nucleotide variant is just a single mutation in an ATG or C, so this would be an SNV, for example. So this would be a SNV from C to A. Good. Then they say these function scores reflect the extent by which the genetic variant has disrupted the protein's function, with lower scores indicating greater disruption. So in this dataset, the SNVs are classified into 3 categories based on their function scores: LOF, loss of function; ENC, intermediate; or FUNC, functional. We start by reading in this dataset. So first they just installed some pip packages, then import some of these packages they just installed. This gave them this output, doesn't really matter that much. We can then just check out the photo structure again. So you see that there is this Excel file right here. So this Excel file contains some of the variants. So they read this Excel file, which is the following one right here, and then they print out the first 10 rows of this data frame. And that gave them the following output. So hopping over to our ArcGL, we can just go ahead and go a little bit more in depth with what this actually means. So each one of these rows is a single nucleotide variant. So a single nucleotide variant has a location. So it has a location on a chromosome. And then the specific location on that chromosome is called the position. HG19 is a human genome, so that is just a reference genome like we talked about earlier in the theory. So the reference is what it was before the mutation, so what it is supposed to be, and the alt is what it is after the mutation, so what it has been mutated to. And then there is the class over here that could be func, int, or loss of function. So loss of function is severely disrupted function. So that basically breaks the gene and makes the body not able to repair the damaged DNA, giving you a much higher chance of cancer. INT, so intermediate, is moderately disrupted function, so these partially impair the gene. And then functional means that you have normal or near normal function, so this mutation doesn't really have that big of an impact on the BRCA1 gene. And these labels right here have been determined from the score mean. And what does these scores actually mean? So the function score is a measure of how well the BRCA1 protein maintains its DNA repair function when a specific variant is present. So these lower scores, as you see right here, so more negative values indicate more disrupted function, so the variant severely impairs the BRCA1 ability to repair the DNA damage, and the higher scores, so closer to or above 0, indicate preserved function. This means that the variant has little impact on BRCA's repair function. So this also means that when you see INT right here, this moderately disrupts the function of the gene, but you also see a very low score, whereas when you see all these high scores, those are more correlated to FUNC, so functional. So the low scores means that there is a higher chance of the label being INT. So that is what you should be able to interpret from these 10 SNVs that has been printed out by the researchers that made this notebook. What they then do is then just group func and int into one group called func int. So instead of having 3 different groups, they have 2 groups. They have one called loss of function, that is a mutation that actually breaks the gene, and then they have one that is both benign and also moderately disrupted function. So they group these 2 into one and then just generalize or abstract those 2 differences away. So that is what they do with this code right here. So first of all, they take these columns up here, they take that and then just rename it to these other names. So chromosomes become chrome, position HD19 becomes pos, for example. So they rename it, and then they take these 2 groups and then just group those into the same one called func int. And the result of this is, of course, that all these rows that would be either func or int will then become func int, so this general class right here. What they then say is that we build a function to pass the reference invariant sequences of 8,000 base pairs window around the genomic position of each single nucleotide variant using the reference sequence of a human chromosome 17 where the BRCA1 gene is located. So this may sound a little bit intimidating, but let's just hop over to the Excalidraw and then just take a look at how they actually go ahead and do this. So they run a variant mutation, just like we talked about over here, where you take a sequence before and after the mutation, and then take a look at the difference in the score. So this difference in the score, which makes this color lighter or darker, is the exact same score as was talked about over here in this function score mean. So it's not much harder than that, basically. So let's say you have a reference sequence that looks like this. Just imagine this to be 8,000 base pairs long, as they wrote right here. So that is our window. Then we would like to introduce a variant. So for example, if the position right here corresponds to this position right there, then we can see that it is a variant. So let's say this was the variant A. So instead of this A, we just turn this into a T. So now we have a reference sequence and we also have a sequence with a variant within it. So hopping back over here, we can see how they do this. They define the window size, and then they open up this other file called fna.gz, so just a zipped file basically. And then this file contains the sequence for this chromosome with this chromosome right here. So this contains this entire sequence of single nucleotide variants. So this is the sequence of single nucleotide variants, which is just one long string from record.sequence. They then define a function called parseSequences, and this will return a reference sequence and a variant sequence. So these are the 2 sequences I talked about. This one and then another one that is the variant sequence where it has been changed from an A to an C. So these are the 2 sequences that are returned from this function right here. So you put in a position and then you put in the ref and then an alt. So the position would, for example, be this number right here. The reference would be the A, and then the alt would be the T. So that is the mutation you induce to the reference sequence. Great. So this position over here isn't 0 indexed. So the first thing they do is just to subtract 1 from that, since they want to convert it to a 0 indexed position. They then define this new variable called full sequence, which is equal to the full sequence for the entire chromosome. So many more single nucleotide variants than we actually do need. And then they cut away all the parts of the sequence that isn't within this window, and within this window, this position should be right in the middle. So that is what they're going for. So let's say this entire sequence right here is the full sequence. This full sequence right there. They then calculate these two variables, ref sequence start end and start. So these are based on the window. So the start of the sequence that contains both the variant and also not the variant, so the sequence we would like to return, are the following ones right here. So let's say this is the entire sequence, and then the window is maybe only 10 single nucleotide variants. Well, that would mean that you would have about 5 single nucleotide variants to the right, and then 5 to the left, and then you would have this one in the middle, and then everything else from this entire sequence is then just cut away. So that is what I've been trying to illustrate with the following illustration. So everything outside this sliding window is cut away. So everything outside these 8,000 nucleotide variants, nucleotides, are just cut away. So this is just a number for where to start the sequence, and this is just a number for where to end the sequence. And ref is then just A right here in the middle, so what the mutation is before the mutation. So this would then be the reference sequence, and this down here would then be the mutated sequence. So you just take the position from ref sequence start, all the way up until the mutation, and then you just insert the variant instead of the reference, and then you take from reference and then all the way over to ref sequence end, and then append that to this as well. So now you have the reference and then the mutated sequence as well. So that is what I've been trying to illustrate down here as well. Again, you just cut these long sequences away outside the window. That is what is done right here. But then instead of changing the variant or changing the reference to the variant like that is done right here, you just cut away this, and then you are just left with the original sequence with the reference being right in the middle. So hopping back into the code, that is what is done here. You have the reference sequence start, reference sequence end. So it can start in a position less than 0. So that is why you say max right here. The position right there is then the position of the reference right here. Then you subtract half of the window. So that is what you subtract right here. So then you'll get the ref sequence start. So all this will be cut away. So that is why you calculate it this way. For the reference sequence end, you do the same. You can't have a sequence a sequence end that is longer than the full sequence length. So that is why you put in a min. Then you can calculate the sequence end from position dot and then position plus and then half of the window size. So that will give you the following position right there. You can then take the full sequence and then just cut away all of the nucleotides. So you cut away all of this and all of this based on these two positions, and that is what has been done right here. And the variant position is then redefined to be the minimum between the position and then half of the window size. So this will give us the position of the single nucleotide variant within this new reference sequence. And now they create the mutated sequence. So they take this half of this sequence, then they put in the alternative, then they take the rest of the sequence, and then just append those together. So that is what happens right here. They take the first part, this one, then they take the variant, and then they take the last part, and they append those together to get the mutated sequence. Then they do what is called sanity checks, so they check whether these things are correct or not, and if they are not correct, then something is obviously very wrong, so they want to throw an error in some way. So first of all, the variant sequence and the reference sequence should obviously be the same length, because otherwise something is very wrong, so this should be the same length as this one right here. Then they would like to check that this position corresponds to a letter that is ref, and this position right here in the variant sequence corresponds to this alternative letter, so the mutation. So they just check that this is actually a T, and then they check that this is actually an A, and then they can make sure that everything is actually correct. Then what they do is just take the first row right here, so this position from a T to a G, and then they take that single row, save in this row variable, then they take the position, the reference, and the variance, and then they pass this into this new method they just created. Then they can print the information of the row as well as the sequence, or at least part of the sequence, before and after introducing the variant. So up here, they change from an A to a T. So down here, there should be a difference between an A and a T somewhere in the sequence. Oh, sorry, I can see that it is a T and a G. So there should be a change from a T to a G. So down here, you can see that there is a T right there in the middle, and there is a G right there. So this means that the mutation has then been introduced in the variant sequence down here. Then the researchers write, then we load the Evo2 1 billion parameter model and score the likelihoods of the reference and variant sequences of each single nucleotide variant. And then they say they strongly recommend using the larger models and they only use the small ones because it's quicker. So they import the Evo2 model. So this is imported from evo2.evo2. So evo2 right here, and then models. And then in here there is a class called evo2 that can be used to run inference with evo2. Good, so hopping back in here, what they then do is just load the evo2 1 billion parameter base model. So this will then download the model automatically from Hugging Face if not already in the computer system in the Hugging Face cache. Otherwise it'll load it from the Hugging Face cache locally. And this will then also load the model into the GPU memory so you can actually run inference with it. The next thing to do is to build out a list of variant sequences along with a list of reference sequences. So this comes down to looping through this entire list of all these different variants and then using this parse sequences method to create a reference sequence and a variant sequence, and then adding these two sequences to these lists just for each single nucleotide variant. So they loop through all of these different rows in the data frame, and then they get the reference sequence and variant sequence with this parse sequence method, and then they do some deduplication right here because the same reference sequence might have two variants in the same place, so you don't have to score the reference sequence twice. After doing this, they have a method called score sequences that can be used to score all of these sequences by running inference on all of them. But first, let's just take a look at how this deduplication actually works. So if I scroll down, for each variant, the sequence is passed to get the reference sequence. The dataset of variants might contain multiple entries of variants for the same position, so that is the issue. And because inference is run for every single reference sequence and also for every single variant sequence, you might score the same sequence twice or even multiple times, and that is just a waste of processing because the result is just going to be the same in both cases. So over here I kind of describe what goes on in this code, and it's a little bit complex, but let me just walk you through it. So we process two different variants, both in position 100, and then we see how this system acts. So when processing the first variant right here, this reference sequences list get the first unique reference sequence added. This dictionary right here then maps the unique sequence to index 0. And reference sequence indexes then stores index 0 because this first variant uses the reference sequence now stored at index 0. And the variant sequences then gets the variant sequence added to that array. So this will happen regardless of what happens because two mutations won't be the same. So when processing another variant down here, also at position 100, then this kind of caching logic kicks in. So this reference sequences array does not change because the reference sequence is already present. The reference sequence to index does also not change because the key, so the reference sequence already exists. The reference sequence indexes gets another 0 appended because this second variant also that is also analyzed also uses the reference sequence stored at index 0 up here. So now this becomes 0, 0. And then again, this of course changes because another variant sequence is added to this variant sequence array. So instead of scoring this reference sequence right here twice by adding it twice to the list, it can be added once and then also scored once. And then by tracking these indexes, we can know that both of these variants should just use the result of this score right here. And this way we'll only need to do the scoring for this reference sequence once. And then down here we can see how it actually did this caching process. So it actually reduced the number of reference sequence inference calls by 2/3. So evo2 had to score 3,800 variant sequences, but only 1,326 reference sequences. So that is quite a lot of requests to the evo2 model, which will save a bunch of compute. And after having built these lists of all these reference and variant sequences, the researchers went ahead and scored those. So that is done right here. And these variables will then be lists of scores for each variant. So index 0 will be the reference score in this variable for the first SNV. The index 0 here will be the score for the first SNV just for the variant sequence. And down here, these two lists can then be subtracted. So the first variant score, the first reference score is subtracted from the first variant score. The second reference score is also subtracted from the second variant score, etc. And then all the way through the list. So this will give us the differences between the scores from the variants and references just for the entire list at once. And this will then be added as a column in the original data frame or original grid right here. So that is what you can see over here. And then the researchers print out the first 10 records, and that is the printout. So hopping over to the CalDraw, let's talk about what this scoring sequences actually means. So this score that is being calculated right here, score sequences, Well, the score represents the syntactical correctness of the sequence, so more harmful mutations will change the score more than less harmful mutations. So the worse the variants actually are, the bigger the difference in the scores will be. So that is how you are supposed to understand that. So to summarize that, reference sequence score minus mutated sequence score is equal to how big of a difference the mutation actually makes. And the last part is then comparing these scores. So the researchers went ahead and created these two diagrams down here. So let me just walk through what they mean. So this diagram has two different categories. It has variations with harmful mutations and variations that are likely benign, so not harmful. So each one of these rows with the delta score and the ref and alt, class etc. is then plotted right here. So on the y-axis there are two categories. There are the func in or int, meaning they are likely benign, and then right here in the second category it says loss of function. So these are all the different rows that has loss of function as the class. And then the first one again is just all the different rows that has func or int as the class. This way the researchers can check whether the general or average scores are lower for the mutation with loss of function than they are for the benign mutations, so the less harmful ones. So by plotting all of these different mutations along with their scores and then tracking the median or calculating the median, you can see that the median is a little bit lower for the mutations with loss of functions than they are for the mutations that is benign. And what this means is that EVA2 is able to predict that mutations that actually cause a loss of function in the BRCA1 gene are more pathogenic or more harmful than they are if they are just benign up here. So let's try out this example in Modal so we can just run it ourselves instead of having to rely on the researchers' outputs. So I'll just go ahead and close this down. And then inside my repo, I'll just say open my integrated terminal. And then I'll say pip install Modal, and just let Modal install. Then I can say Modal and then setup. And this should then just open up the browser. So I'll just copy this link right here and hop into the browser, open a new tab and paste that in. Now I'll just go ahead and say continue with GitHub and then just log in. Alright, so I just logged in and now we're able to create a token. So I'll just go ahead and say authorize. And now it should be redirected in a few seconds, it says. And I was redirected over here to the dashboard. Great. So hopping back, we should be able to see that we were successfully logged in. And then I'm just going to go ahead and test that it actually works. So if we hop into documentation right here, I'll just open that up and then say guides. Maybe there's an introduction. So down here there is hello world. So we just ran modal setup. Now we can just create a new file in here. I'll just go ahead and call that main.py. So I'll just import in this code right here. It'll import the system, create a new modal app, and then we'll just copy in this function as well. This will create a function that can run remotely or locally. Then I'll just copy in this main function. This main function is going to be the one that runs, and that will then call this function up here. It'll call it both locally and also remote. So when you say function.local, this will run on the system you're currently on. When you say .remote, it'll then run on the Modal cloud, and you can then specify what graphics card should it run on, for example. So this means you can run GPU heavy stuff on a GPU function only, while running other stuff on your local system if you don't want to use the GPU. Cool, so I'll just go ahead and save this and then say Modal and then run main.py and just see what happens. So it's running something right now. Great, and stop the app. Good, so let's just scroll all the way up and see what happened. So it initialized and then it created a run, and then the blue runs right here are the runs that run locally, that run in the cloud, and the white ones right here are then the ones that run locally. If I then just hop back into the Modal dashboard right here, refresh, we should be able to see a run. So I'll refresh, stopped apps maybe, example hello world right there, and then we can see the run as well as the logs. So it says world, and down here we say hello 1000. Cool. So now we at least know we are connected to Modal and are able to run the code. So that is perfect. So I'll start out by setting up the Docker image that is supposed to contain the code. So we don't have to build the Docker image itself, but we need to define just what it is supposed to contain. So hopping in here to GitHub, we can see that there is this setup guide. So you need to clone down the repo, then cd into it, and then say pip install dot. So pip install dot will run this setup.py file right here, which will then do a bunch of setup for you. This most likely won't run on your own computer, so I don't really do this. The only reason I cloned down the repo is so I can look at the code and then import stuff I do need, and then everything I'll run, I'll just run in the cloud with Modal. So as you see, there is a setup.py file right here, which will run a bunch of code, and then inside Vortex there is another setup.py which will run some other code. Inside operations there are these other repositories as well, which will also contain setup files. So there is a bunch of code that is going to need to run. And some of this code from NVIDIA will, for example, need a C and a C++ compiler. So some of these things we'll need to add when we build the Docker image to make the requirements available for Evo 2 such that it can actually run in the cloud. So first I would like to just define the image. So I'll say Evo 2 image equals, and then I'll say modal.image. And this will then be dot from registry. I'll then like to use the NVIDIA CUDA and then 12.4.0 and then development Ubuntu 22.04, and then I can say add Python, and then the Python version is going to be 3.12, just like our own local environment. So when this Docker image is being built, it will start from this base image right here, which is based on Ubuntu with support for NVIDIA GPUs with CUDA. So when the image is being built, we'll need to do some apt installs as well. So we'll need build essential, we'll need cmake, we'll need ninja build. We'll also need lib cut cnn 8, and then we'll also need libcut cnn 8 def, like that. Then we'll need git, we'll then also need gcc, and then g++. I'll just save that. So these right here, those I just marked, are essential compilation tools. So many high-performance Python libraries used in artificial intelligence, so the ones used inside Vortex, have parts written in C++ or CUDA that needs to be compiled during installation. So that is what is being done inside these setup files. So these packages right here provide the necessary compilers, so GCC and G++, and also the build systems such as Ninja and CMake to be able to do that. And these two right here provide necessary functions for deep learning functions. So these are the NVIDIA QDNN library. And this is also needed to compile code that uses QDNN. So how might you find out that these are the ones you actually need to install to be able to run Evo 2? Well, if you just try and run Evo 2 and then you do not include these, well then you'll get some errors, and then what you'll do is you'll search online for the errors, and then somebody will say, oh, if you want to use Transformer Engine or you want to use Vortex, for example, then you'll need to add these libraries. And then when you do add these libraries to the Docker image, then suddenly it'll work. So you find out you need to install all these packages just by trying to run it down here. Let me just go back. Just by trying to run this right here on Modal, for example. And then when it does install with just a base image and the requirements installed maybe, but not any of these installed packages, it'll just throw some error you have never heard of before. And then when you search for it, you'll find out that you need to install, oh, maybe you need this, maybe you need this. And you figure that out just from trying to run the code, and then you iterate on that. We also need to set an environment variable within the system. So we'll say environment, and then the name of the environment variable should be cc. This will point to user bin gcc. Then we also need to set cxx. This will point to user bin and then g++. So after having installed these libraries up here, or these packages, we also need to set these environment variables that explicitly tell the build tools which C and C++ compilers to use. And now with these prerequisites done that are needed to actually run these setup.py files, we can go ahead and run this code that actually clones the repository down into our Docker image. And after that, then install the requirements.txt files. So first I'll say .run commands. Inside run commands, I'll put in git clone -- recurse submodules. And that is because we have the Vortex repository inside the Evo2 repository, so we need to also fetch this one since it is needed by Evo2. And it doesn't do that automatically, so that's why we put in this recurse submodules. Good. So hopping up here, copying the HTTPS location, we can just paste that in. And then we need to say and cd evo2, so that is being run sequentially after this command right here. And then after cd evo2, we need to say pip install dot to run the setup.py files, like this. And then one error I've previously gotten when installing evo2 into a Docker container is the following. So I'll just hop under issues, and then just remove these and search for key error. And then I'll say recipe, just search for that. And then I can hop down here. And then you see other users have had the same error with the key error recipe, just a error that doesn't really make much sense. Scrolling down, I found that you can pip uninstall transformer engine, and then you can say pip install and then install a specific version, just like this person right here did. So to just avoid getting this error, because I've gotten it before, we'll just go ahead and run these commands right here as part of the building Docker image. So I'll just hop back here and then say dot run commands, and I'll then run pip uninstall -y, so just -yes if there is anything that requires us saying yes. Then I'll say transformer, and then I'll say engine, as well as transformer underscore engine, like that. So this will uninstall the transformer engine script, a package. Then I'll say dot run commands again, and then I'll reinstall it in the version we would like to. So I'll say pip install, and then you can say put in two single quotation marks like this, and then I'll say transformer engine, like that. And then I'll say PyTorch equals 1.13. And then you can say -- no build isolation, like that. And then lastly, because we are running this example inside the notebooks BICA1, so in here, the first step that the researchers did was to install the following dependencies. So we'll just make sure to have those installed in the Docker container as well. So let me just copy these dependencies, close this down, and then create a new file in here called requirements.txt. I'll paste in Modal first of all, and then I'll put in matplotlib, pandas, seaborn, scikit-learn, and then open PyExcel. I'll just press save, and then we can hop back into main.py and just run a command that also installs those. So we'll say pip install from requirements, so this will install requirements from a file. Then we'll paste in requirements.txt and just press save. So we'll just name our app now. We'll say variant analysis and then evo2. Then we'll pass in the image, so we'll say image equals, and this is then evo2 image. And then we can just create a new function, so I'll say def test, and then this will just print out testing, and then this will be defined as an app.function, and I would like this to run on a graphics card, so the GPU should be H100, like this. So this is one of the available graphics card, and this is actually the one that is needed by evo2. So hopping in here, searching for H100, we can see that this requires an H100 graphics card, or some graphics card with compute capability above 8.9. We'll just use the H100 because it is available on Modal. Then what I'll do is just, let me just remove this function right here. What I'll then do is just delete all the contents of the main function, and then say test.remote to run it on this graphics card, and then I can just press save. This should then build this entire Docker image. This will only have to build for a long time the first time, and then this entire Docker image should be cached such that only the code that will change, it changes every time. So when running this for the first time, it'll take a long time, maybe 10 minutes, because it needs to build all of these things, and then it'll run the code afterwards. Then when we run the code again, it'll only have to print this code out because this has already been built. And because this is building a Docker container and actually, a Docker image and actually works like a Docker image, this will also just cache from the top statements just like a Docker image does. So right here, when the base image has been chosen, this will then be cached, and then the apt installs will be cached after that. the setting of the environment variables after that. So this means that if you run this once and then you change this apt install command, for example, you add another dependency, then it'll have to redo all of the commands after apt install along with apt install. If you change out the environment variables, then it will have cached until here, and it'll need to redo the environment variables along with all commands after those. So the things that change the least should be at the top of this Docker container definition. So that is what has been done right here. So just to summarize, a base image is set, so the operating system for the Docker image is set. The packages for being able to run the setup files has been installed right here, so the C++ compilers. They are made references to where these compiler files are at. Then the evo2 repository is downloaded straight down or cloned straight down to the Docker image along with the pip install command. This will install the setup files and compile the necessary packages required to run evo2. Then to avoid this key around a recipe, the following commands are run that will uninstall the transformer engine as well as reinstall it in the version we would like to. And lastly, the requirements.txt will be installed right here, such that we are ready to run the code from the notebook whenever this has been built. And when all of this has run, we should see the printout testing because the code will run after all of this. So to test this out, we can say modal, and then we can say run main.py, and then press enter. And this should then run all of this in the cloud, and this will then take a while. So I'll come back when this is finished. So this finished after just a couple of seconds, and it said testing already. So it seems like it didn't build all of these layers in the Docker image. It just cached the Docker image that was used last time. So what we can do is just to force the rebuild of the entire Docker image. So up here, I'll say force build equals true, and then just run this entire command. This will force this cache to be invalidated from here on and all the way down. So this will rebuild the entire Docker image. So I'll just go ahead and say modal run main.py and just let this run again. And then as soon as this is done running in maybe 10-15 minutes, we can go ahead and remove this force build equals true. And then this should definitely, because you need to remove this afterwards, otherwise it'll rebuild the entire Docker image every time. So I'll just wait for this to finish and then I'll come back to you. Alright, so my build just finished and this took about 15 to 20 minutes. So as you see, it ran through all of these different steps up here, a lot of downloading, and then it went all the way down and then it ran the code at last with the output we were expecting. So now to just test out that the caching actually works, we can go ahead and try and run this again. So we just need to remove force build equals true, of course, just so we don't have to wait 20 minutes again. And then press enter. And now we see that it loads the image and it runs the code, but it didn't have to go through all of these steps again. So that is the power of caching, like we did right here. This also means that just don't change these things for fun. Like don't just go and add one of these or remove one of these for fun unless you want to wait for 20 minutes. So only change these things if you really have to do it. Otherwise you'll need to wait maybe 20 minutes again until your Docker image builds. Good, so I'll just go ahead and hide my terminal here. And then we can try and port the functionality of the notebook over into our main.py and then try and run evo2 on Modal in the cloud. I will start by creating a function that won't run on a GPU. This function will be called brca1 example. In this function, I'll say print running brca1 variant analysis with evo2, something like that. Great. And then I can say brca1 example dot, and I'll just run that locally. So the computation for scoring the reference and variant sequences should be done on a GPU in the cloud, but obviously we can't really show a plot in the cloud like the plot that was shown right here. So if you scroll all the way down, this plot obviously can't be shown in the cloud. We want to show this locally. So I'll just create this to, let me say, run inference, and then show plot from returned data. That is kind of what I want to do. And then down here, I should be able to just try and run this. So I'll say modal run main.py, and this should then just print out running brca1 variant analysis. And this is in white text, which means it ran locally. We'll then just create another function for lifting the heavy load. This will be called run brca1 and then analysis. And this function should be run on a GPU So I'll say GPU, and this will be H100, like that. I can then say volumes, and I would like to define a volume quickly. So I'll say mount path, and then I'll say volume, and we just need to define these two things further up. So I'll hop up to right under evo2 image, and then I'll define a volume. So I'll say volume equals modal dot volume, and then I'll say dot from name, and I'll say hf cache. That is what we will call our volume, and then create if missing true. So if it doesn't exist, it'll just create that, and I'll then define a string called mount path, which will be equal to root. So from root, I'll say root, and then dot cache dot hugging face, like this. So this points to the place in the Ubuntu file system where Hugging Face will download its models to and then store those in a cache folder. So inside this evo2 code under under models, this is the class that loads the evo2 model. So whenever you create a new instance of this class, what it'll do in the constructor, so the init, will be to download this model from Hugging Face. So if you hop over here, they then provide these model checkpoints that are hosted on Hugging Face. So when you instantiate a new instance of this model, it'll download these model files automatically, such that you don't have to do it manually. So this is what it'll do right here. Then it'll hop in and otherwise download these models from Hugging Face. So to avoid the model being downloaded every single time we run our script, we'll attach a volume to our modal container. So a volume is kind of an external file system which we can attach to our Docker container. So it's kind of like plugging an external hard drive into your system. And this volume will be mounted to the following path. And this path just happens to be the path that that evo2 and Hugging Face downloads to whenever they download the model file for you automatically. So what also happens inside this models.py file is that if evo2 can't find the model for you and is already in your file system, then it obviously won't download the model for you again. So that is why this folder is called cache. So first time you run this function and instantiate the evo2 class, it should then store or download the file down to your local system and place it in this folder right here. It does that automatically. Then because the volume is attached to this specific folder, Modal will handle uploading the model file to Modal and storing it in a file system for you. You don't have to do anything like committing files. And then next time you run the function again, because the volume is still attached to the same place when you run the Docker container again, evo2 won't have to download the entire file again. It'll just take the file that was already downloaded and placed in the volume. So this just caches the model file for us and prevents it being downloaded every single time. So that is why we use a volume. So now we can define that this volume should be attached when this function specifically is ran. Then we can also set a timeout. We'll set the timeout to 1000 seconds. This means that it'll be able to run for about 15 minutes before the function shuts down automatically. And remember, with serverless solutions like Modal, for example, you will be built by second ran. So if you just search for Modal AI and then go to pricing, then for serverless, you'll be built by second ran. So if you say second, for example, it costs this much of your credits per second, and then per hour, it'll amount to something like this. So if you run without any breaks for an hour, it'll cost you about $4. So that is quite a lot of runtime. So let's just hop back and try and get this to work. So the first thing we'll need to do is just call this function. So run inference, I'll say run brca analysis, and then this probably returns something. So I'll say returns, and then I'll go ahead and say .remote. So this function will be run on a local system, and this function up here will be run in the Modal cloud with this GPU available. This way you will also only be built for this function right here, and this function down here will then run locally and won't be built for that. So this function run won't be as expensive as this up here because this has a GPU available and this down here does not. Cool, so let's just see what we need to import to get this example up and running. So first of all, if I just remove that, we'll need to import all of these packages right there. So we'll just go ahead and do that. So I'll just indent that. And then because we added these requirements down here, you can see with these three dots, they aren't really installed. And that is also why we see these yellow squiggly lines. So opening back up the terminal, I can just say pip install and then -r requirements.txt. And yeah, I'll just install matplotlib, which is defined right here, as well as these other packages that are also required. and now they just resolved. So the next thing we need to do is just find out what else happens in here. So we can at least start by defining the Evo2 model, make sure that is ready. We can define the window size. So the Evo2 model should be down here somewhere, right there. Cool. So we can start by saying, we'll go down, and then we'll say from Evo2, import Evo2. Then we'll also define the window size, so that was up here. This is just a constant that should be defined in the start of the script. Then we can say print loading Evo2 model. We can then say model equals Evo2, and then we'll pass in the model we would like to work with. So that is going to be Evo2 and then 7 billion parameters. So this is one of the models available. So hopping into the Evo2 repo, these are the models we have available. This one will require multiple H100 graphics cards. So we'll just use this one for now, and then we can test this out later if we would like to. So I'll hop back, paste this in, and then I'll say print Evo2 model loaded. Now the next step is loading the BRCA1 dataset from the Excel file, so containing all the nucleotide variants. So hopping up here, this is about what should be copied in. So I'll grab those lines, paste them in right here. And I just see a small error. There's a yellow squiggly line here. So we just need to put in Evo2 right there instead. And remember, this code will be ran inside this Docker container. So we'll need to take account for the new file paths that might not match up to the file paths from this research notebook. So we'll need to hop in here and then just change this file path. So we'll call this, I'll just go ahead and remove this join path. So we just have a hard-coded path, and we'll then say Evo2, notebooks, BRCA1, and then into this file right here. So this corresponds to Evo2, so it'll start inside this folder, Evo2, and then notebooks, BRCA1, and then the Excel file. And this we'll just leave alone. This head is just a printout, so we'll just remove that. Hop down and find the next part that is supposed to be copied. So these ones right here, the renaming as well as the converting to the 2-class system, we can paste in right there. And then after loading in all the variants, we should be able to load in the reference genome right here. So I'll just take this line of code, paste it in, and this should then be indented. And then the same way we hard-coded the path right here, we'll just do the exact same instead of the join here as well. So I'll remove that and just add this to the path instead. So this just points to the same path as up here, but instead of the Excel file, it points to this gset file right here. And after that, what they do is create this parse sequence function, and then they use this function inside a loop. So I'll just take all of this right now and just paste that right in. So I'll just skip the parse function and indent this correctly right there and right there. And then instead of this parse sequence function, I'll just take the code and just copy that straight in. So I'll take this, copy it, and then right here, I'll just replace that. So it looks something like this instead. I'll just need to remove a single indent and then remove this return statement as well. I'll just go ahead and remove the sanity checks. I assume it works because we tested it. Then I'll say row and just index for the key instead of having variables. And this will be alt. And then I think there is one more, or the alternative, so the variant and the position might actually be enough. So I'll just go ahead and see what the next part is, and that should be the scoring. So hopping all the way down to the scoring, that is done down here. Then the delta scores can be calculated. So I'll take these, paste those in as well. So right there, indent them correctly. Then this curve can just be calculated down here. So I'll take this and just add this right there. And just print this out. So I'll say print and then something like auroch plus and then auroch, so the score right here. I'll take that, paste it in. Next, the plot can be created but not shown. So I'll take all of this code right here and just copy that. And right on this OROC, I'll indent the code correctly. And then right under here, this is where we return the code back to the local code, our local machine, and then show the plot over here. So we need to have something that returns to this variable, and then we can show the plot right here. So to do this, I'll just encode this as base 40 or 64, and then return it to the other machine, so my local machine, and then show the plot on my local machine. So I'll just go ahead and say import base 64, and also from IO import bytes IO like this. Then scrolling down, I can create a new buffer. Buffer equals bytes IO. Then I can say plot.savefigure like this, and then I'll save it to the buffer in the format PNG. What I can then do is say buffer.seek0 plot data equals base64.base64 encode. Then I can say buffer.getValue and then .decode UTF-8. So the plot up here is saved as an image, and then it is encoded in base 64, and then because this is just numbers, that is decoded to one string. And this encoded string can then be sent back to the local machine from the Modal container and then decoded again. So I'll just say return, and I'll return variance first of all. This will be BRCA1, and then df right here, dot, and then to dictionary. And then I'll say orient with records right here. So this flag right here will convert the data frame into a list of dictionaries where each dictionary in the list corresponds to one row in the data frame. And the keys will then be column names and the values will be the data from that row. We can then just send back the plot as well. So I'll say plot, this will just be plot data. And lastly, I'll just send back the OROC value in case we need that. So I'll say OROC and then OROC like this. And then I just want to do one more quick thing, because right now we are analyzing all the data. So this means that all of these dots will be inferences being ran. So all of these dots are single nucleotide variants. And this is quite a lot. So I want to just run a subset of the dataset and not the entire dataset. So I'll just hop up to right over here somewhere. And then I'll say BRCA1 subset equals BRCA1 data frame dot ILOC. Then I'll say sample size, that will be 500. And then I'll say dot copy. So I'll only take the first 500 samples in the dataset. And then I'll copy these into another data frame in this variable instead. And then of course I just need to say BRCA1 and then subset instead of the actual data frame. And all the places that the data frame is actually used right now, we'll just need to, or at least below this, we'll just need to go ahead and resolve those as well. So going down, it is used right there, right there, right there, and there as well. And then these two last places too. So if we just try and run this, because right now it is being called, we can just see if anything happens or if it completely fails. So hopping into terminal, I can say modal run main.py. And then we'll just see what happens when it is being ran in the cloud. So it just ran for maybe 10 seconds. And as you see, it loaded the image and then it says loading evo2 model. So that is what happens up here whenever you instantiate a new instance of the evo2 model. It then downloaded the model files. That is what it does when it says fetching for files. Then it loaded the model into the GPU memory right here. And then this has been running for a couple of minutes. So now what you see is some progress bars. And these progress bars comes from the score sequences methods being called right here and right here. So because the reason this takes so long is because it has to run inference for all of these different variants, or at least the 500 we picked, so we chose to analyze. And this of course takes a lot of time, even though it is running on a large GPU. So this will just take maybe 5 minutes, and then I'll come back whenever this is done. So we just saw a small error right here after finishing. Print OROC, it can concatenate a string and a float. So I'll just go ahead and remove this line, it actually isn't needed. we do return it down here anyways. And then I can just run the entire thing again, and then we can just see if there are any differences compared to the volume that we have just attached, for example. So what we should see is that when the Evo2 model is being loaded, it shouldn't download or fetch the 4 files and spend like half a minute or a minute doing that. It should happen pretty much instantly. So you see that it's basically done instantly, and then it should say loaded Evo2 model. So that is what we see right here. Now it loads the model into the memory. So what takes a little while here is for Evo2 to load the model into the GPU memory, not the actual fetching of the files. And the reason that we can avoid this is of course because of the volume that we talked about earlier that is attached right here. So that is why that happens. So now that this finishes in a couple of minutes, I'll come back and then we can talk about what we see. So it just finished right here. You see we don't see any errors, and all the inference was done just as it is supposed to be. We don't see any prints either because we didn't really do anything with the return statement, but at least we know we can run the entire analysis without any errors. So now we can go ahead and actually just show the plot. What I'll then do is just call this results instead, and then we can first say if the key plot is in results. In that case, we'll just decode the data. So we'll say plot data equals base 64. We'll just need to import this. base64.b64 decode. And we'll decode the key results plot like this. Now we can say with and then open. And we can say brca1 analysis plots dot png. And then we'll say wb as file. Then we'll write the file to the system. So we'll say f.write, and we'll just write plot data like this. After having written the plot data as a file, as a PNG file to the system, we can go ahead and say we just import the functions needed to read the image and also to show the image. So we'll say import matplotlib.pyplot, and we'll just say as PLT. We'll just also import matplotlib.image as mpemg. Down here we can then show the actual plot. Let me just remove this. Down here we can show the actual plot. So this will be image equals mpemg.mread, and then we'll need to read the bytes actually. So we'll say from IO import bytes IO. Down here we can then say mread and then pass in bytes IO and then plot data. Now we can say plt.figure, and would like a figure with the figure size 10 and 5. Then we can say plt.imageShow, and we would like to show the image, and then plt.axis, and we'll set this to off. And then lastly plt.show like this. So now we should be able to run this command again. So I'll just go ahead and say modal run main.py, and then this should then show the plot whenever the inference up here has finished. So I'll just go ahead and do that and then come back when that is done. So the analysis just finished, as you can see here, but the container is still running. So hopping over here, I can see that this diagram popped up. So it looks very much like the diagram that we saw within the notebook right here, although just with less dots as we would expect because we cut off to only 500 of these different samples. So now we're successfully able to actually run these examples with inference on, or with the Evo2 model on Modal. So with having done this, we should be able to go ahead and build out the backend, which we'll need for the tool, and then deploy that before we build out the front end. But before doing that, let's just hop over to the Excalibur drawing and then just take a look at what we're going to build. So zooming in to backend right here, what we're going to build is a basic structure that looks kind of like this. So we have already built the modal image, but inside the code we'll build a class. So we'll build a class that is called something like an inference endpoint or something like that. And then this class will have two functions. It will have an enter function and an API endpoint function. So this enter function up here will then download the model and then load the model file into memory. And this enter function is only executed the single time when the container is started. So this prevents reloading the model into memory every time the scale down window is hit. So since this is a serverless solution, there will be a cold start of 2 to 3 seconds whenever you hit the endpoint for the first time, and then you aren't inside the scale down window. So let's say we set the scale down window to 120 seconds. Well, then Modal will keep the container running for 120 seconds and then close down the container again. And this means you will be billed for 120 seconds, but that also means you will prevent cold starts for 120 seconds. So it's up to you to decide what the scale down window should be. If we didn't have an inter function like this, but just had an endpoint and then loaded the model into memory in the endpoint, well, then we would have to wait for the model to be loaded into memory every time this endpoint is hit. And this would make all of our requests very slow and also waste a bunch of processing that is definitely not needed. So the next endpoint or the next function we're going to create in our class is going to be our API endpoint. This is what we'll call from the frontend to run an analysis on a mutation. So this API endpoint will receive a mutation. It'll then fetch the relevant sequence inside the window or in a window around this mutation. This function will then score the mutated sequence and also the normal sequence, and then it'll compare these with the mutation analysis which we have looked at earlier, and then it'll determine the harmfulness from some threshold which we'll define. So we can say if the score is less than a specific number, then it is harmful, otherwise it is not harmful. And this will just be a pretty simple post endpoint which will expose this functionality, which you can call with fetch from Next.js. And a Hugging Face volume will also, a normal modal volume will also be attached to the Hugging Face cache folder right here, just like we did with the other function. And this will then just store the downloaded model file from Hugging Face to make sure the requests are even faster as well. So hopping over to the front end here, we can take a look at how the application is going to work. So in the front end, the person or the user will first load the genome assembly. So this might be HG19 or 38, for example, and this is just going to be the reference genome for a human person. So this is of course loaded from an external API, and then the user picks it afterwards. Next, after picking an assembly, all the chromosomes within that assembly is loaded from an external API as well. And the user then has two options. The user can either search for a specific gene within this assembly, or the user can click a chromosome, and then the genes will be searched for, so you'll only see the genes within that chromosome. After selecting a specific gene, the user is then able to create a mutation and then run an analysis on that specific variant. So this step right here is what we'll code right now in the backend. All of the rest of these steps with loading the data will be done with public databases, which just happen to have public APIs for free. So we'll create this endpoint where you can run analysis on a variant, and this will just be done with Modal with Evo2. So that is what we're going to build right now. And this is the structure, which we'll also go ahead and build now. So hopping back over here, we can define a new class. So I'll say class Evo2 model. This will have two functions. The first will be load Evo2 model, and it will accept self as a parameter. So in here, we'll just say pass for now. The next function is going to be def analyze single variant. This will accept self along with the variant position, which is an integer along with an alternative. So an alternative will then be a string as well. We'll also pass in the genome and the chromosome, like this, and this will also be a string. So this is the information you'll need to tell somebody where a mutation is. You'll first of all need the genome, so that is the organism we're working with. This could be a human, for example. You'll then need the chromosome in that genome that the variation is placed at. Within that chromosome, you'll need the specific position for the mutation, and then you'll also need the nucleotide that the mutation has changed the reference nucleotide to. So that is what these four parameters mean. We can then just put on a decorator for this class. So I'll say app.cls for class. We'll give it a GPU of H100, so we'll have this GPU available. We'll of course attach the same value, so we'll say mount path, and this will be the volume. We can then say max containers, so we only want Modal to be able to spin up 3 of these containers at one time. This will make sure we won't get overloaded completely if that happens for some reason. We'll then also say retries, so it should be able to retry up to 2 times. And then we can say scale down window 120 seconds, so it'll keep this container alive for 120 seconds at a time between requests. And now for loading the Evo2 model, we can go ahead and start by saying from Evo2 import and then the Evo2 class, like that. Then we'll just say, then I'll just hop up and grab the code which we wrote earlier. So this code right here, scroll down and just paste that in, like that. And we just need to define with the decorator that this is actually the enter function, just like we talked about over here. So hopping back, I can say modal.enter and just call this function. This is just going to be a normal endpoint, just like a post endpoint, for example. So we'll say modal.FastAPI endpoint, and then this will be a post endpoint. So that is what we define like that. When hitting this endpoint for debugging purposes, we'll always just print out the genome. And then I'll say genome like this. We'll also print out the chromosome. We'll also print out the variant position. And also the variant alternative. So we'll say alternative right there. We'll then also define the window size. So window size, which will run the scoring on. This will just be 80,192. Before we can run the actual scoring, I'll just say scoring down here. We'll need to get the sequence to do the scoring on and then also create the mutation or mutated sequence. But both of these depend on this sequence of nucleotides, which we don't really have. So in the example up here, the sequence was stored in this tarball file right here. So that is where the researchers got the scoring from, the sequence from. So up here, somewhere they loaded the sequence right there. So we'll need to do this dynamically. We can use an API for this, and luckily there are public APIs available for us to be able to do this. So we'll just go ahead and create a new function up here. So I'll say def get genome sequence. We'll pass in a position, a genome, a chromosome, and also the window size. And we'll just set this to the same as this for default. Great. Now we can start by importing requests because we'll use a third-party API to get the sequence. So just hopping back to Excalibur right here, I'll go back to my drawing. So right now what we are experiencing is the exact same problem that we had over here. So we have a super large sequence and we have a position where we would like to introduce a mutation, and then we only want the nucleotides that are inside the window on each side of this mutation. So for that, we'll just need to calculate this starting point and this endpoint, which will fetch the sequence, which we'll use to fetch the sequence. So we'll first define a variable called half window, and that'll be equal to window size divided by 2, and it'll round that if it's an uneven number. This half window size will then correspond to how much we should subtract from this position to get the starting point. Cool. So the starting position will then be the maximum number of 0 because we can't really have a position that is less than 0, and then the position minus 1 minus the half window. Then we can say end, and this will just be, let me just see, position minus 1 plus half window plus 1. So the reason for the minus ones right here and right here is that the genomic positions are 1 based, so we'll need to convert them to a 0 based system. And the reason for the half window plus 1 is that the API we use won't include the last nucleotide in the sequence for 0 based positions. So to make sure we also get that, we'll just add 1 to that position. And then for some debugging statements, we'll just say print, fetching, and then let me just make this a formatted string. So fetching window size base pairs, window, around, position, and we'll pass in the position from the UCSC API, like this. And then under that, we'll just put in another formatted string where we'll say coordinates. And these coordinates will then be the chromosome. We'll also have the start and the end. And then we'll just say which genome it actually is. So we'll say genome like that. So the API we're going to use to fetch the sequence will be the following. So I'll just open up a new tab, search for api.genome.ucsantacruz.edu. And this will then be the documentation for using the API. So what it basically does is that it allows us to browse through genes. So the University of California, Santa Cruz has a genomics institute that has all this data available and then just lets the public use it for free. So they have a genome browser where you can select a specific organism. So if I just click this link, it should take me to this page in just a bit. So it says human right here. You can then select human, for example. So this is a specific human genome assembly. And then you can put in a specific position if you would like to. So let's just try and put in these positions and press go. And then just see what happens. So now you get a big overview where you can browse around inside the genes in a human reference genome. Anyways, hopping back to the documentation for the endpoint, that is this one right here. It says that we can call the API with the following URL, and then we can pass in a genome, which we would like to use, a specific chromosome, a start, and an end. So that is basically what we'll need to find the sequence which we're interested in. So to use this API, we can go ahead and say API URL equals a formatted string, HTTPS, and then we'll say api.genome.ucsc.edu. We'll then use the action called get data and then sequence. So we would like to get a specific sequence. And for the query parameters, we'll pass in the genome as it said. So genome equals genome. We'll also pass in a chromosome, which we'll do this way. Chromosome. We'll pass in a start. And we'll pass in an end. Like that. And then we can use this Python package requests to actually request this URL with a get request. So I'll say response equals requests.get, and we'll then get the API URL. The first thing we'll just check for is whether the response was good or bad. So we'll say if response.status code is not equal to 200. So 200 is the success status code for HTTP requests. In that case, we'll just raise an exception where we say fail to fetch genome sequence from UCSC API. And then we'll just pass in the response.status code so we can see what the actual problem is. Otherwise, we'll take the response and make it into JSON. So we'll say genome data equals response.json. We can then say if DNA not in genome data. In that case, we'll say error equals genome data.get, and we can then get an error. And this will then fall back to unknown error if the error returned from the response up here does not exist. Then we'll just say raise exception, and we'll raise UCSC API error, and we'll just pass in the error like this. So why do this, you might think? Well, if we hop into right here and search for error, we can find an example error down here somewhere. So if we just click this, we should be able to see an example error. So we see that there is a key in the dictionary called error with this specific error message. So that is what we are interested in and we would like to print out. So that is why we do what we do. I'll just close this down and hop back into the code. If the DNA key is in the response though, we'll just say sequence equals genome data and then .get. We'll get the DNA key and say .upper. We can then just do a quick sanity check like they did in here. So remember how they used a search to do a quick sanity check? Well, we'll do the same in here. So we'll go ahead and say expected length and that is going to be end minus starts. Then we'll just say if the length of the return sequence, which we just had returned from this API, is not equal to the expected length, then we'll go ahead and say print and then if warning received sequence length of the sequence differs from expected. So this way we'll know if that actually happens. So this way you can kind of be notified if the APIs that you interact with do not behave as they're supposed to. Otherwise, if that is not triggered, we'll just go ahead and say print, or not otherwise, this will happen anyways. We'll say loaded reference genome sequence window, and we'll say length and just put the length in as well. So I'll say length sequence and then say basis right after that. Lastly, now that we have the sequence available, we can just go ahead and return sequence and also where it started from. So we can start by just trying out this method right here. So if I just say get genome sequence and then hop down and call that in here, I'll then say, I'll just do it right now. I'll say get genome sequence, and I would like to pass in a position. That's going to be the variant position. Also a genome. That's going to be the genome. We'll also want to pass in the chromosome, which will be the chromosome, of course. And lastly, the window size, which will be equal to the window size. It will then return a window sequence and a sequence start. And we can just go ahead and print these out. So I'll say print and then window sequence as well as a sequence starts. Then I'll just go ahead and turn this into something we can test locally. So this is an endpoint right now, and we don't really want to test an endpoint. We want to say modal.method instead. And down here in the local entry point, we'll then say evo2modal equals evo2modal. This will also call the inter function up here and make sure it loads. This needs to be self.model, by the way. Then we can scroll down. Under this, we can say evo2modal dot analyze single variant, and we'll just do this remote. And then we can say variant position. So I have this example prepared. You can say 43, 11, 9, 6, 28. For the alternative, we can put in a G character. For the genome, you can put in HG38, that's the human genome. And for chromosome, you can put in chromosome 17. Now let's just go ahead and save that and then try and run it. So I'll open up the terminal and then I'll say modal run main.py. And I'll just wait for this to run and then come back when it's done. It just finished, so let's just check out what it printed out. So first of all, we see that it loaded the model into memory like it did right here. That is perfectly fine. Then it says evo2 model loaded, printed out the genome, chromosome, etc. We can then see the entire reference genome sequence window of the 8,000 nucleotides which we loaded from the API. So everything seems to be working perfectly fine, so that is great. I'll then just go ahead and print out the following. Fetched genome sequence window, and then first 100, and then I'll say window sequence, and then just cut the first 100 nucleotides off. Good. Now we can just calculate the position within the window and then make it zero based. So I'll say relative position equals variant position minus 1 minus sequence start. Then I'll just say print, and then I'll put in relative position within window, and this will just be relative position. And of course this just needs to be relative position like that. And now I'll just do one more sanity check. So I'll say if relative position is less than 0 or relative position is more or equal to length of the window sequence. Well, this means that the position right here is outside the sequence which we just fetched. So this is obviously wrong. So we'll then say erase and just something like value error, and then put in a format string, variant position is outside the fetched window, and we'll just put in the start, which is equal to sequence starts, and just say +1, and then we can go ahead and say end as well. So end equals, this will then be sequence starts plus the length of the window sequence. Great. Let's just go ahead and find the reference nucleotide, because right now we have the alternative, but we do not have the reference nucleotide. And because we have both the position and now the relative position as well as the sequence, we can go ahead and find that. So I'll say reference equals window sequence, and then just put in the relative position. Now I'll say print reference is, and just say reference like that. And now we just need to actually analyze the variant, but we don't really have any function to do that right now. So we'll just hop up to right below this get genome sequence, and then do it right here. So it'll be about here. So I'll say def analyze variant. So this will analyze a single nucleotide variant. So we'll need a relative position in the window. We'll also need the reference. We'll need an alternative, as well as a window sequence. And lastly, we'll need the AI model. So that is going to be the Evo2 model. Then we'll just create the variant sequence now. So hopping back into Excalibur, that is what we do right here. So we just take the first part of the window. We'll then exchange the reference with the variant, and then we'll append the second half of the window again. So it's basically the same we do over here. So we're going to say variant sequence equals window sequence. So we'll cut off all the way up until relative position in window. And so it's only from the start and up until the relative position in the window. Then we'll append the alternative. So this is the mutated single nucleotide variant. Then we'll say plus window sequence, and then we'll pass in the relative position in window, and then +1, and then cut off. So the first part right here is what corresponds to this part right here. So what this does is that it takes the window sequence and then keeps the part of the window sequence from the start of the sequence and then up to, but not including the specified relative position. So not including the reference. The alternative is then added instead of the reference. And this right here then corresponds to the following part right here. So what this does is that it takes the second half of the window sequence right after the relative position, so right after this new inserted alternative, and then all the way till the end of the window sequence. So this is then the reference sequence, and this is then the variant sequence. So we can go ahead and just score both of these sequences now. So we'll say ref score equals model dot, and then score sequences, because remember, this is what we have done up here as well. And then you just pass in the reference sequence. And because this score sequences is able to score a full list of sequences, we then just take the first result coming back after that. So I'll pass in a new list of sequences, and this will then just be the window sequence, because we only need to process this single sequence right here. And then I'll just take the first result of that. I'll then create another variable called variant score. This will then just be the variant sequence, and we'll just take the first element from the return statement of this function as well. We can then calculate the difference between the reference and the variable score. This will then just be delta score equals variable score minus reference score. So hopping into this right here, that is exactly what they also did in the notebook. I'll just close this down. If we find out where the scoring actually is, that is down here. They get the reference scores and the variable scores and the variant score, and then they calculate the delta score, which is all the different variant scores, and then we subtract all the different reference scores. So instead of subtracting from these two with these two arrays because they have multiple different sequences, we only need to subtract the first element of these arrays. So that is what we get right here, and then we just subtract those two scores manually. Now from the score, we would like to define whether this variant right here is harmful or not. So we'll need to define some kind of threshold to define whether it's harmful or not harmful. So going into the notebook, what they did in the notebook right here was just to plot the scores along with the median and then the category, but they didn't really define whether a specific variant was harmful or not harmful. And the reason they didn't do that is because they already know from the categories, so this is loss of function variants, so these are obviously very harmful, while these up here are not. But if we work with unseen data where we don't have these two labels, well, we need to find a threshold which we can use to categorize variants as either harmful or not harmful. So instead of just picking a threshold ourselves and then hoping that that is a good threshold, we can use some of the data from this notebook right here to actually pick one for us. So we'll calculate some thresholds in a data-driven way such that it isn't just a defined threshold that we did ourselves that doesn't really make any sense. So hopping into the calculator right here, we can talk about how that can be done. So let's talk about calculating thresholds. So the goal of this section right here is that during inference, we would like an output that is either likely pathogenic or likely benign. So these are the two classes that we should be able to classify mutations into. So a numerical score doesn't say much about the severity of the mutation when a user reads it. So this numerical score I'm talking about here is of course the delta score. So if the user sees a score like minus 0.0005, like what does it even mean to a user? The user can't really comprehend what that means, and neither can I. But the user can easily comprehend whether a mutation is pathogenic or benign. So what we're trying to achieve is abstract the score into something that is comprehensible by normal people. So the way we'll do this is we'll define a single threshold / a number where if the score is above this number, it is pathogenic, otherwise the mutation is benign. So if the number is too low, too many variants will be classified as benign, but if the number is also too high, then too many of these variants will be classified as harmful when they actually aren't. And that is why it's important to define a threshold that actually classifies these two categories as accurately as possible. So how do we actually define this threshold number? Well, we can use the data from the BRCA1 example and then calculate a useful threshold from the labeled data. Because remember, in the dataset in this notebook, they had actually labeled the data. So they had the scores right here, along with the class about whether it is harmful or not harmful. So this func_int means not harmful, whereas LOF, so loss of function, means harmful. So with this data available, we can go ahead and use a method, which is based on the ROC curve, to actually calculate this threshold. So down here is an actual ROC curve and how it would look if we illustrated that. So in short, this ROC curve will help us visualize how well the delta score, so this threshold right here, separates the harmful from benign variants across all possible cutoff points, which is the line you see right here. So you can analyze this curve right here, and then you can particularly find the point maximizing Newton's index, so that is what we see right here, and this will then determine the most effective delta score threshold to use for classification. That will then be the perfect threshold for us. So on the axes of this graph, we have two different things. On the y-axis, there is the true positive rate, TPR, also called sensitivity. These are the fraction of true benign variants that has also correctly been identified as benign. On the x-axis, we have the fraction of true harmful variants that has incorrectly been identified as benign. This is called the false positive rate, FBR, and that is also the same as 1 minus specificity. So just hopping up here, we can just see the table that is the excerpt from the notebook. So what we can see is that the score is lower if it's more dangerous. So the more harmful the mutations are, the larger the negative score. So the delta score is therefore negated by us. This means that we just take a negative sign and just put that on to all the different scores. This means that the larger the larger the positive number is, the more harmful it also is. This means that the curve will go up like this instead of going the opposite way. This the function for the ROC curve expects it in that way, so that is why you see this minus sign in front of this delta score thresholds. So it's just to take all these scores and then remove the minus sign, basically. So we'll use a function, so an ROC curve function from a scikit library. And this will then just take all this labeled data and then create this for us and then calculate this Newton J index and thereby also the threshold for us. So the higher we are on the y-axis right here, the more true benign variants are then correctly identified. So we want this number to be as high as possible on the y-axis. At the same time, we want the number to be as low as possible on the x-axis. And that is because the x-axis marks the fraction of true harmful variants incorrectly identified. So the higher the number is on the x-axis, the more incorrect identifications we do have. So that is also the reason we don't just take the highest threshold up here, just because it has the highest y-axis number. So we need to calculate some kind of threshold where we get as many true positives as possible, while getting as many false or as few false positives as possible as well. So it's kind of a trade-off. So what you see right here in the middle is the random chance line. So these would just be thresholds where the true positive rate and the false positive rates are identified equal. So we want the highest true positive rate along with the lowest false positive rate. So therefore the top left corner is the best position if that was possible. But that of course isn't possible because we can only pick points from the curve right here. So one way to calculate this trade-off right here is with the Newton's index. So Newton's index maximizes the vertical line distance you see right here, letting us find this optimal threshold. So this Newton's index is TPR minus FPR. So we take this number and then we deduct this number from that. And this is just one way to balance the highest sensitivity, so the true positive rate, with the highest specificity, so the lowest FPR. So by using this technique, we can then have the threshold calculated for us. So we can actually, because this is based off of the notebook, we can actually go ahead and go back to the code and then find our example with BRCA up here and then calculate the threshold from that. So I want to go up to the right under the ROC score, and then I'll just create a new section right here where I say calculate threshold, because this is something we made ourselves, so we'll just make sure that we know the difference, and then calculate threshold, and then I'll just say end. So I'll just go ahead and do this. Then I'll say y true equals, and then this will be BRCA one subset, and we'll take the class label and then say equals LOF, so loss of function. So what this does right here is that it creates a Boolean array, so an array where the values can be true or false, and true are the classes that has loss of function. So this means that the different indexes in the array will be true when the class is loss of function. So if we go up here, where this class is loss of function, and then otherwise the array index value would then be false if this is func int, for example. So a true positive would then be where a loss of function would be predicted as a loss of function, and a false positive would be where a func int is predicted as a loss of function. So those will be the two axes that we see right here on the ROC curve. Now we can just import the function for actually using this ROC curve. So I'll hop all the way up and then say from, we can actually do it right here. We'll see ROC and then curve. So we'll take this and then hop all the way down here. Then what I'll do is just go ahead and say ROC curve, and then I'll pass in the YOC, Y true, and then I'll negate the scores. So I'll say minus BRCA subset, and we'll only take the scores and select those. This will then return a list of false positive rates, true positive rates, and thresholds like this. So thresholds will just be an array, the same for true positive rate and for false positive rate. So this would be an array of false positive rates calculated at different thresholds. This would then be an array of true positive rates calculated at the same thresholds corresponding to the false positive rates values. And the thresholds array right here would then be an array of numbers. So these would be the different thresholds used to calculate the FPR and then also the TPR. And then if we go back right here, we can then see that the Newton's, if we want to maximize the Newton's index, we need to maximize where TPR minus FPR is the highest. So we'll go ahead and do that. So what we can do is say optimal IDX, so index ID. And this will then be TPR minus FPR. And then we can say dot argmax. So this will return the index corresponding to the point on the ROC curve where TPR minus FPR is maximized. So since this is just the index, we'll need to go ahead and actually get the threshold as well. So I'll say optimal threshold equals, and then I'll say minus threshold because we negated it up here. So we'll need to negate it back. So we'll say thresholds, and then we'll put in the optimal IDX. Then we'll just go ahead and create a new, as we'll say, a new dictionary. This will be confidence parameters, because we'll also calculate the confidence in this optimal threshold. We'll then say, calculate the confidence in the prediction and not in the threshold. So we'll then say threshold, that is the first thing we'll add. This will then just be the optimal threshold. And then below this, we can say print confidence parameters, and we'll then just print out the confidence parameters. So when we run this BRCA1 analysis function again, we should then see the confidence parameters being calculated and also printed out. But before doing that, we can just go ahead and also predict the confidence, the confidence in the model's predictions. And this is the way we're going to do that. So the basic idea is that if we calculate a mutation score, so that is going to be the variance score versus the, minus the reference score, and it is very far from that threshold, we can then be more confident that the prediction is correct. So to give you a visual example, let's say we use this notebook example, so the diagram they used. Well, let's say we then take the category loss of function down here, and then we say that the median right here is going to be our threshold. So if we are on this side of the median of the threshold, then we won't have a loss of function. But if we are on this side of the threshold, so the left side where the scores are getting bigger, then we'll classify it as having loss of function, so being more harmful. So up here, I actually say the same just for the category here. So let's say we use this median as the threshold for harmful/not harmful. So harmful on this side and not harmful on this side. Well, then these variants very close to the edge, we probably aren't as confident in as these variants out here. So if we have a variant all the way out here on the safe side, then this is probably a safe variant. And if we have a variant all the way out here on the harmful side, then this is probably a harmful variant. But these variants very close to the threshold, well, we don't really know if they are that harmful or not, so we can't really be that confident in the prediction. So the further away the variants are from the threshold, the more confident we can be that the predictions are actually strong and correct. So when calculating how far away our predicted variants are from the from the median or the threshold, which we'll use, we'll need to take the standard deviation into account. So the standard deviation is a measure of how spread out the values are from the mean in a diagram like this, for example. So the more spread out the values are generally in the entire plot, the higher the standard deviation is going to be. So down here I say the more spread out the values are, the higher the delta score must also be before we consider it far from the mean. So up here, for example, with the loss of function, you can see that right here on the left side, the variants are very much spread out compared to what they are up here on the functional, in the other category. So let's say that we have a variant in the loss of function that is predicted all the way over here. Well, because there are so many other variants already, we can say, well, probably it's normal that they're over there because they're spread out that much already. But up in a plot like this, where all the variants aren't spread out very much and they are very close to the median, if we do actually see a variant all the way out here, well, then we can definitely be sure that it's very much outside of the normal range, because almost all of the previously seen variants are very close to the threshold right here in the middle. So from this, we can then say the confidence in the prediction can be calculated as the distance from the threshold divided by the spread of scores for the predicted class. So to make this even more tangible, we do the same calculation down here. So we can say the distance from the threshold, so how far away from this threshold a single variant is. Well, that's just equal to the threshold, the delta score minus the threshold. So that'll be, for example, the x position of this one minus the threshold's x position. And that'll give us the distance between these 2 points right here. And then because we also need to take into account the standard deviation, so how spread the values are in this plot right here, we'll then divide that absolute distance with the loss of function standard deviation. And the reason we have a loss of function standard deviation instead of just a single standard deviation for both categories is because values are spread differently based on whether they're classified as harmful or benign. So we need two different standard deviations. So as you see in this plot up here, these ones are spread out way more than they are up here in the functional, in the benign category. So we do need a category for both, a standard deviation for both. So this distance from the threshold is put inside an absolute function. So for example, we might encounter negative scores or negative distances because we have both variants on the left side and on the right side of the threshold. So to always have a positive number, we'll just put it inside an absolute. And we then just divide with the loss of function standard deviation and then put this inside a min function that takes the minimum number of either this one right here or 1. So we cap the confidence to 1. So since we divide by the standard deviation, this means that if the distance is more than 1 standard deviation away from the threshold, we then cap the confidence to 1. So there isn't really any rule for this, it is just a definition we use. So the score is at least as far from the decision boundary as the typical spread of scores within a predicted group if the confidence is capped to 1. So if a variance score is further away than the typical spread of scores used to calculate the threshold, well then we can be very confident in its prediction, so we'll just cap it to 1. Cool. So again up here with this first part of the calculation, so this first term, we then calculate the raw normalized distance from the threshold, and then it is divided by the standard deviation. So this is a normalization step to normalize it to the actual spread. So this will divide the raw distance by the typical spread of scores for the pathogenic class. So this basically means how many of these pathogenic standard step sizes fit into the actual distance. So to actually go ahead and calculate the confidence for a prediction, we'll need to calculate the standard deviation as well. So we can do that as well right here after calculating the optimal threshold. To calculate the standard deviation, we'll first do it for the loss of function scores. This will be equal to BRCA1 and this subset, of course, then .loc. We'll then say BRCA1 subset. We'll grab the class column where the value is equal to loss of function, and then we'll select the evo2, let me just see, delta score right here. So what this basically does right here is that it extracts all the evo2 delta scores values from the variants that are classified as loss of function in the BRCA1 subset data frame. Then just returns a series which we can calculate the standard deviation from. So I'll go ahead and do the same just for, let me just see, just for the functional, or sorry, the benign class. So functional or int. And this will then just be FUNC scores instead. Now for the standard deviation calculation, we can say LOF STD equals BRCA, LOF scores dot, and we can just do .std. And we can do the exact same just for the functional scores. And we'll then just say FUNC score, standard deviation instead. We'll just need to change this to ST and then D. And then I'll just print these as well down here. So I'll go ahead and add a key and a value, and then a key and another value down here. So we'll need to put in a comma, and then this should be printed out as well. So now that we have the math ready to actually go ahead and calculate these thresholds, we can just do that by running the BRCA1 analysis script again. So I can go all the way down and just comment this out, and then say BRCA1 example.remote. So let me just check that this is the correct one. And it certainly is. So that is perfect. So I'll go ahead and do that. Say terminal, modal, run main.py. And then just let this run and then come back whenever we have something tangible I can show you. So it just finished running and now we actually have our thresholds. So I'll go ahead and just copy these in. So I'll take this entire line, copy it, and just make that a comment for now. So down here, I'll just say new comment and then say thresholds, and then just paste these in just so we have them for future reference. Then I'll go ahead and close down the terminal and then just take this comment up to where we actually need it. And that is within the analyze variant function. So up here. So I'll just go ahead and create 3 new variables. I'll create the threshold like this, and that will be equal to, and that is the following number. So I'll take this, just copy it in. Then I'll create the loss of function standard deviation. That is the following number right here. Then I'll paste in the benign standard deviation. That is going to be the following number right here. We can then determine the actual prediction and also the confidence. So I'll say if delta score is less than the threshold, so this is pretty straightforward, then the prediction will be equal to likely pathogenic. The reason we don't just say pathogenic is because we have calculated these thresholds. These aren't absolute universal truths. These are something we have calculated, so we can't be 100% sure. So we say likely pathogenic. And the confidence will then be calculated from the formula right here. So I'll go ahead and say confidence equals minimum, and this will be 1.0. And then The other value is going to be the absolute value of delta score minus threshold divided by loss of function standard deviation. And the reason we say if the delta score is less than the threshold is because we have negated values. So remember up here, I think I showed you this earlier, the more harmful the values are, the smaller they will be. So this is a smaller value than this one, and this one is more harmful. This means that over here, if the delta score is lower than this threshold right here, then we consider it likely pathogenic. Great, so if it is above this threshold, we'll then say else, and then prediction equals likely benign. We can then also calculate the confidence just with the functional standard deviation like that. And then the last thing we want to do is just return the information that we have just calculated. So down here, I'll say return. I'll return the reference. That's going to be just the reference from up here. So I'll return reference. I'll return the alternative. So right there and right there. I'll then also return the delta score. So this will look like that. I'll then return the prediction. The prediction is going to be either likely pathogenic or likely benign. And then lastly, I can return the confidence. So it'll look like this. I'll just call this classification confidence, because that is what it is. And then for these floats, we'll just go ahead and say floats, delta score, and floats confidence as well. So I just want to make sure that these two numbers are returned as actual floats. Good. So I'll go ahead and delete this threshold right here. Then say analyze variant. And then we can go down and call that down here. So this will look something like that. Result equals analyze variant. I pass in the relative position in window. That is going to be relative position. We then also pass in the reference. That'll just be reference. We pass in the alternative. We then also pass in the window sequence. That is just going to be window sequence, as well as the model. That is just going to be self.model. So self.model we get from the enter function up here. So whenever this runs, when the container starts up after a cold start, this will then attach the eval2 model instance to the instance variable self.model. This means we can get it from other methods which we use down here. And lastly, we're going to return something from this endpoint as well. So if I just hop down, we'll just say results and then position. So we'll just attach this new column or this new dictionary item position. This is going to be variant position, and then say return results. And this should then be all the different information which we're going to need in the front end to be able to render out something nice for the prediction and also confidence when the user analyzes a variant. So we can start by just testing it out locally. So I'll comment out this BRCA1 example, and then I'll comment in this right here with the example that we know exists. Then I'll open up the terminal once again, say modal run main.py, and now we should be able to just say result equals, and then something like print results. And then hopefully this should give us a result that we should be able to interpret. So let's just try and press enter and run this, and then I'll come back whenever this is finished running. Alright, so we have our first result coming back. So what is being printed out right here is the reference, the alternative AG, and then we have the delta score, as well as the prediction that is likely benign, and then the classification confidence we can't really see. So I'll just go ahead and run this again, but only print out the classification confidence, just to make sure that we actually have this available and that it isn't just being cut off. This classification confidence is also a float, so we just need to wrap it in a str like this just to convert it to a string. Then I'll just go ahead and say classification and then append that to the string. So classification, and then just go ahead and save that, and then say modal run main.py and press enter. And this should allow us to actually run this code. So I'll just come back when this is done. And now the classification is being printed out as it is supposed to be. So apparently there's nothing wrong with our actual code. It was just the classification that was being cut off somewhere out here on the right. So that is great. So now we should be able to go ahead and make this an endpoint instead of a method we run locally. So I'll comment out modal.method, and then I'll say modal.FastAPI endpoint instead. Just save that. And then down here we can say modal deploy main.py, and this should then deploy the app for us. So it'll create this endpoint, which we will be able to call. And then we should also be able to see this if I just copy the link right here and then hop into a new tab and just open it up here in Modal. So we see variant analysis evo2. We then see this class called evo2 model right here. So that is this one. And then we also have this analyze single variant endpoint. So that is the one we see right here. So now that we know that it is live, we can just try and go ahead and call it with curl here in the terminal, and then try and actually call it and see what we can see in here in the dashboard. Alright, so I just had a small error testing, so that is why I just opened up a new terminal. And then what I want you to do is just take this link right here and just copy that. Then hop back into your terminal, say curl -x, and then it is a post request as we defined up here in the FastAPI endpoint. We can then pass in the URL itself, and then also the dummy data we used earlier. So we'll say query parameter variant position. That should be 43 and then 11, 9, 6, and then 28. We'll then say and alternative is equal to AG. We'll then also say and genome is equal to HG38. And chromosome is equal to chromosome 17. If we then just press enter, that should trigger the endpoint. So going over here, we can see that we have one cold starting endpoint container right now. So that should be possible to see right here in the function calls in just a second. So that is this one. So I'll just go ahead and wait for that to finish as well. So it just changed from pending to status 200. So opening it up, let's just see what it printed out. So we see post 200, which is fine. And then we see these debugging logs that we also printed out locally. So that looks perfectly fine. Hopping back into the terminal, we then get reference, alternative, we get the delta score, the prediction, likely benign, as well as the classification confidence and position right here. So with that done, we have actually deployed our entire backend, which is this analyze single variant endpoint. And right now, even though it is deployed and ready to be run, we aren't getting built because this is running serverless. So we are only getting built for the startup and then the execution of these containers. So with the backend built out and deployed, we should be ready to go ahead and build out the frontend now. What I'll first do is hop into my browser and then search for the T3 stack. That is what we'll initialize our Next.js app from. Then I'll click this copy button and say npm. Hopping back into my terminal, my VS Code right here, I can then say open folder. And then I would like to find my repositories and then just the variant analysis folder right here. Now we see that we have the backend folder already. So we'll open up a new terminal in the root right here and then paste in the command that we just copied. Our project should be called Frontend or something like that. So I'll just say variant analysis and then evo2 frontend like this. Then I'll use Typescript, Tailwind CSS, node to TRPC. I would like to just go without authentication just for this time, and then I'll select none. And then I would like to use the app router and then ESLint. And then I would like to also not initialize GitHub repository. You might want to do this though, if you would like to store this in a GitHub repo. Then I'll just say yes to npm install, and then just enter on this one as well. And then I'll just wait for this to finish and then come back when it is done. So just finished installing all the npm packages. So one thing I would like to do is just rename this to evo2 frontend, and then just this backend to evo2 backend, just so we have some more standardized naming. Then what I'll do is just open up a new folder right here in VS Code. Then just select my folder and then evo2 frontend. And then I'll just open this as the project that is opened up. Cool. Then under npm scripts, we should be able to press def right here. If you can't see npm scripts, you should be able to right click and then select npm scripts. Otherwise, you might need to click the package.json and this menu right here will appear, or at least the option to have it open up. So I've just opened up the VS Code in the left pane and then the browser over here in the right pane. This means we can actually see what we code while coding it. So I'll just copy this URL right here and paste it into my browser. and then we should be able to see the actual application running. So that is what we see right here, and we are hosting it locally. So the first things we can start doing are just renaming the actual page up here. So we would like to create a single page. So if I just open up Excalibur, then we can just go through the flow quickly. So I would like a single page where all of this can be done. So this means that first of all, we should create a simple page where a user can load a genome assembly. So this would be HG38, for example. And the list of genomes we'll get from the following API right here. And then after doing that, we'll be able to build out the next steps, which is loading chromosomes, genes, selecting genes, and then also running an analysis on those variants that the user then creates. So hopping up here, I'll just select my page again, and then we can see that it says, let me just make this a little bit bigger. We can see that it says create T3 app right now, and that is not what we want. I'll just close down these as well. This one, and just leave that. So I would like this to be something like Evo2 variant analysis. So to do this, we'll hop under the app folder right here and then into layout. And inside this layout file, we can define the metadata. So right here, I'll just say Evo2 variant analysis and just do the same for the description and press save. And now we should see that up here. We still see this little T3 logo though, that is called the favicon. The favicon is stored inside the public folder right here, where we can store all of our static assets. So instead, we'll just use a service online to create a new one instead of T3. So I'll just search for favicon generator and then text. And then we should be able to find one where we can actually generate our favicon. So right here, we can say text to icon, and then I'll just say WA as a text for variant analysis. I'll just pick something like enter. And then for the font color, I'll pick green, something like this. And for the background, I'll just pick white. Maybe more of a creamy color. Or let me just go with white. And then a darker color like this. And then for the font size, let me just make it a little bit smaller, like that. So let me just download this favicon right here. And then I can upload it into, I can copy paste it into the public folder here. So I just copied the favicon.ico file into my clip holder. And then I can paste it into the public folder right here. So I'll go ahead and delete the original favicon and then just rename this to favicon. And then going back, we should be able to see the correct favicon if we say reload. And now we can say that it says VA for variant analysis instead of just the T3 stack icon. And then we'll need to install some npm packages as well. So one we'll need is going to be Lucid React. This is just a package for managing icons, so we can use a bunch of different icons through this package. So all of these icons we can just import. So we'll just go ahead and install that. So we'll choose npm, copy the command, open a new terminal, then say npm install Lucid React. We'll also just need to install Shadcn, which we'll use for some of the styling. We'll customize the styling a bit ourselves, but we'll use some of the features in Shadcn, such as the cards, for example. So this card right here, we can at least use, that is very useful, so we don't have to define that ourselves. And then we'll style it with our own color schemes, style, etc. So first we'll just go ahead and install Shadcn for Next.js. So that'll be npm, and then we can just go ahead and install it right here. Paste it into the terminal. Say Y to install the newest one. Neutral, and then just use force. And now that is done. So we can start by just importing a simple button. So I'll just go ahead and copy this and just paste that in. Use force. And now we should, under components, be able to see /UI and then /button. So this is not something you should go ahead and edit, because this is just what was generated by Shadcn. So now, because we just installed this button component into our system, we can go ahead and use the button, as you see right here, along with all its features, styles, etc. You can also style it yourself, which is what we're going to do. So let's get started with the actual application. In here right now we get an error, so I'll just go ahead and restart the terminal in case something wasn't completely noticed by Next.js whenever we installed this. So I'll just refresh the page over here. So I just spent 5 minutes looking into this, and apparently if you go to Shadcn, so their GitHub repository, they changed something regarding Tailwind Animate CSS about 50 minutes ago, so right before installing the actual package, and this is what causes the following error. So I'll go ahead and just install this package manually. So I'll copy this command, paste it into the terminal, and then as you can see over here, when I do install it, it should fix itself over here, and now we'll no longer see the error. So this is just a small bug in the Shadcn repo right now. It's probably fixed by the time this video comes out though. We can now get started with designing this page in itself. So we can go to page.tsx. I'll make this a little bit smaller and hide the terminal. And then just also this page, this pane over here. So what I would like to do is just first of all remove the content of this homepage component. So this page component right here is what renders out this entire page. And this page component is being wrapped inside of this layout.tsx. So it's being wrapped with an HTML and also a body tag. This right here is a server component. So the layout file is going to be a server component, and this page.tsx is also a server component right now, but this will probably become a client component so we can use states within it. So the first thing I would like to do is just change this main right here. So I'll go ahead and just create a single div with the class name minimum height of screen, and then also a background color that is going to be hashtag and then E9 EEE and then A. We just need to close off the parentheses, so it's more of a creamy color compared to a regular white color, as you can see. That is what I wanted. Then we'll go ahead and create a header. So first I'll just put in a header tag. So I'll say header, and then I'll say class name, border, bottom, and then I'll put in the border color, which is going to be hashtag free-C, and then 4F free-D. Save that, and then say BG whites. And now we can put in a height, for example, of 7, just to test that it actually works. So we do see the header up here, so that looks great. And I'll just say divided by 10 over here. So what this does, when I just hover it, is that it says 10% color. So when I go ahead and save this, the border should be way less accentuated. So I'll just say border B instead of border bottom, and then try and remove this. So now this is what the border looks like without the 10% filter. And then when I put on this, which removes 90% of the color, it looks way more subtle. Okay, I would like to just remove this height and then just let the height be determined by the actual contents of this div. So I'll put in another div, and I'll say class name, container, mx auto, px of 6 and py of 4. So the container class right here will set a maximum width for the elements, and then the mx auto will help center it horizontally. And this is just a padding horizontally, and this is a vertical padding. Inside this, I'll put in two elements. The first element is going to be a div with the class name relative. Inside this relative, I'll put in an h1. Inside this h1, I'll put in two spans. The first one will say evo, and the next span will say 2. I'll then just do some styling for this h1. It's going to be text extra large, font light, tracking wide, like this, and then also text, and then the text will be hashtag free-C and then 4F free-D, like that. And down here I'll say class name font normal, and then for the 2 I would like to color that orange, so I'll say class name text and then color it DE and then 82 and then 46, so it looks something like this. Now we can also put in a cool little effect, so a little border, so I can put in this div and then say class name absolute minus bottom 1 left 0, a height of 2 pixels, width of 12, and a background that is the same as the orange color up here. So it's going to look like this. So the reason it can be positioned like this is because it has absolute, and then it is wrapped within a div that has the relative tag or the relative class up here. So it is positioned relative to this class right here. Now I would like to also just add another span right here. And this span should say variant analysis. So it'll look like this, but then instead of this being below the Evo2 logo, I would like it to be to the right of it. So I'll put in another div and then give it the class name flex items center. And just copy this div end tag down here. And now when I save, they should be next to each other like that. Then I would also like to put in some spacing between these. So I'll just go ahead and put in a gap of 3, then put in some classes for this text. So I'll say text small, font lights, and then text. And then I'll put in 3C, 4D, 4F, and then 3D. And then I'll say with 70% opacity. And I just need to spell this correctly. like that. So now we have this pretty cool header down, which can be reused throughout our page. So that is great. So now I would like to put in the actual contents of the page right here. So the first thing I'll do is put in a card. So if I hop into Shadcn, so right there, we can then go ahead and install the card. So I'll find the card and then edit right here. So I'll do this, terminal, open a new terminal, and then add the card. That should then install the card over here under components/UI/card, and then I can just restart the server. And we should then be able to actually go ahead and use it. So under the header right here, I'll say main, put in this main tag, and then I'll put in a container as well, mx auto, px of 6, and then py of 6. I'll then put in a card component, so I'll say card reference to the Shadcn card, and we do need a closing tag, so I'll go ahead and say card. Inside this card, I'll put in some classes, margin bottom 6, gap of 0, border none, bg whites, py 0, and shadow small. We can then also import a card header, so we can say card header like that, and then just put in a closing tag too, and then for the classes we'll say padding top of 4, and then padding bottom of 2. Inside the card header we'll put in a div. Inside this div we'll put in some class names, flex, items, center, justify, between. Inside this div we'll put in a card title like that. The card title should contain Genome assembly, so I'll just save that and then just try and refresh over here to see if it's, yeah, now we see it didn't really load before, but now it loads. Cool. And then we just need to style the card title a little bit because it doesn't really look that great. So I'll say text small, font normal, and then I'll say text, and we just want to copy the same text color with 70% opacity like up here. So I'll go ahead and do this, press save, and now it looks way better. Just need to spell this correctly though, like that. And then I would like to make the user aware that they're actually working with the human genome, and then they can pick a genome assembly within the human genome. So to do this, I'll just say div, and then organism, and I'll put in a span that says human, and the span should just make the human text bold. So I'll say font medium, like this, and we just need to make this text a little bit smaller as well. So I'll say text extra small, and then I'll put in this text right here, and just make it 60 instead, so it's a little bit less visible. So when we put in the picker right here, the user should be able to see that they can pick a genome assembly from the organism, which is the human. We can then go ahead and get the actually available genomes. So I'll do this in another file. So I'll hop under source and then create a new folder called utils. Inside utils, I'll create one called Genome API.ts. In here I'll say export asynchronous function, get available genomes, like that, and then just spell this correctly, available genomes, and then we can just say console.log hello, for example. And then for now I'll just take this and just call it once right here. So I'll say use effect, and then I'll call the following function on use effect, get available genomes, and then just pass in an empty dependency array, making sure that it is only called once. And I'll just go ahead and await it since it is asynchronous, and we can't do that because we're inside this use effect. Let's just see if we can make this async. We can not as well. So what we'll do is say const fetch genomes, and that will be an async function. And then we can put get available genomes into that, and then call fetch genomes like this. So to be able to work with the data that is returned from this get available genomes, we can just say const data equals. We'll need to put it inside an async function such that we can actually await it. Otherwise, we won't be able to get anything but the promise that is returned from this get available genomes function. So if I just hop into the console over here, press F12, we should be able to see a printout. So I'll just hop back in here, see what is happening. So this needs to be a client component, use client, And the reason is that we're using a hook inside the component, so that is just what we need to do. And now it says hello whenever we refresh it, so that is perfectly fine. Cool, so I'll go ahead and just implement some logic to actually fetch these genomes. So I'll say const API URL equals https api.genome.ucsc.edu, and then I'll say /list /UC Santa Cruz, and then genomes, like this. If I just take this URL again and just open up a new tab, we should be able to see the documentation. So we should be able to list out the genomes, and that is what we're doing right here. So that is basically what we're doing as well. If I just go into this URL as well and print that, we should be able to see what it actually returns. So we get out the US or UCSC genomes, and then for each genome we have a description, for example. We have whether it is active or not. And the key for the genome right here, this dictionary key, will then be the ID for the genome. So what I'll do is just define a type which we can use to make the data we get returned from this API type safe in our Next.js application. So I'll call this type, I'll just export the interface. I'll call this type genome assembly from search, because it is something that comes from a search. Then I'll put in an ID, which is a string. I'll also put in a name. I'll put in a default position as well as active. And this default position might actually not be needed, so we'll just leave that out. And then I can just go ahead and fetch this API endpoint. So I can say const response equals await and then fetch, and we'll fetch the API URL. We'll then just check if the response is okay or not. So I'll say if not response dot okay, then we'll throw a new error. Failed to fetch genome list from UCSC API. Then we can go ahead and grab the actual data from the response. So we'll say const genome data equals await response.json. We'll then just check if we can find this key right here, UCSC genomes. So I'll say if, and then not genome data .ucsc genomes. Then we'll also throw a new error saying UCSC API error, missing UCSC genomes. And now that we know that it is not undefined, we can go ahead and say const genomes equals genome data.ucsc genomes. And since genomes can be genomes assemblies for different organisms, so right here it's an American alligator, for example, this is a lizard, this is also a lizard, we'll need some way to handle that. So in case you would like to work with other organisms, we will make the system so it's able to handle that. But for our case, we'll only work with humans. But it's still, we'll like to create a record where the key is going to be the organism and the value is then going to be the data up here about that organism. So I'll say const and then structured genomes of the type record, where we have the string, which is the organism, and then we have the genome assembly from search. So each organism can have multiple of these genome assemblies over here. And then we'll just say that that is equal to an empty dictionary for now. And then we can go ahead and populate this dictionary in a loop down here. So I'll say for const genome ID in genomes. We can then go ahead and get the genome info. So I'll say const genome info equals genomes, and we'll index in with the genome ID like this. The genome info will then be this dictionary right here. So the content of the dictionary in value. So first we'll just grab the organism. So I'll go ahead and say const organism equals and then genome info dot organism, or other if that for some reason isn't there. So this will grab the following key and value right here. Then I'll push this info to the dictionary up here. So I'll go ahead and say if not structured genomes and then organism. So if the organism hasn't been added right here yet as a key, then I can say structured organism or structured genome, organism is equal to a new array. So then we'll add this record right here, but just make this array empty. And this of course needs to be if. Then below this, I'll say structured genomes and then organism dot push. And we would like to push a new genome assembly over here. And the genome assembly contains the ID, which is the genome ID. It will contain a name. We'll just say that is the genome info dot description. So description right here. And I'll just make sure this genome info is spelled correctly, like that. Genome info dot description. We'll also say that if this is not there, for example, then we'll just call it the genome ID. So otherwise, we'll use the genome ID as the name. Then we can go ahead and define the active or not. So this will just be genome data, the genome info dot active. And over here we can see Boolean can't be string. So of course active needs to be a Boolean and not a string. And the reason there are 2 exclamation marks right here is to convert the 1 over here to a true. So it'll evaluate the 1 to a true. And if it's 0, it'll just evaluate to false. So that is basically what happens with these 2 exclamation marks. Good. So with that done, I'll just add one more thing, and that is the source name. So if I say HG38 down here, there's also a source name that is a little bit more in depth about the name. So I'll go ahead and just add that as well. So I'll say source name, and then down here I'll say source name equals genome info dot, and then I can do source name. And if it's not there, then we'll just say genome ID right here, just so it has a fallback value, even though it should be there all the time. The only reason we're doing this is because we can't, in fact, be 100% sure that all of these keys are always present. So we just do this just to have some fallback value in case that happens. Good. So hopping back into the page.tsx, now that we return, we don't actually return anything yet. So I'll just go ahead and under this for loop say return, and then genomes, and this will just be structured genomes. Good. Hopping back here, I can then say console.log data, and then we can just check out whether we are getting something we would expect. So hopping in here and just refreshing, we now see genomes on load, and this contains a bunch of different genomes. So if we just print out the ones that are human only, we can do this, human, but we of course just need to say .genomes first, print that out. I'll just refresh over here, and now we'll see undefined. So hopping back into the Genome API, what we need to do is just check whether this indexing key is correct. And right now it says genome data.organism. So hopping in here, it's actually going to be genome info. So genome info.organism, that will give us the organism right here, and we can use that as the key for the dictionary. Good. Hopping back and refreshing, we now see that we have 6 printouts after we fixed the error. We have these 6 different genome assemblies for the human organism only. If I just remove this key, we should be able to see all the different organisms. So we have African clawed frog, we have American alligator, for example, all with the genome assemblies inside of it. So inside this component, we'll create some use states. We'll create a state for loading. So we'll say const and then is loading. And then set is loading as well. This will just be a global is loading state for all the fetches that we'll have inside of this component. We'll say equals to use states, and this will just be a boolean. It might infer this itself. This will just be a boolean, so we'll just set it to false for default. Good. We then also store the genomes inside a use state. So we'll say const genomes set genomes is equal to use state, and this will be genome assembly from search and an array of those, and this will just be an empty array per default. And up here we just need to do this, so we say type in front of the actual interface. And then we'll also just create a use state for storing an error that might happen if this doesn't work. So we'll say error and then set error, and this error will just be an empty string for now, or actually we'll make it string or null. So string or null. So if there's no error, it will be null. Otherwise, it will be a string. So it'll just be null per default. So what we can now do inside this use effects is the following. We can say try, and then we can say catch error. And then error like this. We can then say set error if there is an error, and this error will just be failed to load genome data. We won't really be able to use this for anything, and we'll just show the user the following message right here. Then we can say finally, and so then the first thing we'll do inside the try is set is loading to true. Then we can in finally, so regardless of whether the error happens or not, we can say set is loading to false. Right inside this try, we'll then say const data equals get available genomes. Then we can say if data.genomes and data.genomes human. So if there is the human organism, then what we'll do is set the genomes to the following right here as well. So this should then load all of the genomes for us on load whenever the page reloads over here. So that is exactly what we want. Then I'll also just create another use state up here. This will be called selected genome. And I'll then say set selected genome. This will then just be a single selected genome. So I'll just use the string. And then the one that should always just be selected is going to be HG38. So that is just a default one. And that should always be available. Good. So hopping back over here, if we scroll down to just below this card header, we'll just put in a card content 2. Inside this card content, we'll put in a class name, which is padding bottom 4. So we'll just need to do this, padding bottom 4. And then for the card content, what we'll put in is going to be a select. This select will just need to install from Shadcn. So I'll open up the terminal and just open a new one, search for Shadcn. We can actually just do it in here. So in here we'll find the select and then just find the install command, paste it in, use force, and then just restart the development server. So I'll just go ahead and open up this menu again, press def, and close down the terminal once again. We should now be able to then import the select over here. So I can say select, and then just import that under components UI. And then for the card content, we can just import that as well. The value of this select will then be the selected genome from the use state up here. So this will store what genome is selected. On the value change, what we'll do is just call a function called handle genome change, which we haven't implemented yet. And the reason we will call this function instead of just calling the set selected genome from the use state is because we might want to do some other state changes as well whenever a genome is selected. So scrolling up here, I'll just create a new function, handle genome change. This will just accept a value string like that. And then we can say set selected genome for now and just set the selected genome to the value. Scrolling back down, this select should be disabled if we are loading. So disabled if is loading. And if this is loading will be set whenever the genomes are being fetched. So obviously you shouldn't be able to pick between different genomes when they aren't even fetched yet. So that is kind of the logic behind this disabled functionality. Then if I just save over here and refresh the page over here, we should be able to see a select in just a second. So first I'll just put in a select trigger. So I'll say select trigger. Inside the select trigger, I'll then put in a select value from components UI select as well. This will also need to be imported from components UI select. The select value should then have a placeholder. So I'll say placeholder select genome assembly. And I'll press save. And then for the select trigger, I'll give it the class names h9 with full border. And then for the border, I'll give it hashtag free, C4, F3, and D. And then I'll cut it down to 10% opacity. And then for the actual contents of the select, and I'll just make this a single select value instead. For the actual contents of the select, I'll say select content from component UI like that. And then inside the select content, I can go ahead and say genomes.map and loop through all of our fetched genomes. So I can get the genomes like this. And then I can return a select item. This select item should have a key because we're inside a map. We'll just need to import the select item. The key should then be the genome. This will need to be genome. It will be genome.id. The value of this select item will be the genome.id as well. And then for formatting how it should look, we can go ahead and say genome.id and then dash genome.name like this. Press save. So now you see what this looks like, as well as I'll just copy this. And then down below, I'll say genome.active. If it is active, then what I'll do is say active. Otherwise, I'll just put in an empty string. So now you can see that these ones are all active, so that is great. Right below the select down here, I'll just put in a conditional, and then we can show the source name. So I'll say selected genome. And if a genome is selected, I'll just return a paragraph. This paragraph should say the source name, so I'll just say source name for now. And then for the formatting, I'll go ahead and say class name, mt2, text extra small, text, and then hashtag free. I can then say C for F3 and D, and then I can make it 60% opacity like this. And we can then find the genome for the genome that has been actively selected as well. So what we'll do is just copy this line, then say genomes.find, and then we can say genome like this, where genome.id is equal to selected genome, and then we can say dot, we go ahead and say source name. So I'll just go ahead and paste this back in, and then copy this entire thing and paste it in instead of source name like this. And then the paragraph statement might have to be inside of this. If I then press save, we should then be able to see the source name just below the picked genome. So that looks great. And let's now just test out the picker as well. So if I just click the picker, we can then see that it switches down here as it is supposed to do. And we can then switch between these different genomes. So it seems to be working just fine. So hopping into the Excalibur, we can check out what the next step is. So now we have loaded the genome assemblies. We can pick between genomes. So now I would like to load the chromosomes. And then after that, let the user switch between picking genes or searching for genes and then picking genes that are part of specific chromosomes. So we'll just implement the functionality right now for just loading the chromosomes and then hop on to this next part right after. So the first thing I would like to do is just hop into the Genome API file and then create a new function that is also asynchronous that will then fetch the chromosomes from also the UC Santa Cruz API. So since we already have the ID of the picked genome, we can then fetch the chromosomes from that API as well. So I'll start with just copying this method right here, or at least some of it, call it get genome chromosomes. And for the input, we should receive a genome ID that is a string. And then for the API URL, we'll need to make this a formatted string like this. And then we'll call, instead of list USC genomes, we'll say list and then chromosomes, where we pass in the query parameter genome equals, and then we'll pass in the genome ID. And then down here, we'll say failed to fetch chromosome list from UCSC API. And then we'll follow kind of the same structure as down here. So first we'll check whether the chromosomes even are within the list. So we'll just do this and just browse chromosomes first of all. And then we'll just add the query parameter. So genome equals, and then we'll say HG38. Pretty print that. And now we should be able to see what it returns. So if it doesn't contain this chromosomes key right here, what I'll do is just say missing chromosomes. Then I'll just add a new type up here as well. So I'll say export interface chromosome from search. This will contain a name, which is a string, and also the size, which is a number over here. So those are the two things you see right now. So I'd like to avoid chromosomes that contains either this underscore right here, since they aren't very telling of what the chromosome actually is, or chromosomes that contain this UN, doesn't really know what that is either, or chromosomes that contain random. So to do this, what I'll do is after this, just say const chromosomes, which is a list of chromosomes from search. Then I can say for const chromosome ID, like this, in chromosome, and we just need to rename this up here. So this needs to be chromosome data. Then we can say chromosome data.chromosomes dot, and then we'll say dot chromosomes. So we'll loop through all the chromosomes, and then if the chromosome ID dot includes an underscore, or it includes UN, so I think this means unknown, or it includes random, well, in that case we'll just continue. Otherwise, what we'll do is say chromosome and just chromosomes dot push, and we would like to push the name, which is the chromosome ID, as well as the size, and that is just going to be chromosome data dot chromosomes, and we would like to index in with the chromosome ID, so that is the ID right here. This means we'll get out the size. And then I would like to also just sort the chromosomes server side here as well. So I'll just do a natural sort. So this means that they'll come out as chromosome 1, chromosome 2, all the way up to chromosome X, chromosome Y, etc. So I'll just do ... Then you can just see what we're doing. So chromosomes dot sort, and we'll do a custom sorting. So I'll say A and B, those are the two elements which we'll pick between during sorting. So first, const, we'll just check whether A is a number and whether B is a number. So we'll say A dot name dot replace chromosome with nothing. So we'll remove this first chromosome part, and this way we can check whether this is a number or not. So this is then for A, and this is for B. Then we can say const is number A. This will be a regex, so I'll go ahead and do this, backslash digits. This will then let us test whether it is a number or not with a simple regex. So this regex you don't really have to understand that much. Just know that it checks whether this string right here is only digits or not. This will then just be for B instead. And for B, if is number A and is number B is then true, then we'll return number A number minus number B number. So if both of these are numbers, we can then sort the chromosomes by the difference between the numbers. So we have 1, 2, etc. So it'll just be an increasing sort. So in this case right here, A might be chromosome 1 and B might be chromosome 2. And it'll then be able to determine that chromosome 1 comes before chromosome 2 because both are numbers. So we can then just take the difference. Well, let's say A is then chromosome 1 and B is chromosome X. Well, then only chromosome 1 will be a number and this won't be a number. So we can say if, and then is number A. So if only A is a number, because this case will be hit up here if both are numbers. Well, in that case, we'll return -1. And this will let the sorting function know that the A element up here should become first in the sort order. Well, if it is the opposite case, so if B is a number and A is not a number, then we'll let it know that B should come first in the sort order. But if it is comparing then also chromosome X with chromosome Y, well, that is another case for itself. So in that case, we'll just compare them lexically, so alphabetically. This will mean that X will come before Y. So in this case, we'll say a number dot local compare, and we'll compare it to B number right here. And then we can go ahead and just return the chromosomes like this in the sorted order. And then hopping back here, we should be able to scroll up and then fetch those chromosomes whenever a genome has been selected. So we'll create a new use effect, and then whenever one of these genomes is selected, this will then re-render the component and fetch the the chromosomes. So this should only happen when selected genome changes up here. And then this will instead be fetch chromosomes. We'll call this fetch chromosomes as well. Here it'll be get available chromosomes. We'll import that. Let me just see if we named it correctly. Get genome chromosomes. We'll pass in the selected genome. This will be failed to load chromosome data. The result of this should then, let me just import that. The result of this get genome chromosomes should then be stored in a use state up here. So I'll say const chromosomes and then set chromosomes is equal to use state, and the use state should be chromosome from search and array of those. And then I can also say selected chromosome. So I'll just copy this up here, call it selected chromosome, set selected chromosome as well. And then this will also just be a string, and this will just be CR1 for default. So the following chromosome right here, so just the first one. And up here, I can then just import chromosome from search correctly by appending type or prepending type to the import statement. I'll then say whenever these chromosomes has been fetched, I'll then just set the chromosomes to data.chromosomes. What I'll then also do is say if data.chromosomes.length is more than 0, which it'll always be basically, but just to make sure, I'll then say set selected chromosome to data.chromosomes, the first element, .name. So if I remove this exclamation mark right here, this might be undefined, but we know that it won't be undefined since we have checked that the length of the list is more than 0, but the Typescript compiler doesn't quite pick that up, so that is why we put in the exclamation mark. And this should then fetch the chromosomes whenever the genome changes. So what I can do is just say console.log and just log the data.chromosomes, and then hopping over to our application and just refreshing. If I then do select a specific genome like this, then it selects the chromosomes for that genome assembly. So maybe this one as well. And then selects genomes still. So it has the name and the size just like we wanted it to be. So the chromosome fetching logic seems to be working out just fine. So now we can move on. So hopping back into the SCALDRAW, we can just check out the next logic. So we need to be able to search genes manually with a search query, or search for genes in specific chromosomes by clicking specific chromosomes, for example, from the ones that we have just fetched. So what I want to do is just use the tabs component within Shadcn right here. So I'll just find tabs. Then there should be two different options. There should be a search option, then a chromosomes picker option. So first I'll start out by just installing the tabs. So I'll open up a new terminal, install tabs. And whenever this is done, I'll then go ahead and restart the development server. So I'll open up files again, click dev, and then just close down the terminal once again. And then I'll hop back into my application and also scroll up, and then under selected chromosomes, I would like to add a new use state, and this is for the search query. So if the user decides to click this search query tab, then the user should be able to enter in their search query, and this should obviously be stored somewhere. So we'll store this in a use state. So I'll say search query and then set search query equals use state. And this will just be a string, and I'll just set it as an empty string for now. And then scrolling down to right beneath the card we have right here, I should be able to put in another card. So I'll say card, and then I'll say class name MT6, a gap of 0, border none, BG white, PY0, shadow small. Then I'll save that and just open up the card. Inside the card, I'll put in a card header. The card header should contain a card title, like this. The card title should be browse. And I'll just refresh over here just to make sure that we can follow the progress. So now we see browse. the card header, we then just need to put in some class names, padding top 4, padding bottom 2, so it looks like this. For the card title, I'll say class name, text small, font normal, and then text with the styling, hashtag free F or free C for F, and then F free D at 70% opacity, like this. And I just put in one F too much, so if I go ahead and remove that one F, it'll look exactly like the card title up here as well. Then for the card content itself, I'll say card content, and the card content should contain the tabs. So first I'll just say class name, padding bottom 4, then I'll say tabs, which we just installed from Components UI. Inside the tabs, there'll be a tabs list. Inside the tabs list, there'll be a tabs trigger. And inside the tabs trigger, it'll say search genes. So that is one tab we have right here. And then I'll just need to create a use state for storing the state of the tab that is currently active. So scrolling all the way up here, I'll just say const and then mode, set mode equals use states. And then we'll just create a new type called mode. So up here I'll say type mode equals either browse or search, like this. So I'll then say mode, and then the mode should be search. Scrolling back down, if I hop into tabs, so in here we can set the value to the mode, so that is the use states. And on value change, what we'll do is just grab the value and then say switch mode, and we'll just need to create a function for that. So scrolling all the way up right here, we can just say const switch mode equals, and then we'll accept a new mode, which is of type mode. And then if the new mode is equal to the current mode, we'll just return, because we can't really switch to the mode we're currently on already. Otherwise, we'll just set the mode to the new mode like this. Then scrolling back down, we can then use this switch mode function where we pass in the value as a mode, which it always will be. Then for the tabs list, we'll say class name, margin bottom 4, bg hashtag, let me just finish it like this, then it'll be hashtag E9EEEA. And for the tabs trigger, I'll say value, the value will be search. So this value right here is what will be the following value right here when the value does change. So since this value is search, which is one of the two options in search in mode right here, it'll then always also be either search or mode. So that is why we can say as search right here. We just need to make sure that with the other tabs trigger as well, the other tabs trigger needs to say browse and not anything else than browse or search. So I'll also change the styling a little bit based on the active state. So I'll say data and then states equals active. And then if that is the case, I'll say bg white. I'll also say state. I'll also say data and then state active again. If this is the case, I'll then say text, and then I'll change the text just a little bit to hashtag free C for ref, and then free D, like this. Then I'll just copy this tabs trigger, and right below I'll say browse chromosomes. And then right here, this value will then be browse instead. So it'll look like this. Then for the tabs content, we'll first of all have a tabs content for the search. So I'll say it's tabs content. Value will be search. The class name is going to be empty0, like this. Inside this, there'll be a search bar where the user can search, and then also a button to actually do the search. So I'll start by putting in a div. I'll then say class name space y4. Inside this, I'll put in a form. In the form, I'll have an onSubmit. This will contain a method for actually doing the searching. I'll just put in a empty method for now. So outside this onSubmit, I'll just put in a class name, say flex column, gap of 3 between the items vertically, small, so on large screens, flex row. Cool, and then inside the form, I would like to put in another div. I'll give it the classes relative and flex1. Inside this, I'll put in an input. The input we'll get from Shadcn. So we'll just hop back into Shadcn, find the input over here, right there, and just install that input. So I'll open up the terminal, install the input, and just restart the development server right after that. So that is what I'm doing right here. And then I should be able to refresh over here. And now we should still see the input server, the development server running. This input would then be of type text. So I'll say type text. The placeholder is going to be, I'll just close it and also just import that. The placeholder is going to be enter gene symbol or name. Under this placeholder, I'll put in a value of type search query. So going up here, we can just see what we call this. So search query. So I'll just put that in, and then we can say on change. We'll then say we'll receive the event, and then set search query to event.target.value. And then we'll also just style it a little bit. Class name h9 border of a hashtag 3c4f and then 3d. Styled down or scaled down to 10% opacity, and then a PR of 10. Then what we can then do is put in a button into this. So I'll say button from Components UI as well. We'll put a search icon into the button. So I'll say search like this, and then just close that. Give it the class name h4 with 4, along with a span, and this span should just say search like this. And I just made a small mistake. We just need to close down the input right there, and now we see this instead. And then for the button, I'll put on some classes. Absolute, I'll put on top 0, right 0, age full, cursor pointer, rounded, large none, left none, background, hashtag 3, and I'll just use the same one as right here, like that. I'll put on text white, and then hover. In that case, I'll say background, put on the same one again, and then just scale it down to 90% opacity. And then I'll make sure this button right here is disabled so I'll say disabled if is loading, or there is nothing in the search query, so if not search query.trim. And the size of this button should then just be icon, like that. And then we should have a type, which is going to be submit. Then this search text right here will just go ahead and set to screen reader only. So I'll just say SR only, and then it'll be the button the icon right here. So if I enter something into this, we should be able to see the button. So it won't really do anything now because we haven't made it do anything when the form is submitted, but we're at least able to use the search input field. And then I just want to make the user able to load in an example. So this should just set the input field search query for the user. So this button will say try BRCA1 example, like this. And then we'll just style the button to be variant link, like this. And then for the class name, we'll say hauto, cursor, pointer, padding of 0, text, and we would like an orange text. So we'll say hashtag DE8246, and then we'll just copy this color right here, and then say on hover, then we'll change the text to become this, just with 80% opacity instead. So it'll look like this. And then we'll just create a method for handling actually clicking the button. So hopping up here, we can say const load BRCA1 example equals a new method where we say set mode. So we set the mode to search, because you can't really click chromosomes and then load this BRCA example, which is a specific gene. This specific example by searching for this BRCA1 gene only makes sense if you are on the set or search genes tab. So after this, I'll then say set search query to BRCA1. So it'll just search for this for the user. And then we of course just need to handle the actual searching. We haven't really implemented that yet, so we'll just leave a comment about that for now. Going down to the button again, we should just put in an onClick. So onClick, we'll then just call this method right here. So clicking it then just sets this BRCA1, and then we can actually go ahead and search for that. We'll just create another method up here as well. So if I scroll up, then I can say const handle search, and this is equal to an async function that takes a React event. So React.form event. Then we'll say if event, then event.prevent default. So this is what we'll call whenever we submit the form. Then we'll say if not search query.trim. So if there is no content in the search query field right here, then we'll go ahead and say return. And we're just missing the rocket notation to make sure it's actually a function. And then when this has been done, so we have extracted the search query, also we made sure that we have a search query that actually exists, then we can do the search, gene search actually. So we'll say perform gene search, which we'll do in just a second after having implemented the tab content for the browse chromosomes as well. But for now, I can just take this handle search and scroll down to the form, which we defined right here, and then just pass that in as the handle search for the onSubmit. And then for this handle search, we can actually also just call it up here. So we'll go ahead and do this, and then just make this React form event optional, because we check for whether the event is there or not anyways. And then for the tabs content under the browse chromosomes menu right here, we need to implement that as well. So we can just scroll down to tabs content. I'll just copy this right here and then paste it in. And then this should be browse instead, because we browse different chromosomes. And then I would like a list of chromosome buttons. So first I'll put in a div, and I'll say class name max height 150 pixels, like that. Then I'll say overflow y auto PR1, and then put in a div that actually wraps around the a div. And then the class name should be flex and flex wrap, and this will wrap the buttons around. So if there are more buttons than the screen width allows, then they'll wrap to the next line. So I can then say gap2. Under this, I'll say chromosomes.map, and I'll get the chromosome out like this. And then for each chromosome, I would like to render out a button. So I can do a button like that. And then for the button, the content, so the text should be chromosome.name. So these are then all the chromosomes we can pick between. Then for the key, because we need to set a key when we're inside this map function, we'll say chromosome.name, because they'll be unique. It wouldn't make sense for each of these buttons, or for any of these buttons for that sake, to have the same chromosome anyways. Cool. So after the key, we can say variant, and then outline, like that. Then we can say size small. We can then say save. Then we can put in a class name. The class name should be a class name that allows for optionals, like this. We'll then say height of 8, cursor, pointer. We'll put in a border with this color right here. So I'll say border, put in the color, add opacity 10%, and then hover. In that case, we say background, E9, E, and then we'll say hover, and then text, and then just put in the background color once again. So that'll look like this. And then when you do hover it, you see the background color changes. And cool. Then for the optional, what we'll do is say if there is a selected chromosome that is equal to the chromosome.name. So chrome.name. So the one that's currently selected will highlight in some way. So if that is the case, we'll then say background color hashtag E9EEA and text in the color we always use. Otherwise, we'll put in an empty string like this. So this one is currently selected. Then you can click another one. We can't do that right now. We need to actually put in the onClick. So I'll just say onClick, set selected chromosome, and then I'll just select chromosome.name like that. So when I click one over here now, we should be able to select different ones like we are. So that is great, and you can see which one is selected. So it seems to be working out just fine. Then we can just put in an error state and also a loading state now. So if we just set error to null or test error, and we set is loading to true, we can now just put in those two states under these cards, under these tabs. So this global loading state, both for searching for the genes, but also for loading the genome assemblies, should just be shown in this card right here, which is kind of the main card on the page. So under the tabs, I'll say is loading. If we are loading, so if is loading is true, then we'll return a div. This should have the class name flex, justify center, py of 4. Inside this, we'll put in another div. I'll just save this. And this should just be an empty div, but we'll put in some classes that makes it a spinner. So height of 6, width of 6, animate spin. This comes from Tailwind, by the way. Rounded full, border 2, border, we'll put in the same color at 30% opacity, and then a border top of, and this will be an orange color, so DE82 46. And I'll just need to make this a color. And then I think this is loading state was overridden somehow by set is loading up here maybe. So I'll just go ahead and remove these statements, setting it to false, and just reload over here. And now we see the loading state that just spins around like this. So that seems to be working just fine. Now, if I just save this, and then up here at the loading state, I just set loading to false by default. Then if I do refresh the page, it loads for just a second whenever this genome assembly is loading. So now the loading state is working, we can go ahead and create the error state. So the error state will be a little bit like this. So I'll say if there is currently an error, then we'll return a div like that. This div will contain the error message, so test error, and the styling will be MT4, rounded medium, border red 200, BG red 50, padding of 3, text small, text red 700. So an error that you'll see will look exactly like this. But of course we don't need to show the error every time, we just need to hide it again. So it's only shown if there is actually an error. So I'll go ahead and just insert a null again, and then the error should be gone. Okay, let's just create the function in here for actually searching for the different genomes. So this function will say, for this function will say export async function, and then search genes. And then we'll pass in a query, which is a string, along with a genome, which we're searching within. And that is also going to be a string. So this genome we're searching within is going to be the genome assembly that has been chosen. And then for the API we'll be working with, we'll say const URL equals HTTPS, and then we'll say clinical tables dot nlm dot nih.gov / and then it's going to be API ncbi underscore genes underscore version 3 and underscore search. So just hopping into the base URL of this right here, we can just check out what it actually is we're using. So what this is a public database we can use to search within genes. So what we're going to do is use this NCBI genes right here. So if I click that, we'll be able to see what we need to do to actually search within these genes. So right here it says the process data only includes the human gene data from NCBI's gene dataset, and because we are constraining our software to only working with the human organism, that is perfectly fine. It is also provided as is and free of charge, so we can just call this API without any problems and without any authentication tokens as well. For the genes you are searching for, you can get all of these fields out, and you specify whenever you call the API which ones of these you want and which ones you do not want. So what we'll do is hop down here to under the URL and then say const params equals new URL search params. And then we'll say terms query. We'll then also say df, and these are called display fields. So in here we'll put chromosome, we'll also put symbol, we'll put description, we'll also put a map location, and a type of gene like this. Then I'll just copy this line to right below it, and then I'll call these EF, so for expanded fields, and then I'll also just add genomic info and gene ID like this. And this should allow us to retrieve the following properties down here, or the following fields. So I can then say const response equals await and then fetch, and we'll then just fetch the URL, and then we'll put on the query parameters as well. So I'll say params, and then I can say if not response.ok, then we'll throw a new error, which will just be something like ncbi API error, like this. And I'll just make this inside the curly braces so it looks a little bit nicer. This needs to be URL, for example, as well. And then after having done this fetch, we can just go ahead and get the JSON from the response. So I'll say const data equals await response.json. Then I can say const results. This will have to be a type which we'll make ourselves, and then this is just going to be equal to an empty array for now. So hopping up, we can just go ahead and define the type. So we'll say export interface gene from search. This will contain a symbol. A symbol will be a string. It will contain a name that's going to be a string as well. It'll also contain a chromosome, a description, as well as a gene ID. And this gene ID we can use to get more information about the specific gene whenever you do click a gene and is navigated to that page as well. So now we can say that this list is of type gene from search in an array. So hopping over here, if we scroll down to the API documentation, we can see that this is a sample API query along with the result that we are getting back. So right here it says returns all 5 genes matching MTX. The number 5 is the total amount of available result count, and that is the number 5 they're talking about right here. So first we'll check for whether we have an amount of results coming back. Otherwise we don't really need to get all of these specific keys out of the results if we aren't even getting any results back. So I'll say if, and then data, and then 0, because this was an array, is more than 0. Well, then we'll have to get the specific properties out. So I'll go ahead and say const filled map is equal to data 2. So over here, if we just take a look at the description, we see that this is index 0, this is index 1, and this is index 2. So right here we can see that the subsequent null means that no extra parameters were requested via the EF parameter. So we actually passed in this EF parameter right here with the gene ID. So this null should not be null for our request, so we should be able to actually get this extra requested data out of that. So that is why I say data 2 right here. Then I'll say const gene IDs is equal to filled map.gene ID, or just an empty array. Then we can loop through the results. So I'll create a classic for loop for let I be 0, and then if I is less than, and I'll say math.minimum. So I'll process at max 10 different results, and if the data contains less than 10 results, I'll just default to that. And then I'll say plus I. And then we can get the display properties. So I can say if I is less than data 3.length. But first we'll just do this little check to just make sure the for loop doesn't run out of bounds. So if the for loop runs for 10 times, for example, but that there are only 5 different results returned, we won't really be able to get any results out of the last 5 iterations in the for loop. So we just want to make sure that we don't run out of bounds with this little safety check. And then again, we'll also put this into a try catch, since we don't really know that much about the APIs. We want to make sure that we always have the props available. So I'll say in the catch continue. This means that if we get an error trying to access some of these fields, we'll just continue and skip that specific record. Cool. So I'll then say const display equals data free I can then say let the chromosome be equal to display 0 if the chromosome and not chromosome like this dot starts with chromosome will then we'll say chromosome, and I'll just put this into brackets, chromosome will be equal to chromosome like this. So what we do is if the chromosome is just a number such as 22 for example, then we'll just append or prepend chromosome to that number such that it says chromosome 22 like it does right here for example. So this is chromosome 2, so we don't want this to just be 2 for example. So we just append chromosome to that. Hopping back in here, we then just push these results to the results array. So I'll say results is equal to, dot push. And then I'll push the symbol, which will be display 2. We'll push a name. This will be display 3. We'll also push the chromosome and a description. So the description will be display 3 again. So this will be the same as the name. And then for the gene ID, we'll set this to gene IDs at index AI, which we're currently at. Otherwise, just an empty string. Then if we hop outside this if statement right here, we can then say return and then query, genome and results. And then hopping back into the page, if we just scroll up to under this use effect at fetch chromosomes, we'll just create a new function called perform gene search, which is an async function that takes a query, that is a string, a genome, that is a string. It'll also take a filter function. So this allows us to filter the gene search results after they have been fetched for the API. So how we're going to do this is by just saying filter function. This should be optional because we probably don't want to use it all the time. We'll then say gene from search, boolean like that. Then we'll also just open up the function. So I can go ahead and say record notation, open up the function. Inside this, I'll put in a try along with a catch. Inside the catch, we'll just say set error message. So we'll just use this global error message, error like that. We can just say failed actually to search genes. And then we'll just put in a finally. Inside the finally, we'll set is loading to false like this. And then we'll set is loading to true inside the try as the first thing. Like this. Cool. And then inside this try, what we'll then do is say const data is equal to await search genes, where we pass in the query along with the genome. This just needs to be written in the correct way. After this, we can then say const results is equal to, if we do have a filter function, then we apply that. So then we say data dot results dot filter, where we pass in a filter function. Otherwise, we just say that results is equal to data dot results. Then we can just set a search results use state. So up here, we just need a use state to store all of these search results. So under search query, I'll say const search results set search results is equal to use state. And this will be an array of gene from search like that. And then if I scroll down, we should be able to just set these down here. So right about there, we can then say set search results and set the results like this. And when do we want to actually perform the gene search? Well, one of the cases is whenever a chromosome is selected. So we'll say use effect and just listen for selected chromosomes. So we'll pass in a dependency array. So we'll say selected chromosome, selected genome, as well as the mode. Then we can go ahead and say if not selected chromosome or mode is not browse. So this should only happen for the chromosomes, and that is when the mode is browse. Well, in these two cases, we'll just return. Then we can say perform gene search, selected chromosome, selected genome, and then we can pass in a filter function where we say gene from search, where gene.chromosome is equal to selected chromosome. Okay, so the reason we are actually passing in this filter function into this perform gene search, which then also filters away genes outside the chromosome after having performed the API search, so this one in here, is because this API call, which we perform right there, performs a broad search based on this search term, and this happens to be the chromosome name in the browse mode. But this also actually might return genes that aren't actually on chromosome 7, for example, if that is what you search for. So we do some client-side filtering to make sure that the genes that are returned are also actually filtered to the chromosome which was searched for, to make sure that only genes within the chromosome which we're searching for is actually shown to the user whenever they use that filter. So with that out of the way, let's just go ahead and find out where else we need to perform the gene search. So whenever you change the gene, whenever you handle the search down here, so that corresponds to if we hop into the code, and then you search for something like this, and then you click right here to search. Well, in that case, we'll need to search for genes. So we'll call perform gene search, pass in the search query, as well as the selected genome, like that. And then whenever you click this browse chromosome, I also wanted to search for genes because this chromosome 1 is automatically selected. It doesn't really make sense that you have to select a chromosome before anything even pops up. This should just search for genes automatically, such that you can see the results for chromosome 1. So I'll hop down to switch mode, that is actually up here. So whenever the user switches the mode, I would like to first of all immediately clear the search results. So set search results, I'll immediately clear that list to make sure the user doesn't see any search results from the other pane, for example. And then I can also say set error and also clear that, because now it is a new browser, a new way of browsing. So if you're here and you switch to this, you don't want to see the error from this search genes pane, for example. So we'll just clear those two, and then we'll say if new mode is equal to browse and select chromosome is actually there, then we'll say perform gene search, and we'll just copy the exact same thing from up here so that it will be these 4 lines. And I'll paste these in right there. And then if we hop into perform gene search and just print out the results, console.log results, hop over here, refresh the page, and I'll just open up the console like that, and then we can try and load the BRCA1 example and press search, and now we see these genes that were searched for. If I click browse chromosomes, they also load automatically, and when I switch, they also load automatically. So all that there's left to do now is just show the genes. So I'll just close down the terminal and then scroll down again, all the way down to below this error. And then I can say search results dot length is more than 0. So if any search results are there and we are not loading, then we'll say and like this. And this just needs to be put in a parentheses. Then we'll return a div. This div should have the class name mt6. Inside this div, I'll just put another one. This div should have the class name margin bottom 2. Inside here, I'll then put in an h4 and say if the mode is equal to search, then we'll return a reg fragment. Otherwise, we'll just return another reg fragment like that. And then inside this reg fragment, we'll say search results, put in a forced spacing like that. And I'll then just write out how many search results were found. So I'll say search results dot length, and then I'll say genes, save that, and then we can just format this a little bit. I'll just refresh over here to make sure that we actually see what we're supposed to see. So if I just load this BRCA1 example and press search, we can now see search results 7 genes. For the H4, we'll then say text extra small. We'll say font normal. And we'll just say text hashtag 3C 4F, and then we'll say 3D at 70% opacity, like this. And then we'll just take this text and copy it, hop down into the span, say class name, font medium, paste this in again, and just remove this opacity. So now it'll look like this. So this was for the search. Then I'll just copy this right there, paste it down into what happens if we are in browse mode. So if we're looking for chromosomes, so we'll say genes on, and then we'll show the selected chromosome. So selected chromosome, and then I'll say found right here instead. So hopping into browse chromosomes, genes on chromosome 1, 7 found. Then I would like to show a table with all of the different genes. So first I'll hop into Shadcn, just find the table component, that one right here, and just install that. Terminal, new terminal, and then just restart the development server just because we added new files. Close down this terminal again, and then we should be able to first put in a div. This will just serve as a container for the table, making sure that it doesn't overflow in the screen, for example. So I'll say class overflow hidden, rounded MD, border, and then a border color of 3C 4F 3D, like that, at only 5% opacity. So hopping back in here, we should be able to refresh and then just search for something. And now we see this border right here. So in here, I can just open up a new table element. So I'll say table from components UI table. And then inside here, I'll put in a table header first of all. This table header should be table header and table header right there. And I'll import those. Then I'll say table row. The table row should have a table head. Inside the table head, we'll have a symbol first of all. We'll also have, let me just style this first before copying it. So I'll say class name, text, extra small, font normal, as well as text, and then just the same text as up here. So I'll paste this in at 70% opacity. Then I'll just take this table head and copy it twice and call this name and this location. So now we have symbol, name, and location for each gene. Then for the table row styling itself, we'll just say class name, BG, and then we'll say E9, A at 50%. Then we'll say hover, BG, and then we'll do the same, but just cut it down. Increase the opacity a little bit, so maybe like 70. And then for the table body itself, so table body, inside this, oh, I just made a spelling error. Table body, inside this, we can go ahead and say search results, dot, and then we can say map, and then we can get each gene and the gene's index, and then render out the different table rows. So I'll say table row. Inside here, I'll put in a key. The key for the table row should be a string consisting of a gene.symbol, just to make it unique. So we'll make it consist of both the gene.symbol as well as the index, like this. And then for the table row, we can then say table cell. Inside this table cell, we'll put in the gene.symbol, like that. Import the table cell, and then I can say class name, py2, font medium, and text, giving it the same color again. I'll then just try and search for a gene, so now we can at least see the names here. Then I'll just take the table cell and copy it twice. This one will then just be name, this one will be chromosome. So now we see the different results like this. Then for the styling of the table row, I'll say class name, cursor, pointer, border bottom, and then also a border color. So the border color is going to be a little bit darker gray. So I'll say hashtag 3c 4f 3d at 5, and then we'll say hover background hashtag e9 eea at 50, like this. So when we do hover it, it tells the user that they're able to actually click these. So now we just need to be able to click them. So hopping up here, you obviously need to be able to select a gene. So I'll just create a new one here called const selected gene, and then set selected gene. And this will just be a use state containing a gene from search or null if there isn't anyone selected. Then I can just say null for default value, scroll all the way down again, and then inside the table row we can then just define an onClick. And then onClick will then just set the gene, so we'll say set selected gene, and then just pass in the gene like that. We will also just set the selected gene up here in the handle genome change. So if I just find that right here, then we'll also just set both the search results to an empty array, as well as setting the selected gene to null. So whenever you select a new genome assembly up here, it should just reset the entire state down here. So delete what is right here, or sorry, delete the search results that have currently been there for a previous genome assembly, as well as a gene that you might have selected before. And when you switch your mode down here, we might as well also want to set the selected gene to null, since the only reason you would actually switch the tabs up here would be to select a new gene anyways. Now we can just implement a state that shows the user that no genes were found on this chromosome, for example, if no genes were found during the search. So down here we'll just say if not is loading, and if not error, and search results dot length is equal to 0, and then we'll return some HTML. So this will be a div. Inside here we'll first of all put in a search logo, so we'll just say search like that. So let's say we just put in something that definitely isn't found, like this. Then just save over here. Well, that is what we see now. So under this search logo right here, I'll put in a paragraph saying if you are in mode search. In that case, what we'll do is say enter a gene or symbol and click search. Otherwise, what we'll do is say if you have a selected chromosome, then we'll say no genes found on this chromosome. Otherwise, we'll say select a chromosome to view genes. Cool. And then obviously this looks terrible, so we'll just go ahead and format that. So we'll say class name flex height of 48 like this, flex column items center, justify center to center everything, text center as well, and text gray 400 like this. Then for the search logo itself, we'll say margin bottom 4, age 10. We'll say width of 10 as well, and text gray 300. For the paragraph right here, we'll then say text small and leading relaxed. And this should make everything look much better like you see right here. So this is what you see. So if I just refresh the page, enter a gene or symbol and click search. So that is what has been triggered right here. Then if I click browse chromosomes, then maybe we can find a chromosome that doesn't contain anything. This one, chromosome M, no genes found on this chromosome. That is triggered by the following string right here. So this if condition. Hopping back into search genes, if we just search for something, this works fine. If I put in a bunch of stuff that doesn't make sense, then we just see the empty state again, like we see right here. So right now, when I click this try BRC1 example, it does fill in the search field for me, but I would like to also have it do the searching for me, such that it actually fulfills the entire promise of trying out this BRCA1 example. So instead of calling handle search right here, I'll just call perform search gene manually like this. And then for the search query, this is just going to be BRCA1. And then for the selected genome, that won't really change. So if I now click, it should then do the entire thing for me. So that is exactly what I was looking for. Perfect. And then lastly, whenever a gene has been selected, I would like to go ahead and show a page for that specific gene instead of showing all of this. So in the main right here, I'll just put in a conditional and I'll say if there is a selected gene, then I'll say H1 and then select the gene. And then I'll just say selected gene like this. Otherwise, what I'll do is just return all of this below. And this will have to be within a React fragment. So right here, we also need to say select the gene dot something like name. And then I'll just take everything from the bottom right above main right here and all the way up until this card. Copy that. And then I'll put this inside this React fragment and press save. Now, when I refresh over here, what I would like to see is that when we do select a gene, we then just see selected gene, and then we can render out the entire page for that specific selected gene on this page you see right here. So that is the expected behavior, and that is also what we see. Now, one thing I do notice over here is that it says select chromosome twice. It is actually supposed to be selected genome, such that this code is only being run if any of these changes, and obviously you don't need to put in selected chromosome twice. So I'll just save that, and then we'll be ready to build out this component instead of the hard-coded text right here. And this component will do under the components folder. So I'll say new file, and then I'll call this geneviewer.tsx. In here, I'll create a new component. So I'll say export defaults function, and then I'll call this gene viewer. I'll pass in a gene as well as the genome ID as well as an onClose. So whenever you do go back from that page, well, what happens then? That is what we need to define here. I'll just open it up like this and say gene is a gene from search. And then a genome ID is going to be a string, so that'll be genome ID like this. That'll be a string. And lastly, onClose will just be a function that returns void. Cool. And in here, I'll just first of all return an H1 and then just say gene.name again. So if I take gene viewer and paste it in right here instead, we can now import that. So I'll add the import, say we'll pass down the gene, will be the selected gene. We'll pass down the genome ID, that will be the selected genome. We'll also pass down the onClose. So what happens when you go back to this general page or the first page from that gene page? Well, in that case, we'll set the selected gene to null. So I'll say set selected gene and set that to null. This way, whenever you click the back button inside the gene viewer, it will set the selected gene to null and then stop rendering this page right here and instead go back to this page down here. So if I go back here and then just try out this example, click a gene, now we can see the exact same thing. And then we just need to implement the back button inside the gene viewer. So the first thing I'll do is just change out this H1 for a div. Inside this div, I'll say class name space y6 because we'll have multiple items inside this div. I'll then put in a button from Shadcn. Inside this button, I'll say back to results. I'll also just put in an arrow left icon. So I'll say arrow left from Lucid React with the class names mr2, h4, and then width 4. The mr2 is to distance this a little bit from the text right here. Cool. Then I can go ahead and say class names. Let me just put on a variant first, actually. Variant, and then ghost. And then over here, I'll just refresh and hop into a gene. So I'll click this and hop into a specific gene. And then I can see back to results. Now for the class name, I'll actually make it small as well. So I'll just say size, small. And then for the class names, I'll put on cursor, pointer. And before I save, let me just show you why. So right now, when I hover over here, you can see that the cursor just stays the same. When I save here and then hover now, you can see that it is more visible to the user that this button is clickable. Then I can just put on a text color as well. This will be the usual one of hashtag 3C 4F and then 3D again. And then for the hover effect, I would like to put on the background color of 3 and then 9 E 9 E and then A at 70%. And then when I hover it, it looks like this. And when I do go ahead and click it, I'll be taken back. Oh, we just need to implement that. So we'll just say onClick, and then we'll call the onClose, which will clear the state of the selected gene in the parent component. So it'll call this function right here pass down. Now when I click it, we should be able to navigate back and forth between these two pages. So what I would like to do is that since we have a specific component for a specific gene, and then we pass down the gene as well as the genome ID, whenever you do click a gene, it should then load the data for that specific gene within this child component gene viewer. And then we can show specific information about, okay, what nucleotide is inside this gene, as well as what is the gene called, and more information about the gene itself. And then you should also be able to just create a mutation and test it out with the backend that we deployed on Modal. The first thing I would like to build inside this empty page is a card that shows information about the specific gene. So this will basically be the simplest feature of the page. So what we'll do is just fetch some data about the gene because we have the gene ID right here. So we can say gene and then dot gene ID. So to fetch the gene details, we'll scroll all the way down and then create a new function. So we'll say export asynchronous function. This will be called fetch gene details. It will accept a gene ID of type string and return a promise. This promise will contain gene details of type, we'll just say any, but I'll define it in just a second. Gene bounds. This will also be a type I'll define in a second. I'll just say any for now, and then an initial range. And this will be a type containing a start, which is a number, and an end, which is also a number. This can also be null, like that. Then I'll just go ahead and open up the method and then define these types. So scrolling all the way up, the gene bounds will be pretty simple. It is going to be I'll say interface and then gene bounds. It'll be a pretty simple interface just with a minimum, which is a number, as well as a maximum, which is also going to be a number. So I'll just spell this correctly, and then we'll also define the interface for the gene details. So gene details from search, like this, and this will contain a couple of props. So first of all, we'll have genomic info, which is optional. Then we will have where the gene is placed on the chromosome. This will be called chromosome starts, and that is a number. So remember, a entire DNA genome or the entire genome is broken down into chromosomes, and then specific genes are placed on these chromosomes. So genes will have a start position and an end position in the chromosome. So we'll have a chromosome start and also a chromosome stop. Then we'll also have a strand. So I'll say strand, make that optional, and then just make that a string. And the API can then also return multiple genomic info per gene. So we'll just make that an array. The API can then return a summary. This is going to be a string. It can also return the organism. So that will be a dictionary like this with a scientific name of a string, as well as a common name, which is also a string. And then scrolling down, we can go ahead and use these. So I'll go all the way down, and then the gene details will be gene details from search or null, and the gene bounds will then be the gene bounds or null as well. And then the reason it's complaining is because we aren't returning a promise of this type right now. So first I'll just say try, and then I'll say catch, and then error, and then if an error happens, we'll then just say return gene details as null, gene bounds as null, as well as initial range as null, like that. Inside the API, we would like to first define the detail URL. So I'll say detail URL equals, and I'll make this a formatted string because we need to pass in the gene ID to the URL. So I'll do this, https, and then it'll be eutils.ncpi.nlm.nih.government. And then I can just take this URL, paste it into my browser, and this will give you all the documentation for actually using the endpoints. These documentations are quite old and not very nice to work with, so that is why I just defined the type up here, and that is because I've worked with it before, so I kind of know what it will return. But if you need any more documentation and want to expand on what we're building, you can go ahead and head to this URL right here and then check out the full documentation. So after this, I'll then say /entress /eutils / and this is eutils, not eutils, so like that. And then I can say summary .fc gi, and then I'll put on the current parameters. So I'll say database we're looking in is going to be the gene database, and this will be equal to, and the ID we're looking for is going to be this one, gene ID, and we'll then say read mode equals JSON and save that. And now we have the full fetch URL down, so we can go ahead and say const detail response equals awaits fetch detail URL. Then we can say if not detail response.ok. Then we'll return the same as down here. And then also just say console.error, and then something like failed to fetch gene details, as well as the error message. So something like, I just need to make this a formatted string, like that. And then I can say detail response dot status text. Now when it does succeed, which it probably will do, hopefully, we can say const detail data equals awaits detail response dot JSON, and then convert it to JSON format. Then we can say if detail data dot results and detail data dot results gene ID. Then what we'll do is then copy this right here and say const the details for this gene is going to be the contents of this dictionary value right here. Then I'll say if detail dot genomic info and detail dot, and we'll do genomic info again, dot length is larger than 0. We'll then say const info equals detail dot genomic info index 0, because remember this genomic info up here could be an array, because the API might return multiple genomic infos per gene, so we'll just take the first one. And now we can then grab the position of the gene on the chromosome. So genes can then either be on a positive or a negative DNA strand. So if the gene is on the positive strand, then chromosome start will typically be less than chromosome stop. But if the gene is on the negative strand, then chromosome start might be greater than chromosome stop. And I want the chromosome start to always be the smallest one. So therefore we'll say const minimum position equals math.minimum. And then I'll say info.chromosome starts and info.chromosome stop, like this. And then for the maximum position, that'll then be the exact same. And this will then always be the chromosome start, and this will always be the chromosome stop. So I can then say const bounds equals minimum min pos, and maximum is max pos. And then down here we can say return a dictionary with gene detail. This will then be detail. We can then also return the gene bounds, which will be the bounds. And I would also like to return one more thing, and I'll just call this details. So the last thing is the initial range up here. So later on we'll build another card that is not this gene viewer, that is not this gene details card, but instead another card also shown within the gene viewer, and this card will show the entire sequence or part of the sequence if it is a massive sequence of the gene that we're currently working with. So if we're working with a massive sequence, I would like to only show 10,000 nucleotides within this gene, and if the gene is less than 10,000 nucleotides, we can just go ahead and show the entire gene. So this initial range up here is going to be the range from and to which the nucleotide sequence should be fetched from in this other card. So to calculate this initial range, the first thing I'll do is say const gene size, get that. This will then be max position minus the minimum position. We can then get the sequence start. So the sequence start will simply be equal to minimum position, and then the sequence end. So sequence end will then be dependent on whether the gene size is above the 10,000 or not. So I'll say gene size is more than 10,000. If that is the case, then the sequence end will be minimum position plus and then 10,000. Otherwise, it'll just be max position, so we'll go ahead and limit it. So max position, and the range down here will then just be start, sequence start, and the end will then just be sequence end. And then the initial range will then just be range like this. And then we're missing a return statement down here in case this if statement isn't true. So what we're going to do is just take this right here in case no genomic data was found, and then just return that. That should also clear away the error you just saw up here. And now we can hop back into the gene viewer. So on load, we'll just run a use effect that'll go ahead and call this method. So I'll say use effect. And this use effect should be ran on initialization. So I'll say gene and then genome ID. So it'll only depend on these two. So it'll run when the component is first rendered and if these were to change. And then the thing that is returned, or the result that is returned from this function right here, we can just store in some use states as well, so we can show it. So first I'll say const and then gene detail and then set gene detail. This will then be equal to a use states, that is a gene details from scratch. So let me just see if we exported that. It does not seem like we did that, so I'll just scroll up. So maybe I just have to fix some bugs here, and then this will just be null as the default value, and this can then also be null. And now we are able to import it along with the use states. This just has to be a client component now because we're using states within the component. And then just like we did inside this page, I also want a loading state and as well as an error. So I'll just copy these two right here into the gene viewer like that. And then for the initial range, I would like to just keep use states for those two. So that is the start and end position we also return right here. So I'll say const and then start position, and I can then say set start position, and that'll just be equal to a use states. That can then be a string for now, so I'll just say string and make this an empty string right now. So this will be an input field where the user can enter a string, and then the string will be interpreted as a number. And this will then of course throw an error if it is not a number. So I can then say end position and set end position, just so we can set these from the initial range. Good, so in this use effect, we can go ahead and call the new method we created in Genome API. So first I'll say const initialize gene data is equal to an async function. And then down here I can say initialize gene data like that. And then of course we need to set the isLoading to true. We need to set an error to null. So set error to null. So we'll basically just reset all of these states. So I'll also just set the gene detail to null, as well as the start position to an empty string. And I'll do the same for the end position. This just means we have a clean slate to work with. And then if I say gene.getData, gene ID and then hover this. This might actually be null. So right now it is not optional, but it might actually be undefined. So I'll just go ahead and fix that bug right there. So if the gene ID is not returned from the API where we fetch it from, we'll then say if not gene.geneID. So if the gene ID is missing, we'll then say set error. And the error will be set to gene ID is missing, cannot fetch details, like that. And then we'll also set is loading to false. We can actually just do that inside the finally. Oh wait, that won't even be run. So what we'll do is just say set is loading to false because we started it up here. And then we'll just return and then don't run any of the code below this right here. And next up we can say try and we can say catch. And then we can say set error inside this catch and say failed to load gene information. Please try again. Like that. And then we can also create a finally that is run either way. And then we can say set is loading to false. And then inside this try, we'll then say const results is equal to await API or just fetch detail. So fetch gene details and we'll pass in the gene.geneID like this. And then what we get back is the gene details, gene bounds, and also initial range. So I'll just destructure this right away. So gene details, gene bounds, as well as the initial range. And then we can go ahead and say set gene detail. This will be gene details. Then we can say set gene bounds, which we just need to create a use state for as well. So I'll hop up here and then say const gene bounds and set gene bounds is equal to use state gene bounds or null. And then we'll just set this to null per default. And then down here we can now say set gene bounds and pass in the gene bounds. And then we can go ahead and say if we have the gene bounds right there, then what we'll do, or this is actually the initial range. If we have the initial range, then I'll say set start position to a string. We'll convert the initial range dot start, and then we can also set the end position where we say initial range dot end like that. And then we just need to fetch the gene sequence. And we might as well just go ahead and create that now. So hopping into Genome API up here, I see we need to just type here though. So hopping into the Genome API, we can create a new function. So I'll just go down to under this one and then say export async function, fetch gene sequence. So what this does is that you give it a gene, or at least the gene's positions, and then it'll fetch the nucleotides for that gene, or at least a range within the gene, and all the nucleotides in that range. So what I would like to accept is the chromosome where the gene is on, that it'll be a string, then also a start position on the chromosome, which is also a string, and end position. And we'll just need to make these numbers actually. And then also the genome ID, and that is also a string. What we'll return is a promise, because it is an async function. This promise will return a dictionary with a sequence that is a string, so A, T, Gs, and Cs, an actual range. It will have a start, which is a number, and an end, which is also a number, as well as an error. If that happens, that is just going to be a string. Then I'll make that a function. And first we can just define the chromosome. So we'll say const chromosome is equal to chromosome dot starts with. So if it does start with a chromosome, let me just fix this. If it does start with chromosome, then we'll just use the chromosome. But if it isn't prefixed with chromosome like that, we'll just go ahead and prefix it with that. So we'll say chromosome and then chromosome like this. This is just to ensure that we follow the standards that the API uses. Then we can say const API starts is equal to start -1. So for the UC Santa Cruz API we'll use, they use a 0-based start and then an exclusive end. So for the start it'll be start -1, and for the end we'll then have to say just end. because the end is exclusive, so we don't need to subtract one right here. And then for the actual fetch URL, we'll say const API URL is equal to formatted string, so https api.genome.ucsantacruz.edu get data, and then sequence, where we pass in the query parameters. Genome is equal to genome ID, and we just need to put in a semicolon right here, and the chromosome will be equal to the chromosome, and the starts will be equal to API starts, and the end will be equal to API end, like that. So we can just go ahead and fetch that URL. So we'll say const response is equal to awaits fetch API URL, and then we can say const data is equal to awaits response.json, like this. And then we can see the documentation if we just hop into this URL. It is the UC Santa Cruz API which we've worked with already, but we'll just use a sequence endpoint right now. So get data / sequence to return a sequence from a specific database. And then one thing I just wanted to show you as well is the errors. So I think it should be down here, return examples. If we just click this one and then say pretty print, we see that we get an error key right here. So we can use this to find out what the error actually is by, for example, printing this. So let me just hop back out of this and then into our application. We'll just create a variable called actual range, which is the actual range requested, which will be equal to start, comma, end. Then we can say if data.error, which we just saw. If that is there, then there obviously is an error. So we'll then say return sequence, which is nothing, and then the actual range as well as the error, and that'll just be data.error. So this right there. Then we can get the actual sequence from data.dna. So we'll say sequence is equal to data.dna. So just to make sure that this property is here, I'll then also say or not data.dna. Then down here I can say .to uppercase. So we get A to GCs instead of A to GCs like this. And then down here I can just say return sequence dot actual range, and then just leave out the error, which obviously isn't going to be there right now. Then I'll just take this entire thing and wrap it inside a try catch. So I'll say try and catch, and I'll catch the error. So this, and then in here I can just return an error message as well. So I'll just go ahead and say error like this. And then the actual range will then just be the start and the end like that. So I'll say actual range. And then I'll just call this error something like internal error in fetch gene sequence. Great. Then we can hop back into the gene viewer, and here we'll just create a function that can fetch the gene sequence for us, set the gene sequence up here, and then also keep track of loading, etc. So I'll just say const fetch gene sequence is equal to use callback, and then I'll say async starts, which is a number, end, which is also a number. Inside this, I'll pass in a dependency array, so it'll depend on the gene.chromosome and also genome ID. So this function will only be recreated if these ones change right here. Then I can say try and catch error. And then I'll quickly just create the use states, which will need to store what is returned from the Genome API. So up here, I'll just say const and then sequence and set sequence. I'll just call this gene sequence and then set gene sequence is equal to use state. And that is just going to be an empty string for starters. I'll then create a loading state as well. So I'll just copy this actually, and then say const is loading sequence and set is loading sequence. And then there's going to be the bounds of the sequence, which is the max and minimum, so the minimum and maximum that the sequence view can show, but the user will always also be able to pick these bounds themselves, so there'll be an actual range. So I'll say const and then actual range, set actual range is equal to use states, and this will just be null for now. And then I'll go ahead and say a start, which is number, and end, which is also a number, or null. So that is what we'll work with. So the first thing we'll do is say set is loading sequence. We'll set that to true, then we'll set error to null in case there was an error. We'll then clear the previous error. Then we can say const result is equal to fetch gene sequence, and we'll not reference this one, we'll reference the one from over here from the Genome API. So I'll just take this one, import it up here, and then call it as API fetch gene sequence. This way we can differentiate between the local one and the one we're importing. So this one will then be awaits API fetch gene sequence. Oh, sorry, that is details. It is right up here. So API fetch gene sequence. Then I pass in the gene.chromosome. We'll pass in start, end, and the genome ID. And we of course just need to await this as well. And then we can destructure the output to the sequence, actual range, and the error. So we'll say sequence, actual range, and we'll just say, we'll call this fetched range because we already have a state up here called actual range. And I'll then also say error. I can then go ahead and say set gene sequence, set that to the sequence. I can say set actual range to the fetched range. And this one I'll just call API error as well. Then I can say if there is an API error, then I'll set the error to API error. And if an error was then caught down here, we'll then set the error to fail to load sequence data. And I'll then say finally set is loading sequence, and I'll set that to false, so that always happens. So now the gene data can automatically be loaded whenever the gene details are loaded as well down here. So I'll say wait fetch gene sequence, where we pass in the fetched gene, or actually the gene details. So I'll say fetched detail just to avoid name conflicts, fetched gene bounds, as well as an initial range, which will just be fetched range, like this. So now I'll just call this fetched bounds, fetched details, and then also fetch range instead of the initial range. And then we can say fetch range.start, and we can say fetch range.end. Then if we go ahead and just try and print out this after it has been done, we can say console.log and then fetched. Let me just try the details first of all. So fetched details. And then I'll also like to just print out the fetched gene sequence. So I'll hop in here and just console.log, and I'll say sequence, and then refresh over here. And then we can try and load the BSA1 example, click one of these. So no details about the gene is actually being printed out, and I had to spend a couple of minutes figuring out why. So the reason is that in here in the Genome API, I made a little spelling error. So instead of gene ID with a small d, you have to make it a large D. So you save now, and then you can refresh over here and try the example again. And now it should print out some information over here. So you see all this information about the gene, as well as a single G from the sequence right here. So I just checked out that error as well, because the sequence should definitely be longer than just a single nucleotide. So what I found is that if I hop into API fetch gene sequence, then in here I can scroll down to, up to the range. So this initial range, which is used to fetch the gene sequence. The problem is that this is minimum in both places. This has to be minimum and this has to be maximum. So I'll just save and then we can try it again over here. So try BRCA1 example. And now we see the entire sequence being printed out. One thing is though that you see it being printed out twice. So it seems like it is doing it twice. And the reason for this is that there is something called React strict mode that hasn't been enabled. So what I usually do when I have problems with things rendering twice when they should only render once, and it actually causes problems for me, is that I hop into my Next.js and set React strict mode to false. If it is not false, then it might render twice for some components when you're using the development server. So hopping back over here and just refreshing everything, I can then just try it out again and see if it does it twice again. So now we see the sequence being printed out only once, so that fixed that issue. So I'll just hop back into the gene viewer. So now that we basically have all the data we need, or we do have all the data we need for showing the gene details in this card as we talked about earlier, I can just clean up the console.logs because we don't really need those anymore. So this one, that one, and that one. And then I'll create a new component. So under components, I'll create a new one called gene information.csx. In here I'll say export function gene information. This will take a gene detail, as well as and let me just make this a little bit smaller again, as well as a gene bounds. And I can just define the types of these properties. So this gene is going to be a gene from search. The gene detail will just be gene details from search or null. The gene bounds will be gene bounds or null. And I'll then just return, I'll just return an H1 for now that just says hello. So I can then take this gene information, hop back into the gene viewer, and then we can plop that in right here. So I'll say gene information, import the component, and then I can pass down the gene, I can pass down the gene detail, and I can pass down the gene bounds, like this. And now we are able to see the component being rendered out right here. So I would like to render out a card from Shadcn. So I'll say card, import that. Then I can say class gap 0, border none. I can say bg whites, py0, shadow small. I can then just put in a card header. The card header should have a card title. The card title should be gene information. The card header should have the styling pt4 and padding bottom 2, like this. And the card title should then have text small, font normal, and it should have text 3C, 4D, 3D at 70%. And this just needs to be an F instead of a D. And for the card content, we can start out with a card content component. And inside this, we'll then say class name, padding bottom 4. We can then put in a div with the class name grid, gap 4. At medium or above, we'll say grid calls 2. Then we can put in another div inside here where we put in the class names space y2. We can then put in another div with the class name flex. And lastly, we can put in a span that says symbol. For the span, we'll style it a little bit. We'll say width of 28, minimum width of 28 as well. To force it, we'll then say text extra small. And we can say text with this color up here. And I'll just tone that down as well. I'll just copy this span right here, the flex. And I just forgot one thing quickly up here. I'll just say span, and then I'll say gene.symbol. And this I'll just put on some classes for as well. I'll say text extra small, and then font medium, like that. And then you can see where we're going with this. So down here, we'll just do the same for gene.name. And then this will then just be name instead. I can then copy this entire thing again. And then I can say if gene.description and gene.description is not the same as gene.name. If that is the case, then I would like to render out the following right here. And then I'll just say gene.description and description right here. Then I can just show the chromosome property as well. So I can go ahead and say chromosome, gene.chromosome. And then we also just want to show the gene position within the chromosomes that is going to be determined by the gene bounds. So I can just go ahead and say if gene bounds, like that. Then I'll say position. I'll then just remove the font medium, and inside the span I'll go ahead and say math.minimum gene bounds dot minimum gene bounds dot maximum dot toString. And then outside this I'll just put in a dash like that, a forced space as well. And then I can put in the same just for the upper bound. So in this line instead I'll say max as well as the difference so the user can see that. So I'll say base pairs, and then I can go ahead and say math.abs, and then we'll put in the gene bounds dot max and subtract the gene bounds dot minimum, and then just add 1. And that is because of the zero-based positioning. So I just want to put this inside a parentheses, so I can go ahead and do this and just add in one more of these forced spaces right there. So now we see the actual amount of base pairs as well. And I can just say to local string for all of these 3, and when I save, it should then be formatted with your local string. So in this case, it is a comma that is the thousand separator. So this is way nicer to look at than just the normal string. And then lastly, we also saved whether it was a reverse strand or not. So going down here, I can say if gene detail dot genomic info, and then we can say dot 0 dot strand is equal to minus, then we'll say reverse strand like that. And then down here we can just do the next part of the box. So I'll say diff and then class name space y2. I can then say gene.gene ID. So if that is there, then we'll render out a link to the actual gene so you can click it and then see where we fed the gene from, as well as more information about that specific gene. So I'll put in a diff here. I'll just close the diff again. Inside this diff, I'll say class name flex. I'll put in a span, so this will have gene ID inside of it. So we'll just style that as well. So I'll just grab this from up here, put it into the span, so class name, and actually I'll just make this font medium as well, just so it looks the same as the other ones up here. So now that we have the gene ID, we can also go ahead and render out the link. So I'll put in this other span too. So I'll say span class text extra small. Then I can say anchor. Inside the anchor, we'll put in the gene.gene ID, and we'll also put in an external link from Seed React. We'll give it the class names margin left 1 to space it a little bit from the gene ID text, inline block, and then h2 and width 2. So it'll look like this. So this is kind of what you expect whenever you click a link that opens up a new page. And then for the anchor itself, we'll then say href, and the href will be a formatted string. So we'll go to https and then www.ncbi.nlm.nih.gov/gene/ and then we'll put in gene.gene ID. So whenever we hover it up here and click it, right now it can't be reached because it is GIV instead of GOV. So I'll just try again. So when I click it now, it should take us to the other page. Perfect. So now we can see the BRCA1 gene, which is the one we're currently on. Great. So we just need to open this in another page because it isn't very nice of us to redirect the user's page to another page. We should instead open it in a new tab. So to do this with an anchor, we can go ahead and say target and then underscore blank like this. And I'll also add some classes to that as well. So I'll say class name and then text blue 600, hover underline, and then when I hover it over here, it lets us know that we're able to actually go ahead and click it. So this is what I mean by opening it up in a new tab. That is what is going on right here. Good. Then what I'll just do is just test this with a little bit of big of a size, see if it looks better. Maybe we can say inside this anchor, we can do flex and then items center. It looks a little bit better now, I think. Yeah, let's go with that. Cool. So under this, we'll then say gene detail dot organism. And if that is the case, we'll render out a div that contains information about the organism. The first thing we'll render out is a span that says organism, like this. I'll then just save that and format this a little bit nicer. So class name flex, organism with 28, text extra small, and then I'll grab the usual text color from up here. Then I'll put in one more span, class text extra small, and then say gene detail dot organism dot scientific name. I'll then also just show the common name. So if I just copy this entire thing, I can then say, or I'll just do a conditional as well. Then I'll say if this common name is there, then I'll render it out. So I'll say formatted string, and then go ahead and do that. So we then see the scientific name, Homo sapiens, and the common name, which is just human. And lastly, we can put it in the details. So, the summary. So I'll say gene detail dot summary and div. Inside this div, I'll just say class name empty form. I'll put in an h3 summary. I'll put in class name mb2 text extra small font medium, and then text in the usual text color like this. And then under this, I can just put in a paragraph and say gene detail dot summary and say class name text extra small leading relaxed, and then just take the text color again and tone it down to about 80%. So this will then be the entire info box for this specific gene, and this is how it is going to look both on larger screens as well as on smaller screens. I think I'll just remove this font medium up here, since it is there sometimes but not there other times. So I'll remove it right there, and also these two places. And then I think I need to remove it right here as well. So now it all looks the same, so it's a little bit better in my opinion. I will then create a gene sequence card as well. This will also be a component. So remember how we printed out this large sequence of up to 10,000 nucleotides for this specific gene? Well, I would like to visualize these in a card as well. And these should also be formatted in a nice ways with colors that mark each single nucleotide. So to get started with this, I'll just hop into the UI, components, create a new file in here. This should be called gene sequence.tsx. I'll then say export function gene sequence, and then I'll say I'll just leave out the properties for now actually, and then in here I'll just return a h1 saying hello. I'll then just make this a client component, So use client, save this, hop back into the gene viewer, and then I'll place it right over the gene information component, like that, and save that. And now we should see hello, and then we can stack these cards above on top of each other, because we have them inside this space y6 div, and this div will then just be a column with a little bit of space between the cards. So first things first, I'll hop into the gene sequence, and then we can start defining the properties that will be needed to show the gene sequence. So the first property is going to be gene bounds. So gene bounds, and the gene bounds will just be of type gene bounds like this, or null. So there's going to be a range slider. So let me just find this range slider. So range slider is something like this. So this is a range slider for the nucleotide, or that is what we're going to build. So there will be a minimum bound right here. That is the minimum position of the nucleotides in a gene. There will also be a maximum bound for that. And then the user can then specify a range right here. So a minimum shown range and a maximum shown range. And all the nucleotides within this range is what is going to be loaded and then shown on the screen. And there will then also be a maximum range, so that is a maximum distance these two knobs can be from each other. And the reason is that if the minimum and maximum bounds are very far apart, so maybe hundreds or thousands of nucleotides, that is a lot of data to load. So we'll limit the range between these nodes, or the distance between these knobs, to be only 10,000 nucleotides. So that is also the reason why we're going to need stuff such as gene bounds and other props, which you will see in just a second. Cool, so hopping back, I just closed it right now. So hopping back into the gene sequence, the next property is going to be gene detail. So the gene details are going to give this component detailed gene info. So right here it's mainly used to show the DNA strand, so whether it is positive or negative. So we'll then just call this gene details from search, or null. Then we'll also supply a start position. The start position is going to be a string. We'll give it an end position. The end position is as well going to be a string. So that is going to be the two knobs on the range slider. This is going to be the minimum knob, and this is going to be the maximum knob. And the gene bounds is then going to be the minimum and maximum for the entire gene. And these should then of course also be able to be changed. So the start and end position, because those are two knobs. And whenever they are changed, we'll then fetch or refetch the sequence that is loaded in the parent component, so gene parent component, so the gene viewer. So we'll need to have some way to let the parent component know that these has been changed. So we'll say on start position change. So this will be a function with the value that it has been changed to, and that'll just return void. And we'll also have an on end position change, which will just do the exact same thing as well. We of course also need to pass down the sequence data. So the sequence data is just going to be a string of A to G's and C's. And then for the sequence range, that'll be start number, end number, or null. So I'll just say number here as well. And the sequence range right here then specifies the genomic coordinates corresponding to the displayed sequence data. So the coordinate of the start, and then also the end position in the chromosome. We'll also then pass down the is loading boolean flag from the parent that indicates if the sequence is currently fetching. And this is of course just going to be a boolean. We'll pass down any fetch errors, such as a string if that is that, otherwise it'll just be null if there's no errors. We also want to pass down a function that can just trigger refetching of the sequence data, such as if the start and end positions are changed without these being called. So we can go ahead and say on sequence load request. And this will just be a function returning void. Then we can say on sequence click as well. And this is for when a single nucleotide is clicked. And when a single nucleotide is clicked inside this big list of the nucleotides, then this should be passed back to the parent component. And then in the card which we'll make in the future containing the variant analysis, that specific nucleotide and its position will then be highlighted for during the variant analysis. So to actually select a nucleotide, we'll need the position of that nucleotide in the chromosome. And we'll also say nucleotide, which is the base. And this will then just return void. And lastly, we'll pass down a max view range. And this max view range will define the maximum allowed sequence length for both the validation and slider constraints. So this will then just be the number right here. So now I just gotta go ahead and destructure these. So I'll just copy paste them in one by one. Start position, end position as well. And then on position change, sequence data right here, sequence range is loading, error as well, on sequence load request, on sequence click, and max view range. So to be able to follow along with what we're actually building in real time, I would like to just pass down the props such that we don't get an error over here. So right now, if I refresh and then go to this, we don't really see anything, and also we get an error over here, but I would like to pass down the props such that we can work with the live data. So the gene bounds is going to be, I can just say add missing attributes first of all, and then the gene bounds are going to be gene bounds like that. The gene detail will be gene detail. The start position is going to be start position. End position will be end position. On start position change, we'll say just set start position. For the end position change, we'll say set end position. For the sequence data, we'll say gene sequence. For the sequence range, we'll say actual range. Is loading will be is loading sequence. Error will then be error. The max view range is going to be 10,000, so that is the maximum amount of nucleotides we will be able to load. And then these two right here we'll just leave unimplemented for now, and then we'll do a little bit later whenever we come to that point. So first, hopping into gene sequence, we can return a card. So I'll go ahead and say card. We'll say class name, and then let me just implement the card like that. Oh, sorry, import the card. We'll then put on the class name gap0, border none, bg whites, py0, and shadow small. We'll then also put on a card header like this, and inside the card header we'll put in the card title as well. So we'll say card title. We'll then say gene sequence, and then just import the card header as well. So now we should be able to hop into this menu and see gene sequence. Then for card header, we'll just put in pt4, pb2, and for the card title, we'll put on the class names text small, font, and we just need to spell this correctly, font normal, text, and then 3c, 4f, 3d, and then -70. Oh, sorry, /70. Good. And then for the actual card content in itself, card content, we can just go ahead and import that. Then we'll say class name, padding bottom 4. And then we can say if gene bounds is not null, then what I would like to show is a diff. In here we can say class name, margin bottom 4, flex column. And we can then show some range information. So we'll say diff. Inside here, we'll just put in a span. So I'll say span, and I'll put in a paragraph into the span that says from. I'll put in another paragraph that says math.minimum. So math.minimum, gene bounds.minimum, gene bounds.maximum, to local string like this. And we'll just format this just a little bit. So I'll say class name, margin bottom 2, flex column, items center, justify, between, text extra small, small flex row. So on larger devices, it'll be a row. And for the styling of the span right here, we'll say flex, items center, gap 1, text, and then we'll put in the text color from up here as well. So this one, I'll then put in this paragraph and say class name, hidden on larger devices. And you'll be able to see the full example of why in just a bit. So let me just take this and copy the span right here. So I'll copy the span down below and then say 2. This is then going to be .max instead. And now we have a from and we have a to. And then I'll just show the selected in between that. So I'll paste in the span once again. Oh, this needs to be text, both there, as well as right there. And for this one in the middle, I'll only keep the text parts. And then in the div or inside the span, I'll just go ahead and say selected. And then pass integer, start position, or just 0.to local string. And then outside this, I can say dash, put in a forced space. Under this, I'll say pass int. So I'll just do the exact same thing again. And then instead of start position, it's going to be end position. And then I'll say some number. I'll just do 1, 2, 3 right now. And then say base pairs right after this. So I'll say BP. And I would like to just enclose this in parentheses like that. So it's going to look like this right here. And I'll just force in a spacing right there. So on smaller devices, it'll look like this right here. And on larger devices, it is going to look like this. So you have the lower bound, the upper bound, the bound that is selected in the range slider, as well as how big the size of this selected range is. And we of course just need to calculate the actual size of the range instead of just hard coding 1, 2, 3. So I'll say const current range size is equal to a use memo. This means that the calculation done inside, or the computation done inside this statement right here, is only going to be done every time the dependencies or the variables in this array right here changes. So we'll put in the start position as well as the end position. So whenever these variables change, it should recalculate the range size, which makes sense. So we'll say const start equals pass int start position. We'll do the same just for the end position. Call this end. Then I'll say return. And this depends on is not a number. Start. So if start is not a number, or end is not a number, or end is less than start, then what we'll do is say 0, otherwise it'll be end minus start plus 1. And this plus 1 again is because of the 0-based positioning. So what this does is that we are using this pass in right here. So if start is not a number, or end is not a number, or end is smaller than start, which doesn't really make sense, then we'll just set it to 0. Otherwise, we'll actually calculate the range size between start and end, and then return that right here. So now we should be able to hop down and just use this. So instead of 1, 2, 3, we can go ahead and call it and say dot to local string and call that function. And now we see 10,001 right here. And it seems like there is a little problem with this plus 1. So right now, the last number is the same, but with a 10,000 base pair difference. So this should of course also be 10,000. So I'll just go ahead and remove the plus 1. And then we can move on to the slider component that is actually the range slider. So under this div here, I'll put in a slider component. I'll just make a comment around this just so we know what is what. So this will be a div with the class name space y4. We'll put in another div inside of this. This div will have class name relative. Inside this div, we'll put in another one with the class name relative h6 with full and cursor pointer. And inside this, we'll create the actual slider. So first we'll start with the track background. So I'll just make that a comment. Then I'll say div, and I can put in some class name, absolute, top 1/2, height of 2, width of full, minus translate, y 1/2, rounded full, and background should be the color we have used a lot of times, so E9, and then E A. So that is going to be the track background for the range slider. Then we can put in the selected range. So I'll just say selected range, make that a comment as well. So this will be a div2. Inside this div, I'll put in some classes. This will be absolute as well. top 1/2 h2 minus translate minus y minus 1/2 cursor grab. So we can just press save right now. And then if I just put in with full right here and then go up here, we should be able to see cursor grab. So that is what you see right here. So that is cursor grab. Good. I'll just remove the with full again and leave cursor grab. And then I can say rounded, full, background, free C, for F, and then 3D, like that. I'll then just say when active, then we'll say cursor grabbing. So that is another state. There is cursor grab. So when the cursor is about to grab, then there is cursor grabbing when we are holding down the cursor. So when we're clicking basically, then it'll look like we're grabbing something. Then we'll put in some manual styles as well that needs to be calculated inside the style property right here. So we'll put in a style with a dictionary inside of that. That is supposed to contain the CSS style properties. The left will then be a formatted string. This will be slider values, which we'll go ahead and create in just a second. So this will contain the slider values. So we'll just say .start for this one. And then this will be a number, which we'll attach a percentage to. We'll do the exact same thing just for the width. So we'll say width. This will be slider values dot end minus slider values dot start, and it will be evaluated to a number, which we can then append a percentage sign to. So I'll hop up here and then we can go ahead and create these slider values, which is going to be some kind of data structure which can contain these values for the slider. So I'll just use a use state for that. So I'll say const slider values set slider values is equal to use states. We'll have the start, which is just 0, and the end, which is just going to be 100. I'll import the use state and then scroll back down here. I'll just load the example once again. And then if I scroll back up, for example, and say 50, we can see that the width just changed right there. So I'll just hop back down and then just create the start handle. So that'll be the start handle. So the knob that is supposed to manage the start number. This will be a div. We'll put in a class name of absolutes. Then top 1/2. We'll put in flex, height of 6, width of 6 as well. Minus translate x 1/2. We'll also do minus translate y 1/2. We'll do cursor grab, items center, justify center as well, rounded full, border 2, border color of hashtag 3C, 4F, 3D, PG whites, shadow, and then active cursor grabbing. So I'll just go ahead and save that. And now we have some kind of knob over here. And then when I click down, we can see the cursor grabbing right here as well. Then we'll just need to calculate its position based on the start up here. And then for the same, just the end knob, we'll just calculate the position based on the end. So I'll take this and just add a style property to that as well and press save. And then we'll just calculate the left value just like it is done right here. And then inside the div, I would like to just put in another div. Here I'll say class name h3 with 1, rounded, full, and then a bg of green. So that is going to be hashtag 3c 4f and then 3d again. I'll press save. So that is what we're seeing right here. That is going to be the start knob. I'll then just take this start handle and copy it down here and call this one end handle instead. And the left value of this one, of course, just needs to be calculated based on end instead of start. So when hopping up here, we can set this to 10 maybe, and this one we can set to 60. So that means it'll look kind of like this. And then we just need to implement some logic for being able to drag these around, because right now we can't really do that. So to keep track of the dragging itself, we'll need to keep track of dragging on the x-axis. You won't be able to drag on the y-axis, so we only need to keep track of the x-axis dragging. So we'll need a few use state variables. First, we'll need const is dragging start and set is dragging start. This is also just going to be a use state that is a boolean. So this will be false per default. So you can do three things right here. You can grab onto the start handle and drag that, which will change only the start number right here. You can grab onto the end handle, do the same only for the end number. And if you have a a range that is maybe 60 to 70, then I want you to be able to also grab onto the green part right here in the middle and then drag both handles by grabbing onto the green part instead of the handles themselves. If you do this and drag, it should update both these numbers with the difference in dragging at the same time. So to do these things, we'll need to keep track of whether you're dragging the start handle, whether you're dragging the end handle, as we're doing right now, and then whether you're dragging the entire range. So range there and range right there. We'll then also create a reference to the slider. So I'll say const slider ref, and then I'll say is equal to use ref. I'll just import that and say that is an HTML div element, and that is null per default. We'll also need to implement a drag start x. So this will keep track of the value that we have currently dragged on the x-axis. And this will implement as a use ref instead of a use states to avoid unnecessary re-renders and just make performance better generally. So this will persist something across renders without triggering a re-render when the value is updated. Inside this, we'll put in an x that is going to be a number, a start position that is also going to be a number, as well as an end position, which is also going to be a number. And this can also be null. So I'll say or null. And we'll just set it to null per default like this. So I see we have an error right here. So I'll just go ahead and remove that. And then I would like to implement a method. So we'll say const handle mouse down. This will be a use callback that will receive a react.mouse event. So react.mouse events. And I'll say comma handle, which will also receive, that can be either the start handle or the end handle. Inside this, we can then say e.prevent defaults, because it is an event. Then we can say if handle is equal to starts, then we'll set is dragging start to true, and then the same if it is the end. So I'll go ahead and say end right there, and then put in an else right here. And I'll then just also add in a dependency array because it is a use callback. So we'll only want to create this method once. So that is what we'll do right here. So I can copy this function declaration, hop down to the start and the end handle, which we just created down here. And inside these handles, I can say on mouse down. So on mouse down, we'll then copy this function. We'll call it like this though. So pass in the event as well as the start handle. And then I'll do the same just for the end handle down here, and then say end instead of start. This should then let us know in the state right here. So the set is dragging start and is dragging end, that we are actually dragging. And then when we're dragging, we can also keep track of how much we have dragged with this variable right there. Now we just need to create a method for the handle mouse down just for the entire range, instead of just for the start or the end dragging. So this one I'll say const handle range mouse down is equal to use callback. I'll pass in the React event again. So react.mouse events. And then I'll say e.prevent defaults. Put in a dependency array. This will depend on the start position as well as the end position. So this ref we created up here, the slider ref, we'll just need to attach to the actual slider. So I'll hop down so we can find the slider. And we have cursor pointer right here, so that is the slider. Then I'll say ref is equal to slider ref. This means we can get information about this HTML element through this variable right here. So hopping back up, what we can do is say if and then not slider ref dot current. Then we'll return. Then we can get the start position and the end position. So we'll say const start number is equal to parse int start position. We can then also say end number, and this will then just be end position. I can then say if is not a number, start number, or is not a number, end number, because the user can fill these in themselves. And then I can say, then we'll just return, because then we won't really need to drag it. Otherwise, we'll set is dragging range to true. And then I can get the information about the current slider from this slider ref. So I'll go ahead and say const slider rect is equal to slider ref dot current dot get bounding box client. Get bounding client rectangle. Then I can say const relative x is equal to e.clientx minus slider rectangle dot left. So what this does right here is that it calculates the horizontal position, so that is this one here, of the mouse click relative to the left edge of the slider element. So this right here is the mouse position relative to the viewport, and this right here is going to be the slider's left edge position relative to the viewport. And the reason we are calculating this is that we need to record the starting point of the drag operation, so where the user clicked within the slider, and then what the genomic start and end positions were at that exact moment. And this is important because we later need to calculate how much the range should be shifted based on the mouse movement. So let's say, let me just open this up. So if I click right here, for example, and then drag to the left, then this will be the relative x that will be dragged from. So if I click here, then we need to let the system know that this is where we drag from and not this is where we drag from, for example. And this information about where we're dragging from, we can store on this drag start x, which we created earlier. So we'll say .current is equal to x will be relative x. We can then say start position, which will be start number. And we can say end position, which is going to be the end number. So it'll just record the value of this one, and then where we start dragging from. And then I can take this method right here, function, and then copy it down to the selected range, which we should be able to drag on mouse down, like that. And then when I actually do go ahead and click it, we don't see any errors, so that is perfectly good. We'll just listen for when the mouse has been unclicked also, so when on mouse up is fired. So going up here, I can just say const handle mouse up, and this will just be a function. In this function, we'll say if is dragging start, or is dragging end, or is dragging range, and start position, and end position as well. And that should then trigger loading the new sequence that was just dragged to. So right here, I'll just go ahead and fix these braces. So this needs to be in the brace right here. And then if that is the case, what I'll then do is say on sequence load request, which we will pass down from the parent right here. So this will just trigger loading the new sequence that we just dragged to whenever we drag one of these sliders. So for example, when dragging this slider over here, for example, and then dropping it again, then I want it to automatically load the new sequence of nucleotides down below without the user having to click any buttons. So I'll go ahead and do that. And then right below, I'll say set is dragging start. And I'll set that to false. I'll do the same for the end because now we aren't dragging anymore. I'll do the same for the range. And I'll also set drag start x.current to null. Then we'll just create another method. This method is going to be handle mouse move. So whenever you do move the mouse, we need to calculate where these should be dragged to, and then also change these positions. This will be a new function that takes an event. That will be a mouse event, like this. And I'll take both of these functions and just copy them and say use effect. And then we'll add them to a use effect where we can then fire these functions when their given events require them to be fired. So I'll add these two functions, and then I'll say window dot add event listener, and I'll add an event listener for mouse move. And then when mouse move is happening, we'll then say handle mouse move, call this function, We'll do the same just below when we say mouse up, like that, and then we can call mouse up for that as well. And then we'll just need to unsubscribe to these events as well. So we'll go ahead and do this. Do the exact same thing, but just say remove event listener instead for both of these. So when the mouse moves, we should calculate the new positions as well. So first we'll check whether we are actually dragging or not, because if we are moving the mouse but we aren't dragging, well, we obviously don't have to calculate anything. We should just skip this handle mouse move method. So I'll say if not is dragging start, and is not is dragging end, and we are not dragging the range, so is dragging range. Then I'll just return. Then I can also say if sliderref.current, because as you can see when we hover it, it can be null, so we want to make sure that it is not null, or not gene bounds. Well, then we'll just go ahead and return. So hovering gene bounds, this can also be null, so we want to avoid that as well. So remember that when we click down on the range right here, we recorded the relative position within this range that we clicked, so we can be able to calculate the distance between that, and then whenever we have dragged it, so that position after having dragged it. So we'll need to take the difference between those. So I'll actually just copy these two lines right here, because we'll need to do the same. So I'll just paste those two in right here, along with the slider width. This will then be slider rect.width. Now we can calculate the mouse's horizontal position as a percentage along the slider's width. So we'll go ahead and say let new percent be equal to relative x divided by slider width. And then we'll say times 100. We then also want to ensure that the calculated percentage right here stays within the valid range of 0 to 100, preventing it from going outside the slider's bounds. So we can then say new percent is equal to math.max. And then we'll say 0, math.minimum, new percent, and then 100 right here. This will always keep it inside between 0 and 100. Then I'm going to say const min bound is equal to math.minimum, genebounds.minimum, genebounds.maximum. And I'll do the same just for max bound, and this will be max, but we'll put in the same parameters. So basically, if min is larger than max for some reason, then max will become min, and then the opposite for max. So we just get the smallest one and the largest one. And then we can calculate the gene size. So const gene size is equal to max bound minus min bound, and just calculate that. And now we can then calculate the new position. So this will translate the mouse's position percentage into the corresponding genomic coordinate within the genome's bounds. So we can go ahead and say const new position, and then is equal to math.round, and we'll pass in minimum bound plus gene size times new percent, and divide that by 100 like this. I will then just get the current start number. So I'll say current start number, which is going to be parseInt start position. I'll then also get the current end number, like that, which is going to be start and then end position. Then what I can do is handle these different cases. The first case is going to be if they're dragging the start handle right here. The second case is going to be dragging the end handle, and the third case is going to be dragging the entire handle, the entire range. So that means both handles at the same time. So if you are dragging the start handle, then we'll first check whether this current start number right here, current end number, is a number or not. So we'll say if is not a number, current end number. So this current end number will act as a fixed point to calculate how wide the range is. And this way, if the range is stretched to more than 10,000 base pairs, it can be constrained to 10,000 base pairs, so it doesn't move past that level. So first I'll say if current end number minus new position plus 1 is larger than the max view range, so that is the 10,000 number. Then what I'll do is then say on start position change, so we'll notify the parent, and we'll pass in the new position. So this will be a new string where we put in current int number minus max view range plus 1. If we do not need to constrain it, we'll say else if new position is larger than the current start number. Then we can just go ahead and say on start position change and pass in a string with the new position. Then if we're dragging the end, we can just take this entire statement and say else if instead of just if. Then this will be is dragging end instead of is dragging start, because we're dragging this handle right here. This will be the current start number. And for this case instead, if the new position minus the current start number plus 1 is larger than the max view range, then we can say on end position change, and then we can say current start number plus max view range minus 1. So this will have to be current start number. And we'll need to do the same right here as well, and just save. Then if we're dragging the entire range, that is another case. So we'll say else if is dragging range. And if that is the case, we'll first say if not drag start x dot current. If that is null, then we'll just return, because then we won't really have the information we need to be able to calculate how much we've dragged the range. So I'll say return. So first I would like to define how many pixels on this slider represent one genomic base. So we can do this by saying const pixels per base is equal to slider width divided by the gene size. So this will be the amount of nucleotides in the sequence. Or sorry, not in the sequence, but in the entire gene actually. We can then calculate the horizontal distance in pixels the mouse has moved since the drag started. So this will be const drag delta pixels. This will be equal to relative x minus drag start x dot current dot x, like this. And then we can find out how many delta or how many bases we've actually dragged now. So we'll say const drag delta bases. So we'll need to convert to how many pixels we've dragged to how many bases we've dragged. So we'll say equals math.round, because you might not hit a specific base whenever you do these divisions. So we can say math.round, drag delta pixels divided by pixels per base, and save that. Then we can shift the original start position by the calculated number of bases. So what we'll do is say let new start be equal to drag start x dot current dot start position plus drag delta bases. And we'll do the same just for the end. So how much we should change the position. So this will be end position, and the range size, so const range size, will then be equal to drag start x plus current plus end position minus the start position, like that. And then we also want to make sure we don't exceed the minimum or maximum bounds of the gene itself. So we'll say if new start is less than the minimum bound of the gene itself, then we can say new start is equal to the minimum bound. So we'll constrain it to the minimum bound, and new end will then just be minimum bound plus the range size. And for the max bound, we can say if new end is larger than max bound, then new end is equal to max bound, and new start is equal to max bound minus the range size. Lastly, I can say on start position change, then we'll change the position to new start, and we'll also set to new end the end position. So on end position change. And then we just need to add all of these variables or all these state variables which we depend on to the dependency array, such that the use effect re-renders the component whenever these changes, so we can actually see the difference. So we need to add dragging start. We'll need to add is dragging end as well. Also is dragging range, gene bounds, start position, end position. So down here. as well as the on start position change, on end position change, max view range if that changes, on sequence load request. So on sequence load request, like that. And then I'll just save that. Now I just ran into a few errors because I overlooked a couple of things. So hopping back up, we can see that this isDragon range is just an empty else if. So this obviously needs to include all of this. So I'll just go ahead and include all of this code inside the isDragon range. And secondly, there is another issue up here that I just need to fix. So this down here needs to be changed a little bit. So this has to be new position is less than current end number, like that. And then whenever I do save it, we shouldn't run into these problems later that I just ran into. And then right now, these start position changes, end position changes, then updating these states in the parent, but we aren't really updating the slider that actually controls where these knobs and also the range should be. So what I'll do is create a use effect up here, listening to the start and end positions, so these two, and then updating the slider values to match those. So hopping down here, I'll say use effect. And then the first thing I would like to do is just check for whether gene bounds are there or not. So I'll put in an empty dependency array and then say if not gene bounds. If that is the case, we'll just return. Then we can say const min bound is equal to math.minimum of gene bounds.minimum and gene bounds.maximum. We'll then do the same just with max and call this max bound. We can then calculate the total size again. So we'll say const total size is equal to max bound minus minimum bound. Then let's pass the start and end position to integers. So we'll say const start number is equal to pass int start position, and we can get the end position as well. So end position right here and end number. I can then say if is not a number, start number, or is not a number, the end number, or the total size is less or equal to 0. Then we'll set these slider values to just start 0 and end 100. And then we'll return and do nothing more. Otherwise, we'll say const start percent will be equal to the start number minus the minimum bound. And we'll divide this by the total size and multiply by 100. So this will make it 2%. We'll do the same just for the end percent. So I'll say end percent, and this will then be the end number minus the minimum bound, and then the same over here. And then what we'll do is just say set slider values to the start, which will be math.max 0, and then math.minimum start percent and 100. This will constrain it between 0 and 100 such that the percentage never goes over. Then we'll just do the same just for the end. So now we have start, and we'll just set end here as well, and call this end number. And this use effect loop should then run every time the start position is changed, the end position is changed, or the gene bounds are changed. And this will happen whenever these on changes, on position, end position change, for example, is called, and then updates these in the parent. Cool. I can see we just need to do this though, because I made a little spelling error. So I'll just do that and save. And now if we hop over here, we should be able to drag around the slider. So that looks great. If I just refresh, I saw we got a little error. If I just refresh, we can also drag around the end slider, and then we get this error. Let me just check out what that is. Function not implemented. Perfect. So that is because of this over here. So if I just make this a function doing nothing, instead of throwing an error, hopefully we won't really get an error that lets us use all of the functionality. So I can load up an example and then drag this around like this, drag this handle, I can drag this handle, and I can drag both at the same time. And then we'll go ahead and implement these after just implementing the last parts of the gene sequence.tsx. First, I would like to implement the manual input position controls. So if I scroll down to the gene bounds, hop all the way down, then about right here, I can put in the position controls. This will be a div first of all, and this will have quite a lot of classes. So I'll say flex column, space y of 4. On larger screens, we'll say flex row, and items center, and also justify between. We'll also say space y0, and then inside this div, I'll put in one more div. I'll say class name, flex, items, center, and gap2. In here, we'll put in a span that says starts. We should also have a class name with text extra small, and a text color that is FreeC for F, 3D at 70%, like this. And now we can just add an input field. So let's see if we have that already. We do, from Shadcn. And this should have a value. The value comes from the start position. Whenever it is changed, we can fire the onChange event, or the onChange event is fired. So we'll say onChange right here, and we'll receive an event and say on start position change, and we'll pass in the e.target.value. Then we can pass in the type of input field. So the type will be text, like this. Then we can also put in the input mode. This will be numeric, and we could put in a pattern to match. So this will be 0 to 9. So the characters 0 to 9. So only these numbers right here, and then unlimited of those. And this of course needs to be start position, and then save. I can then take this entire div right here and copy it down below, and just call this end instead of start. So I'll say end, and just put in a colon as well. And then I'll say on end position change. The value will be end position as well. So with these two input fields done, we can go ahead and try and change the range, and we can see the input field changes as well because they are connected by the use states. And I can also change this input field to maybe 0, for example, or 54. So it seems to be working out just great. I'll then also put in a button to manually trigger the loading the new sequence after changing some of the numbers in one of these input fields. So I'll do that right here. I'll put in a new button. This button should have the text depending on whether it is loading or not. So first is loading. If it is loading, we'll say loading ... if the input sequence being fetched is loading. Otherwise, we'll say load sequence. Then we can just import the button and style it a little bit as well. Let me just open it up so we can see it. So the button should be styled the following way. We should have size small. We should then have disabled when it is loading. When I click it, so on click, I'll say trigger load sequence. So this will trigger a load sequence from the parent. And then I'll also put on some classes. So I'll say class name, height of 7, width of full, cursor pointer. And then I can say background, hashtag FreeC for ref, 3D, text extra small, text whites, hover, background, free, and I'll just need to fix this. The background is supposed to be FreeC for F, 3D, at 90. And then I'll say on larger devices, we'll say with auto. So I'll just comment out this right here, and then we can check out the actual button and see if it looks like we expect it to. So that is what we see right here. So it seems like these input fields are a little bit larger than the button. So I'll just go ahead and style those so they become a little bit smaller. So for the input field, I'll say class name, and then I would like to say height of 7, width full. I'll put in a border as well. So I'll just grab the border right here, just this one, or at least the color for the border. And then I'll say add 10, and then I'll say text extra small. And then I'll say on larger devices, width of 28. So it'll look like this. So now it's the same height as the load sequence button right here. I can then take this and copy it down into the other input field too. And now it looks alike. So if on smaller devices, it'll look like this. And then when going to larger devices, it'll just scale up so they'll become side by side instead. So I think it looks pretty good, so now we can move on. First of all, whenever this load sequence is clicked right here, I'll then call the function passed down from the parent to actually load the sequence. So that is the onSequence load request. So I'll just call that directly down here. So right there. Then right now we won't be able to click the load sequence because hopping back into GeneViewer, we see that this is not implemented. So I'll just go ahead and do that first of all. So I'll hop in here and create a new function, const handle load sequence. This is going to be wrapped in a use callback. To avoid unnecessary re-renderings, I'll put in an empty dependency array for now, which we can populate after implementing the method. So first I'll say const start is equal to pass integer, and I'd like to pass the start position. I'll do the same just for the end position. So I'll say start and end. Then I can say let validation error string or null be equal to null. So this will just store any validation errors that might happen. Then I can say if is not a number start or is not a number end. Then we'll say validation error is equal to please enter valid start and end positions. We can then also say else if start is more or equal to end. Then the validation error is going to be something like start position must be less than end position. We'll then also just check whether these positions are outside the gene bounds. So I'll say else if gene bounds. So if gene bounds are actually there, then we can say const minimum bound, which will be equal to math.minimum. And this will then be gene bounds dot minimum and gene bounds dot maximum. We can also say max bound by just putting in a max instead, and then say max bound. I'll then just check whether we are inside or outside the bounds. So I can say if start is less than minimum bound, then the validation error is going to be something like start position start dot to local string is below the minimum value. Then I'll put in the minimum bound dot to local string as well. And then I'll just take this line and just paste it down, or sorry, take this entire if statement, paste it down, say else if, and then I'll say if the end is more than the maximum bound, which is the opposite, then we'll say end position, and then end is below the, I'll just say exceeds the maximum value of maximum bound dot to local string. And lastly, we'll check if their distance between each other is more than the max view range, so 10,000. So I'll go ahead and say if end minus starts is more than max view range, so this will be 10,000. Then I'll say validation error is equal to selected range exceeds maximum view range of 10,000 base pairs. So because this validation error is null to begin with, so the default value is none and it'll only be set if any of these if statements actually hit, what we'll do is just hop outside this and say if validation error, then we'll set the error to be the validation error and then return. Otherwise we can say set error to null. So if it was currently set, we'll just set that to null. And then we can go ahead and fetch the gene sequence. And then we can pass in the start and the end position, and then update the dependency array down here with the variables we depend on. So this will be the start position, the end position, the fetch gene sequence, as well as the gene bounds, like this. And we can then take the function definition, hop down to where we're going to need it, so on sequence load request, just paste it in right there, save. And now if I put in a number such as 7 right here, so now we should be below the 10,000 base pairs, and then load, we see that something is at least happening. If I drag this, it'll load again. If I drag this, it'll load again. If I drag this, it'll load. So it seems to be working just fine. Let me just check the validation logic as well. So first we can check whether we have a valid start and end position. So okay, if I drag this all the way up and then make this even smaller, so if I do this, now we're 10,003 base pairs, press load sequence, then we aren't really able to see anything. So we don't really show the error anyway, any place right now. So I'll just take this error being passed down in gene sequence. So that comes from the parent up here, error. And then we can go down and just show that whenever it is actually there. So if I just scroll down to right here and then say if display error, then we'll put in a div with the display error, so the error right here, with the class name margin bottom 4, rounded, medium, bg red 50, padding of 3, text small, and text red 600. I'll just save that. And now we can see that the selected range exceeds the maximum view range. And this is then calculated or determined, that is probably the more correct way to say it, in the parent component. So if I just make this a normal range again, it should load perfectly fine. And then for example, if I put in just ASD and say load sequence, please enter valid start and end positions, and we get another number up here. So this all makes complete sense. So the validation seems to work fine. So while we're down here in this code in the gene sequence, I would like to also just show the strand information. So if you scroll up to the parameter we passed down with the gene detail, hop into that, we can see that we have the strand right here. So to do this, to use this, we can show what strand we're currently at. So what strand we have this sequence from. So we can say diff, then we can put in a span. This span should then show either forward strand or reverse strand, or strand information not available if it's not available. So first I'll say class name, margin bottom 2, flex, items center, justify between, and text extra small. I'll then just spell this correctly. Then for the span, I'll put in a new text color, so I'll say text. And then for the color, I'll say hashtag 3C 4D, or sorry, 4F, and then 3D at 70, and then put in the actual strand. So what we can do is say, if we have gene detail dot genomic info, and then we can say dot 0 dot strand. If that is equal to a plus sign, that means we have a forward strand. So we'll go ahead and say forward strand. And this can be marked by 5 to 3. So this notation right here, 5' to 3' just refers to the directionality of the DNA strand, and that is just a notation. So unless if we do not have the forward strand, well then we'll have the other strand, so the reverse strand. So what I'll do is just say if, I'll put in another if statement as this other case. So I'll just take this and then check whether that is equal to minus. If it is equal to minus, then we'll say reverse strand, and then put in 3' to, I'll go ahead and do that, and then to 5'. And if we are not either plus or minus, well that means we probably do not have any strand information available. So I'll then say strand information not available, like this, then just save. So we can now see that we do not have any strand information available. I can just go ahead and actually just print this out just so we can see what it actually contains. So if I refresh over here, hop into a gene, we see that it doesn't really print anything out at all. So it means that we neither have a plus or a minus, we don't really have anything available. Under this, I'll just put in the maximum window size as well. So I'll copy this span start tag, then just put in maximum window size, and that is going to be max view range to local string. And then it's in an amount of base pairs, so I'll just say base pairs like that. So the flex up here within the justify between will then spread these apart. So the one is in the left side and one is in the right side, because this flex div right here is the full width of the entire box. So now we can create the div for showing the sequence itself. So I'll hop down here, put in another div, and then first I'll put in some classes with full, rounded, medium, background, E9 EEEA at 50, and then I'll say P3. So you see this this dark gray rounded box up here. Well, that is what is supposed to contain the ATGs and Cs themselves. Then I'll put in a if statement where I'll say if is loading. Then we can put in a spinner. So if that is the case, I'll go ahead and put in a div and the closing div as well. Inside this div, I'll say class name, flex, items, center, justify, center, py of 8. Inside this div, I'll put in another div. This will then have the class names h5 with 5, animate spin, rounded full, border 2, border gray 300, border t hashtag 3c, 4f, 3d. So I'll just click load up here, and then we see the spinner loading. And if we're not loading, we can go ahead and show the actual sequence. So I'll put in a div. This is just going to be a div that prevents all of the nucleotides from overflowing and gives them a fixed height. That means we can scroll up and down in the height if it's more than that specific height. So I'll say div class name h64. Then I can give, this will then be the fixed height. Then to fix the height, I'll also say overflow x auto and overflow y auto as well, like this. Then because the nucleotide just consists of letters, we'll put in a pre-tag. This is just to format these letters. We'll say font mono, text extra small, leading relaxed. We'll then just create a function for generating all of these ATGs and Cs. For now, I'll just say ATGC something. And then if there is an error, I just want to be able to handle that as well. So first I'll copy this part right here, put in another if statement. So I'll say sequence data. If that is available, we'll put in the sequence data. Otherwise, we'll put in a paragraph. So a P right here. And then I'll say, if we have an error, then I would like to say error loading sequence. Otherwise, we can say no sequence data loaded. If that for some reason isn't there, I'll just say no with a large capital letter. Class name, text center, text small, and then also a text color. And the text color will just be the one we usually do, so the one up here. And then I'll just say /60. Okay, so right now the ATs, Gs, and Cs are just hard-coded, as you see right here. But I want them to both be clickable, but also formatted in a specific color such that every single nucleotide has its own color. And the reason for wanting them to be clickable is because the next card we are going to build is for running a mutation analysis on a specific position with a specific nucleotide. This means that I want to be able to click a specific nucleotide, and then that nucleotide's position will be inserted into that card. And then you just specify what nucleotide you would like to change that to before running the mutation analysis. And this also means I'll create a React component or just a little piece of HTML with some React in it for each one of these nucleotides that allows for being able to click it. So that is what we'll go ahead and do right now. So let's scroll up to right where we start here, right in the JavaScript part. And then we can create a function, or actually a variable, that contains this formatted sequence of nucleotides. So this will be wrapped inside a use memo. And this also means that rendering this entire list of formatted nucleotides will only be done when the dependency array inside the use memo changes. So hopefully not every time the component re-renders otherwise. So I'll say const formatted sequence is equal to use memo. And then we can pass in the dependency array. And then whenever the variables inside this dependency changes, then this formatted sequence will be re-rendered again. All of the code inside this formatted sequence will then be rerun and then saved into this formatted sequence variable. And that is then also what we're going to use down here. So scrolling down to where we hard-coded in these nucleotides, we'll then just say formatted sequence. So right now, this just returns void. It doesn't really return anything. So we can just go ahead and say return ATGC, for example, and save. And now it says ATGC up here. So first things first, we're going to need a sequence, the sequence data and also the sequence range. So I'll say if not sequence data, and let me just say or not sequence range, then we'll return null. Just to not do this, then we'll say const start is equal to sequence range dot start, we'll then say const basis per line, and this will then be 150. So just a definition. And this is just how many nucleotides we want per line, so we know how to format it further down in this code. Then we'll just create a list containing all the lines, and this will then be a list of JSX elements. So I'll say const lines JSX dot element, and we'll just need to import JSX probably. Yep, dot element, a list of those is equal to an empty list. So now let's create a for loop. So we'll say for let I be equal to 0, and I would like to say if I is less than sequence data dot length. In that case, we'll hop. So we'll do one iteration in the loop that is basis per line long. So let's say we have 1,500 nucleotides. Well, in that case, this loop will then run 10 times because we jump 150 nucleotides at a time. This then means we can chunk up the nucleotides into chunks of 150 as well. So first I'll say const line start position is equal to start plus I. Then I'll say const chunk is equal to sequence data dot substring to extract this substring or this chunk from the sequence data. So that's basically what I'm doing. Then I'll say I to I plus basis per line, like this. Then we can say const colorized chars to just contain the JSX element of the actual colorized and formatted chars. This will be a JSX.element and array of those equal to a new empty array. Then we'll just loop through this chunk. So I'll say for let j be equal to 0. Then I can say if j is less than chunk.length. Then we'll say j++. So we'll loop through all the different nucleotides in this chunk. First we'll get the nucleotide, so I'll say const nucleotide is equal to chunk at position j, or I'll just put in an empty string if that is not there. Then we can say const nucleotide position is equal to line start position plus j. And then I would like to determine this color based on what the nucleotide actually is. So I'll say const color, and then it is going to be equal to some CSS class like text green 200 for example. So we'll just create a function inside utils for doing that. So I'll say new file coloring utils.ts. In here I'll say export function get nucleotide color class. We'll pass in the nucleotide, which is just going to be a string because it is a single character. We'll return a string as well. And then we'll just put in a simple switch here that takes the nucleotide dot to uppercase. This means it'll always be uppercase, so we'll avoid confusion about a lower and uppercase nucleotide. That means it won't really match, or sorry, that could cause not really matching any of the cases inside this switch. So I'll then say case A, return, and then for the A's we'll say text red 600, and then we can say case T. If that is the case, we'll then say return text blue 600 instead. And I'll just spell this correctly. I'll just copy these two cases, paste them in right here. So we have A, T, G, and C. And then for the C, we'll go with, for the G, we'll go with green. For the C's, we'll go with amber. And then we'll just have a default. So if none of these are hit for some reason, then we'll just return text gray 500 like this. Then I can just copy this function name, hop back in here, and then just determine the color based on this class where we'll pass in the nucleotide itself. So now we have a string containing the CSS class that is required to color the nucleotide in the HTML up here. So now we can push the actual HTML to colorized chars. So I'll say .push. And I would like to push a span, so just a string. Inside the span we'll have the nucleotide, so just the letter. And we'll just put a key onto this as well. So I'll say key, and the key is just going to be J. The class name is going to, first of all, be a formatted string. We'll put in the color, because that is a CSS class. We'll put in group, relative, and cursor pointer. Save that. Then when it is clicked, we can go ahead and say on click. And when it is clicked, we'll just call the following function. So on sequence click, we'll pass in the nucleotide position as well as the nucleotide itself. And then when any of these nucleotides up here is are hovered, then we'll need to track that as well, so we can show some kind of pop-up about the position of the nucleotide we're currently hovering. Because let's just say we have 10,000 nucleotides in this window and we can scroll up and down and to the left and to the right. It might be hard to determine what position of this specific nucleotide you're actually currently looking at. So by being able to hover one and then seeing the position will make it easier for the user to pick out the specific mutation they would like to test out. So under this drag start x, I'll create a new one. So I'll say const hover position and then set hover position is equal to use states. And the use state should be a number or null. And then I'll say null. And this just needs to be or instead of a comma. Now this hover position up here is to display, to know what to display in this tooltip will show whenever we hover a nucleotide. But we're also going to need the mouse position, so when hovering up here, to know where to display the tooltip on the page. So therefore I'll just say mouse position up here as well. So I'll say mouse position and then set mouse position as well. And the mouse position, if I just define that, that needs to be an x and I and a y value. So I'll say x, number, y, number, or null. And this will just be null by default. So I'll say on null right there. Then I can scroll back down. So right here. And first we can say on mouse enter. So on mouse enter like this. In that case, we'll receive an event. And we'll say set hover position. And we'll set that to the nucleotide position. We'll then also say set mouse position to x is going to be event.clientX, so the mouse position x, and y is going to be e.clientY, so the y position for the mouse. And we'll then just do the same for the on mouse leaf. And then in this case, we'll just set both of these to null because we aren't really hovering this nucleotide anymore. Then after rendering this entire line of nucleotides, we can then say lines.push and then push the entire line. So inside the div, so I'll say div like this, put in a key that's just going to be AI and a class name, which is going to be flex. I'll put in another div in here. I'll say class name mr2 with 20, text to right. So this will align the text on the right. I'll then say text gray 500 and select none. Inside this, I'll put in the line start position .to local string. Right outside of this, I'll put in another div containing the colorized characters, and then I'll say flex 1 and tracking white like that. Now outside this line.push, I can then say return lines. So return lines, save that, and now we can see the line positions up here as well as the colorized characters. We can scroll to the right or left, and we can scroll up and down. So we just need to change a couple of things for the dependency array, because right now this is only going to be rerun and then updated over here the first time the component is rendered. But we would like these to be changed whenever the load sequence is clicked, for example, as well. So we'll need to say whenever sequence data changes, redo all of this. Whenever the sequence range changes as well, redo all of this. And whenever handle sequence, let me just see what it's called, on sequence click, whenever that is called, we'll need to redo all of this as well. So if I drag this now, it redoes everything. So we can see it says A right here as the first one. Over here now it says G, so it does update it as it is supposed to do. If we make a very small window of only 116, then we only see this single line as well. And I see this one error right here. It says text gray as well, so we need to make it actually gray by spelling it correctly. I'll just make sure that I haven't left out any of these or spelled these correctly. So it looks like I spelled these correctly there, there, and there. So hopefully you didn't do this. That is just me spelling incorrectly sometimes. Cool, so now that we have this tooltip up here, I would like to be able to hover some of these and then use the information stored from this formatted. Let me just hover all the way up in the gene sequence, then use this information we store in these set hover position and mouse position to then also show the tooltip, because right now when we hover something, nothing actually happens. So hopping down here, we can render out the tooltip based on this information. So I'll just scroll down to beneath this, And then I can say if hover position is not equal to null and mouse position is not equal to null, and then we'll render out a div. This div will have class name, pointer events none. It'll have a fixed positioning, set 50, rounded. We'll have background of 3C, forref 3D. We'll have px2, py1, text extra small. We'll have text white and shadow medium like this. Then inside this div, I'll put in the position text and then also hover position dot to local string, just to save that. And then this should also be positioned depending on where the mouse is. So look at this right now, it is just positioned down here, but I'd like this to move so it's right above the mouse at all times. So what I'll do is put in a custom style tag with an empty dictionary, and then in here I'll say top. So the top should be based on the mouse position dot y minus 30, just to take account for its height. So now you can see it aligns with the y position of the mouse. We can then just do the same for left to align with the x position. This will then be mouse position.x. We can then just say transform. So if I just comment out this line, you can see why we need transform. So right now it is in the middle of, it is aligned such that the mouse is in the bottom left corner of this label, but I would like the label to be right above the mouse in the center on the x-axis. So I'll just move it half a width to the left. And to do this, I'll say transform, translate x. Now I'll translate it by minus 50%, so half of the width of the tooltip. And then just save that. And now we see it being positioned exactly over the mouse, along with the position of the nucleotide as well. And lastly, we can just make a little reference so the user knows what nucleotide corresponds to what color. So I'll put in a div down here, say class name. In here, I'll then put in I'll just put in mg3, flex items, center, and gap 4 to create some space between these different nucleotides. So we'll have A to G and C along with their respective colors. I'll then put in another div like that. Then say class name, flex items, center, and gap of 1. We can then also say div inside of that. So I'll put in a div, and then this div just needs to be a circle, so just h3 with 3 and rounded full to make it a circle, along with the color BG red 600. We can then just pass in a span as well. This will then be A, so now you can see what I'm talking about. I'll then say class name, text extra small, text, and then we'll use this text color up here at /70. And I'll then just copy this div 3 more times. So A, T, G, and C. And this C will then be amber. The G is going to be green. The T is going to be blue. So now we have the annotations for the nucleotides as well, just to make it a little bit more obvious why we color them in this way. Before moving on, I would like to just do 2 things. First of all, as you can see up here, it says 999 base pairs. That's because we have a small miscalculation up here. So going up to the current range position and where it is being calculated, it should be all the way up somewhere. I'll just say range just range, and then we should be able to find it. So right here, if I put in a +1, we should be able to see the range being calculated correctly. So that was the first thing. Next, I think I made a little bit of a spelling error down here. So it says 4D instead of 3D. So I'll say 3D and 3D, just so we keep consistent coloring around the entire application. Cool. So just to summarize, we're able to click these positions right now. It doesn't really do anything because we haven't changed the gene viewer to be able to handle that with this on sequence click. But we are able to load different sequences, so I can drag around this slider. I can make it smaller or larger. I can scroll around inside of this window of our sequences. I can manually change these as well, like this, load a sequence. I can see error messages. And we have some annotations. So with that done, I would like to consider the gene sequence completed. So we can now hop back into the gene bureau and then move on to the next card. The next card is going to be a card containing the known variants within this gene that we're currently looking at. This means that you can either test out mutations on known variants that currently exist to see how EVO2 performs against those variants, or you can click a position or a nucleotide like this and then manually test a position that you would like to test. So those are going to be two options. So first we'll need to create this card called known variants, and we'll do this inside a new component. So I'll hop under components, say new file, and then call this known variants.tsx. Then I'll just say export defaults function known variants, and just leave the properties empty for now. And then I would like to return just something like a rag fragment until we have it implemented, like this. And then I'll just say return, import this, I'll make it a client component. In here I'll just place it right above the gene sequence. So I'll go ahead and say known variants, import it in, save, and then it should be up here right now. So the first step in showing these known variants is actually going ahead and fetching them, and this will be done inside the gene viewer since we might need these known variants in other components than just the known variants component up here. So first I'll just create the logic for actually fetching the known variants. So under Genome API, we can go ahead and scroll down and then create a new function that can fetch these known variants. So this will just be called fetch ClinVar variants. So I'll say export async function fetch clinical variants like this. I'll pass in the chromosome, which is a string. I'll pass in the gene bounds. So this is where we search for variants in between. This will just be gene bounds. I'll also pass in the genome ID, which will also be a string. And then what we'll return is a promise. Inside this promise, it will resolve to a list of clinical variants. So we'll just define these up here as well. So we'll say export interface ClinVar variant. It will contain a clinical variant ID. This is going to be a string. It'll contain a title, a variation type. It'll contain a classification as well as the gene sort. It will contain a chromosome, a location, and then it'll contain a couple more things probably, which we'll append if we do need them later on. So going down here, we'll return a list of these. So we'll say clinical variant and then a list of those, and then just open up the function. To search for clinical variants, we're going to need the chromosome, the minimum and maximum bound within that chromosome. We'll search the variants within, and then we can build out the UL search parameters from that. So first I'll just get the chromosome in the way we're wanting it. So I'll say chromosome formatted is equal to chromosome.replace, and I would like to remove this CH or CHR text right here that is in front of some chromosomes. And so if, for example, this is the chromosome, it'll remove this CHR and just leave us with the number 17. So I'll go ahead and say replace, and then I can put in a regex. So I'll say / and then up, and then CHR / I, and then we'll replace that with nothing. This will just replace this CHR with nothing. Then we can say const minimum bound is equal to math.minimum, genebound.minimum, and genebound.maximum. We can then also say max bound. This will then just be max right here. Then if you hop into the following URL right here, that is the endpoint we're going to use. So we'll use ncbi.nlm.nih.government/clinical variants and then /docs/linking. So in here we can see what we can use to search. So for the chromosome which we're searching within, we'll need to specify either chromosome position with 37 or 38 down here. These are for the two assemblies HG19 or HG38. So I'll just hop down and say const position field is equal to, if the genome ID is equal to HG19, then we'll use chromosome position 37. Otherwise, we'll use chromosome position 38. And then we can construct the search query. So I'll hop down and say const search term is equal to a formatted string where we put in the chromosome formatted. I'll then put in chromosome right after to live up to their standards. So chromosome. And I'll then say and the minimum bound, the maximum bound, and then also the position field. So I'll say position field like that. The search URL which we're using is going to be defined as this. So it's going to be HTTPS and then eutils. So we actually use eutils up here already. So we can just copy that. So we'll take this entire part, copy it all the way down here. And then we can say const search params is equal to new URL search params. We would like to search within the database called clinical variants. The term is going to be the search term. The read mode is going to be JSON, and the read max is going to be 20. Then we can say const search response will be await fetch, where we pass in the search URL. So I'll say search URL will pass in the search parameters after this query parameter string, question mark. So this will then be search params, and this can be transformed to a string with toString like that. First, we'll just check whether the response is okay or not. So I'll say if not search response dot okay. Then we'll throw a new error saying ClinVar search failed, and then we'll put in plus the search response dot and then status text. Otherwise, we can say search data is equal to await search response dot JSON to just get the JSON for the response. Then we can say if there is no results, so if search data dot search results, if that is not there, or each search search results dot ID list is not there. We can then also say or the length of this list, so dot length being 0. If that is the case, we'll then also just say that okay, now we know that there probably aren't any variants, so we'll just return an empty list of variants, and also say console.log and then no ClinVar variants found, like that. If there are actually variants, we can then get the variant IDs, so we can say const variant IDs is equal to this list right here of variant IDs. And then because we have the IDs but not the information about the actual variants themselves, we'll need to go ahead and create a new call to a database again. So I'll copy this down and then say summary URL, and I can call this summary params as well. So here we can go ahead and say we'll remove read max, and then we'll pass in the ID. That'll be variant IDs dot join, and we'll join them together with commas like this. So it'll take the entire array of variant IDs and make that a string joined together by these commas, and then we can use all those variant IDs to actually get the information about the specific variants instead of only having the IDs. We'll then just fetch in the same way as done up here as well, so we can do the exact same things. I'll paste this in and then say summary URL and then summary params, and here it is going to be summary response, and then I can say fail to fetch variant details, and then I'll also say summary data, and that'll be equal to summary response dot JSON. Now we can then create a list of variants, so I'll say const variants, which is a ClinVar variant list, and that'll be equal to a new empty array. Then I can say if summary data dot results and summary data dot results dot UIDs. Then we can say for const ID of these UIDs in this list. Then what we'll do is then say const variant is equal to summary data dot results ID, and then variants dot push, and then we'll push a new variant to the list. So each variant will have the ClinVar ID, which will just be ID. It'll have a title. The title will be variant dot title. We'll have a variation type that is going to be either variant dot object type or unknown if it is not existent. Below this we'll need I'll actually just format this a little bit. So what this can be is it can return single nucleotide variant, for example. So instead of this, I would like to transform this to single nucleotide variant, but this is uppercase, this is uppercase, and this is uppercase. This means that it'll look way nicer. So I'll say a new line in the dot, and I'll split this text by spacing. So I'll have 3 different elements in the array. I'll have single, I'll have nucleotide, and I'll have variant. I can then say .map to loop through all of these. So .map. I can then get each word inside this map, which will be a string. This will then be mapped to word.character at position 0 .to uppercase plus the rest of the word. So I'll say word.slice 1 .to lowercase. So the rest of the word will then be lowercase. And then I'll join all of these together. So I'll say join by just regular spaces again. Cool. So this will transform the single nucleotide word into with uppercases as I showed you just before. So the next thing I would like to get is the classification. The classification will be variant dot germline classification dot description or unknown if it is not there. We can then also get the gene sort. This will be variant dot gene sort or just nothing if it is not there. We can then also get the chromosome, which is just going to be chromosome formatted, as well as the location. This will be variants dot location sort. If that is there, we'll then say pass ints, and we would like to pass the variants dot location sort dot to local string or unknown, like this. And it seems like we have a small error. We just need to put in a colon instead of the question mark. Then I see a small error up here. It says variation type. It needs to be variation type. So I'll just hop up, and then we can see variation type. We just need to correct this spelling issue. And then hopping down, that should actually fix that. And then this is also chromosome and not chromosome. So I'll say chromosome like this, and just scroll up and fix that as well. So these are just small spelling errors that just needs to be fixed for this software to work. Then I can just return the variants. So I can return the list of variants like promised by the method up here, a list of clinical variants. Then hopping back into the gene viewer, I would like to create some states for tracking the both loading of the clinical variants as well as the error and then the clinical variants themselves. So I'll say const clinical variants, and then we'll set that as well. So set clinical variants is equal to use states clinical variant. So just an array of those, and we'll set a empty array as the default value. Then I'll just copy this line and say is loading, and then just ClinVar, so to keep it a little bit simpler. And then we'll say set is loaded clinical variant again. And then this should just be a boolean, so we'll just set this to false because it is not loading per default. Then we can just say ClinVar error and set ClinVar error. and then this will just be null, and it can either be a string or null, like this. Then hopping up here, we can then import, this just needs to be type, we can then import the fetch clinical variants as API fetch clinical variants, like this, because our method right here in the gene viewer for doing this will probably also be called something like fetch clinical variants, so we'll just want to avoid a name conflict. So scrolling down, we can then create a function for actually fetching these clinical variants. So I'll hop down and say const fetch clinical variants, and then this will just be equal to a new function. So I'll go ahead and say async function. First I'll check whether we have the gene.chromosome and whether gene bounds is available. Then we'll return if they are not. Then we can say set is loading clinical variants, and we'll set that to true, as well as just reset any errors we might have for the clinical variants. So set clinical variants error. Then we can try and fetch these clinical variants. So I'll say try catch the error, and I'll put in a finally as well, wherein we set the is loading ClinVar to false. Inside the try, I'll say const variance is equal to awaits API fetch clinical variants. We pass in the gene.chromosome, the gene bounds, as well as the genome ID, and save that. We can then set the clinical variants to be variants like this. And then if an error happens, we'll just set the clinical variant error to be filled to fetch clinical variants like this. And if that happens, I'll also just clear the clinical variants. So set clinical variants to an empty array like that. And these clinical variants should then be fed whenever the component loads. So I'll say use effect. And then I'll pass in a dependency array only depending on the gene bounds and also the function itself. So I'll go ahead and say fetch clinical variants, and then I can say if gene bounds are there, then I'll just call this function and fetch the clinical variants. Now in here, I would like to just print out the clinical variants. So I'll say console.log variants, just to make sure that we actually see something. So hopping back, variant analysis. First, we just need to go ahead and remove this though, because we don't really want to depend on that. It might cause it reloading a bunch of times. Over here, I'll just refresh, open F12, and then we can see we have an array of an empty array and then no clinical variants found. If I try some other genes, no clinical variants. What about this one maybe? Still no. So what I did was I looked into this and I found that under Genome API, we made a small error. So instead of eSummary for both the summary URL endpoint up here, and also for just searching up the clinical variants is not the greatest idea. We need to use eSearch instead. So for eSearch, with eSearch, you can look up the IDs of the clinical variants, and in the eSummary endpoint URL, you can look up the expanded and more detailed information about each clinical variant. So I'll go ahead and just save that, and then load up an example over here. So I'll just take the first gene and then just click this array over here. So now we have all of these different clinical variants. If I go back and try the other ones, we get no clinical variants for that one. We do get clinical variants for this one though. Also for the fourth one. So we are getting clinical variants now. So that is a very promising sign for our application. Cool, so with the clinical variants fetched, we can go ahead and use them inside our new component, known variants in here. So first we'll define the props. These will obviously include things such as the clinical variants, but also functions to go ahead and refetch the clinical variants if that is needed. First we'll have refresh variants. So refresh variants is going to be a function called when the user clicks a refresh button to just refetch these variants. We can then also pass down a function called show comparison. So whenever a user clicks the analyze button for a specific variant, then this function can be called to display a comparison modal for the variant analysis and results. So the refresh variance is going to be a function returning void. And the show comparison is going to be a function taking in variant, which is of type ClinVar variant, and it'll return void as well. The next one we're going to pass down is going to be update clinical variant. So I'll say update ClinVar variant. And this is going to be a function that can update the state of a specific clinical variant in the parents component list of clinical variants up here. So this will just be a function that takes the ID of a clinical variant as well as the new variant, which will be a ClinVar variant. It'll just return void as well. Then we'll pass down the actual clinical variants, so ClinVar variants, and that is going to be a list of ClinVar variant is in an array. We'll also have is loading clinical variant, which will be a boolean. We'll have ClinVar error, which will be a string or null. There'll also be genome ID, which will be a string, and gene, which will be gene from search. So the card itself, which we'll return, is going to look like this. We'll first put in a card component, and then we'll put in class names gap 0, we'll put in border none, bg whites, py 0, and shadow medium, or sorry, small, like this. Then for the card, it looks like we need to import that. So if I hop in here and just select the first one, we should be able to see a card whenever we just pass down the properties. So I'll hop in here and just pass down the properties so it actually is fixed. So I'll do this, and then refresh variants is going to call the fetch ClinVar variants. The show comparison we'll just leave as an empty function for now. We'll implement that later. The update ClinVar variant will also just create a function for. I'll just hop up here under these functions and just say const update ClinVar variant. And remember, this took a ClinVar ID, which is a string, and it took the updated variant, which is just a, let me just say just updated variant, which is just a ClinVar variant. And then we can say set ClinVar variants, and we'll first of all get the current variants, and then we'll say current variants dot map. So we'll loop all over all of these variants, and then if we have one variant where the ClinVar variant matches the parameter, so the one we're looking for right now, this one up here, then we'll update that. So we'll say updated variant, otherwise we'll just use the old one. And this one of course just needs to be a normal equal sign. So we pass in the ID of the clinical variant we would like to update along with the clinical variant that we are updating, so all the new properties in this clinical variant. Then we set the clinical variants. To do this, we'll loop through each of these to recreate the list with map. So map just returns a new list based on the updates which we define in here. So we just return the old one if the clinical variant of the current one in the we're iterating over is not the same as the clinical variant we're looking for. Otherwise, we just use the updated variant if that is found. So that is the basic premise of how this works. So we can scroll down and just use that right here, like that. And then for the clinical variants, we can say ClinVar variants is loading ClinVar. We can say is loading clinical variants. We can also say clinical variant error. Genome ID will just be genome ID, and the gene itself can then just be gene. So now we should be able to render out the card in just a second. So over here, I'll put in a card header. The card header should have the card title, which should say known variants in gene from clinical variants. And then we just need to import the card header and the card title. Save that. And then let's just open this up and see if we can see it. And now we can see the card. So first, the card header, we'll put in flex row, items center, justify, between, pt4, and padding bottom 2. And for the card title, I'll say class name. I'll then put in text small. I'll put in font normal, and then text, hashtag 3D, forref, and then 3D again. And then I'll make this 3C actually, and then /70. So now it'll look like this. Then we'll just put in a button that allows us to refresh the variance. So right next to the card title, we'll then say button, import that. And then the text should be refresh. And now we just need to style that to make it look a little bit better. So we can put in, first of all, a variant that is going to be ghost. We'll then put in a size that'll be small. We'll then put in an onClick. The onClick is going to be refresh variance. So this one right here. And we just need to pass down all these properties, destructure all the properties. So that is what I'll just do now by copying in each of these from the list of types. Genome ID as well. And then also the actual gene. So now we can say refresh variance. And then we'll also just disable it. So we'll say disabled when is loading clinical variance. I'll then hop back in here and then say class name h7, cursor, pointer, text extra small, text, and we'll just use this style again. I'll just say hover actually, and then background. Then I can say E9EEA/70. And then this is what we'll get up here. And then when I click refresh, it should refresh for a second and then be available to be pressed again. So that was the card header. Then for the card content, what I would like to do is just go ahead and say class name, padding bottom 4, save that. Inside the card content, we can put in the error first of all. So if there is a ClinVar error, then I would like to render out this div right here, where we say clinical variant error. So I'll just save that and then say class name, margin bottom 4, rounded, MD, BG red 50, padding 3, text extra small, and then also text red 600 like this. So we can just test it out with an error. So let's say this is just going to be a default value of haha. And then in here, we'll just pass down nothing. So the error, let me just remove that. Let me just pass down something instead. That is way easier. So I'll just do this. Now we can see the actual error, clinical variant error. So now we know it works. I'll just remove this again. And then under the error, we can say if we're currently loading. So is loading clinical variant. Then we'll put in a div. We'll then put in another div here with a loading state. So this div will be h5 with 5, animate spin. We'll also put in rounded full. We'll put in a border of 2 and a border color. And this border color should have 3C, 4F, and then 3D at 30. And we'll put in a border top with the same border color as well. So this one right here. For this div, so the outer div, I think we have an error. We need to just put in the end quotation mark, and then also just a reg fragment or something right there. So for the outer div, we'll put in a class name of flex, justify, center, and py8 to create some spacing above and below this spinner right here. So when I save that, we can then just put in the loading, set that to true, save that, and now we see the loading state right here. So I'll just say is loading ClinVar, and we do get an actual error over here. So that is probably something we want to take a look at a little bit earlier. Failed to fetch. So that might be because we're fetching a little bit too often actually. So we might want to put in some logic that doesn't fetch too often, but let's just take a look at that a little bit later. Alright, so moving on, hopping down to if we're not inside the loading state. So if we're not loading, well, we want to show these clinical variants. So I'll say if clinical variance.length is more than 0, then we'll show some regs. Otherwise, we'll just show a reg fragment for now on. And then inside this div, I'll give it a class name, age 96, and then max age 96 as well. And then I'll say overflow, y, scroll. This means that it won't become larger than age 96. So this is going to be this many pixels. And if it is larger than that, then it'll just be scrollable inside the box. Then we can say rounded, MD, border, and then give it a border color as well. Like that. And then I'll just say /5 and save that. So this is going to be the box containing the known variants. Inside this box, I would like to put in a table. So I'll say table from UI table, so from Shadcn. Inside this table, I'll put in a table header. The table header should contain a table row, and a table row should contain a table head, and a table head should contain variant, for example. So I'll just import the table row, and then for the table header, I'll say class name, sticky, top 0, and set 10. Save that, hop back in, and then let's see if we can see the table. So now we see variant right here in the top. And then for the table row, I'll say class name, background, E9, EEE, A at 80%, hover. And then when we hover, we want to change the color a little bit. So I'll just say, let me say 60, for example. So it'll look like this. And that is too little of a difference. I'll just put in, let me say 30. So now we can also hover this table header. For the head itself, we'll give it some classes too. py2, text extra small, font medium, text. And the text should then be the same color as up here. So that one right there. And now we can just take this table header and copy it 3 more times. We'll have a type as well, and then also clinical significance, and we'll have actions. So these will be the 4 table columns we'll have in our table. And then for the table body, we'll say table body, and I would like to put in, I'd like to loop through the clinical variants. So I'll say clinical variants.map variants, and then for each variant, we can then return a table row. The table row should have a key first and foremost, so I'll say key, and that'll just going to be variants dot clinical variant ID, and then also some table cells. So I'll say table cell, import that, and I'll also just do some styling. So I can say class name border. It has to be out here. Class name border bottom and border. And I'll just take the same color as we always use up here at 5%. So the first table cell right here should have class name py2. We then want to say div variant dot title. Save that. And then we have a little issue. So you see we have this big red error. So error. So we need to exchange these yellow ones with normal braces instead of curly braces, and that should fix that error. Then if I save again, we should be able to see the variant names over here. So after the variant title, and I'll just style this, I'll say text, extra small, font medium, and I'll say text, hashtag 3C for ref, and then 3D at 70%. So it'll look like this and be a bit smaller as well. And remember, we can scroll through the table like this if it is larger than it is supposed to be. Then we can just put in the location. So I'll say div, and I would like to put in just the same classes right here in this div, but I'd like to also just put in margin top 1, flex, and gap 1, and remove the font normal. So if I save this and then put in a paragraph saying location, and then I'll put in variant dot location, save that. Now we'll be able to see the location as well. Then up here, I'll just remove this 71 to make it a little bit more distinguished. And then I can put in a link where you can click and then be taken to the actual variation itself if you want to. So I'll say button, Inside the button, I'll say view in ClinVar, save that. And this is pretty ugly, so I'll say variant. And the variant should be a normal link. Size should be small. The class name should be h6. Cursor points up. It should be px0. Text extra small. It should be text. And then DE, so I want it orange, 82, 46. I then want, whenever we hover it, I should have the same text color right here, but just toned down to 80 instead. So when I hover it, it'll look like this. And then you see how this location and the link isn't aligned on the same place on the y-axis. Well, that is because we need to put in an items sensor to align items on the sensor on the y-axis. So when I save that, it'll look way more aligned and way better. Whenever this viewing ClinVar link is then clicked, I would like to control that behavior as well. So we can say on click, and then we can say window.open. So we open up a new link, and this will be https // and then www.ncbi.nlm.nih.government/ClinVar/variation/ and then we'll insert the variant, the clinical variation ID, like this. And we would like to open this as blank as well, so we open it in a new tab. So whenever I save that and hop over here, if I click view in ClinVar, that should open up that specific clinical variation right here in a new tab. So that seems to work fine as well. And then we can also just put in a small link here. So we'll say external link. We can put in class name, that'll be margin left 1, inline block, h2 and width 2 as well. So now we have this massive link. So if I click that, the entire thing will just take us to the clinical variation webpage. So let's just create another table cell. So I'll say table cell. In here, I'll just put in the variant.variation type. And I'll put in some class names, py2 and text extra small. So it'll look like this, single nucleotide variant, but there are also deletions, duplications, insertions, and other variants in a sequence that you might experience. We can then just put in one more table cell. So if I take this and copy it down, inside this one we can put in a div. This div should contain the variant.classification or unknown if it's not there. So I'll just save that, make this a little bit larger over here, and then we can see uncertain significance, likely benign, pathogenic, etc. for each clinical variant. Then I would like to color these based on the significance, so pathogenic should be red, for example, and then likely benign should be green, such that it is more obvious to the user which ones are most harmful and which ones aren't. And using colors is a great way to do this, so you can quickly gather the information about which ones are in certain categories without even thinking about it, just because you see the colors. So hopping down here into this div, I can say class name, and I'll make this a formatted string. Then we can put in width fits, we can put in rounded medium, px2 to create some spacing on the x-axis, py2 to create some spacing there as well, text center, font normal. So let me just hop back, open this up. So right now there is really nothing, so we need to put in some coloring, and this should of course be determined dynamically based on if it says uncertain significance, benign, pathogenic, etc. So we'll hop into utils and just create a function for that. So inside coloring utils, we'll create a new function. So this one will be called get classification color classes. So I'll say export function get classification color classes. Classification is going to be a string. So that'll be the classification right here, for example. So benign, pathogenic, etc. And it'll return a string as well. And these are going to be the CSS classes to color this. So first I'll say if not classification, then I'll return bg yellow 100 and text yellow 800. Then I'll say const lowercase class is equal to classification dot to lowercase. Then I can say if lowercase classes dot includes pathogenic. Well, then it is pretty bad, so I would like to color that red. So then I'll say return bg red 100, text red 800. Then I can say else if. So else if lowercase class that includes benign. Then I would like to just color this green instead. So green and green. Else I'll just return bg yellow 100 and text yellow 800 as well. I'll save that, copy the function name, hop back into known variants, and then we can plop this function in right here. So I'll go ahead and do this, and then pass in. Let me just import this function though, and then we'll just use variant dot classification, press save, and now we see the coloring. So likely benign, pathogenic is yellow though, so we just need to fix that. So hopping into the function again, so I'll just click this one, press F12. We should be able to see that, oh, this needs to be text, and also, let me just save that. And I also need to spell it correctly, so this needs to be pathogenic right here. So now it is colored correctly. So pathogenic is red, the uncertain is yellow, otherwise they are green if they are likely benign. Good. Then under actions over here, I would like a button where you can press analyze with evo2, and then it'll compare the result from analyzing this mutation with evo2 to what was the result of what is the actual clinical significance of the clinical variant. So when you click over here on analyze with evo2, then hopefully evo2 also predicts that it is likely benign, but then you can at least compare the evo2 result with the clinical significance. So to store the results of the evo2, I would like to hop into the variant, clinical variants over here, and then let me just find out where that definition is. So I would like to add a property called evo2 result, and that should be optional, so it doesn't come from the API, but it's something we can attach later on when evo2 has predicted some result. So this evo2 result is going to contain a prediction, which will be a string. It'll contain a delta score, which will be the number, so that is the number we saw from the backend that we calculated. It'll also contain a classification confidence, which will be a number as well. Then I would like to also just have is analyzing. That'll be a boolean that is optional, and I'll have evo2 error, which will also be optional, and then be a string. This means we can show the data on a row basis whenever evo2 is both analyzing and when it has analyzed. So scrolling down, we can then use this data right here. So if evo2 has analyzed the current variant in the table, I would like to create an if statement for that. So I'll say variant.evo2 results, and then and diff class name mt2. I'll put in another diff into this. This should have the class name that is a formatted string. So I'll put in flex with fits. I'll put in items center, gap 1, rounded, medium, px2, py1, text, center, and I would like to put in the get classification color classes, and this should be based on the evo2 prediction result. So this will be variant.evo2 results.prediction, because evo2 is also going to predict either benign or pathogenic. Inside here, we'll put in a little shield icon. This shield icon will just have a class name of height free and width free, like this. And we just need to import that, I think. And under this, we can put in a span. This span will just contain evo2 along with the prediction results. So a forced spacing, and then I'll just say variant.evo2 results.prediction, like that, and then just press save. So it would look something like this if we just have to test it out. This would then be benign, for example, and in here this would then also be benign. So it would look like this. So you have the uncertain significance from the actual database, and then you have the prediction from evo2, and then we can compare these two directly like this. Good. So that is what we would expect to see whenever it runs in real life. Then under this table cell right here, we'll copy that, put in a new one with a div first and foremost, with the class names flex column, items end, and gap1. So currently, if evo2 has not been analyzed for this specific row, I would like to show a button that says analyze with evo2. If the evo2 analysis has been run, then there should be another button that says compare results. When clicking this button, it should open up a modal where the results, where the difference between the actual database results and then evo2 results can be compared. And then if you just click the analyze with evo2 button and it is loading, then it should change the button state to be analyzing instead of analyze with evo2. And the analysis with evo2 should also only be available for single nucleotide variants. So our backend with Modal, which we created, only allows for analyzing mutations with single nucleotide variants. It does not allow for deletions, duplications, insertions, etc. This can be created pretty easily, but right now we only support these single nucleotide variants, so we also only need to show the buttons in rows that says single nucleotide variant. So that is what we'll do. Let's go ahead and implement these different cases and get started. So first we'll say variant dot variation type, and then we can say dot to lowercase, dot includes, and then we only want to show these in rows that says single nucleotide. So if this is the case, then we'll show some HTML, otherwise we'll just show null. So that is the first case. This filters the buttons away from all rows that aren't single nucleotide variants. So we have put in h1 for example and say hello, like this. Then we can see that this hello is only in front of the single nucleotide variant rows. The next statement we're going to base our rendering off of is whether evo2 has already been ran or not. So we'll put in another statement. So we'll say not variant dot evo2 results. So if evo2 has not run, we'll create a button. So we'll say analyze with evo2, something like that. Otherwise, we'll create a button that says compare results. So I'll just put in a button up here as well. So I'll say button and button here. So because we haven't run evo2 with any of these, they're all going to say analyze with evo2. So that matches what we're expecting. If I remove this negation sign and save, now they say compare results. I just need to say compare results instead of t-results though. Then inside this button, I'll create another if statement. I'll say if variant.is currently analyzing, then we'll go ahead and say span. I'll put in a span, and this is going to be the loading state. And then I'll say, let me just remove this and let me just add analyzing next to that dot. Otherwise, we'll need to put in an else statement, and here we'll also return some React fragment. In here we'll say sap, so put in a sap icon from Lucid React, and then we can say analyze with evo2, and I'll just move this text outside the sap button, so it'll look like this right here. And then inside the sap button, I'll say class name mr1. I'll put in inline block, age free, and width free as well. And then we can just style the general button up here too. So I'll say class name, and then first of all, I'll say variant outline. I'll say size small for the button. Then we can say class name over here. The class name should be h7. It should be cursor, pointer. It should be border, hashtag, 3c, 4f, 3d, at 20%, background, e9, eea, px3, text, extra small, text, at 3c, 4f, 3d, and then whenever you hover it, then we'll change the background just a little bit. So I'll just do this and change it to 10. So now the button is going to look something like this. And the button should obviously be disabled whenever the variant is currently analyzing. And then when you do click the button, we say on click. And then we call the analyze variant function, which we'll go ahead and create in just a second. So right now I'll just say analyze variant and just copy this right here. Oh, sorry, comment out this because we haven't really implemented the analyze variant method yet. Then let's just design this is analyzing. So I'll just copy this line and set it to true instead. This way we can see what it is going to look like whenever we're analyzing. So when analyzing, we'll have class name mr1 inline block age free with free animate spin rounded full border 2 border, and then we'll just use the same color at 30%, border top same color again like this. So this is going to be how the analysis state is looking. So I'll just set this to true as well. So it'll be disabled. So this is how it's going to look whenever eval2 is analyzing. Cool. Then I'll just go ahead and say, bring this back to normal. So this needs to be commented in again. And this needs to be removed. And then down here at the bottom, we'll say variant outline size small, class name, and the class name should be something like, and let me just go ahead and test it out. So if I remove this negation sign, we can then see the compare button or compare results button. So the class name should be h7 cursor pointer. We should have border green 200, bg green 50, px free, text extra small, text green 700, and when it is hovered we say bg green 100. So if I save that, we can see a green compare results button, and then I can just put in a bar chart 2 as well, and this should have a class name, so mr1 inline block. Let me just do this correctly, class name, and then mr1 inline block age free with free, and I can just import this from Lucid React. So the compare results button is going to look kind of like this, and when clicked it is going to call the show comparison function from the parent. So on click we'll say show comparison and pass in the variant like this. And now that that is done, I'll hop back up and bring back the negation sign right here, because we haven't really analyzed these with eval2 yet. So right now they do nothing. So we can actually go ahead and create this method called analyze variant and just get this done. So we can use this both here, but also in the next card, where a manual analysis can be run by putting in a position, for example by clicking one of these, and then putting in the mutation you would like to change it to. So I'll say const analyze variant is equal to async. So when you analyze a variant, you pass in the variant, which is ClinVar variant. Inside this function, we can say let variant details be equal to null. The position of the variant we can get from the location right here. So as you see, it includes some commas. So what we can do is say const position is equal to variant.location. If we can pass it to an integer, so I'll say pass int and say variant.location.replace, and I would like to replace commas with nothing. Then we'll use that, otherwise we'll just set it to null. And now we should then have the position as an integer or null over here. Then we can say const ref alternative match is equal to variant.title.match, and I would like to match with a specific regex. So I'll say /, and then if you see up here at the single nucleotide variants, this is going to be the title. So from this title we can then find out both what the reference and what the alternative is. So right here we have the reference, and then right here we have the alternative. So this is what was before the mutation, and this is after the mutation. So we'll need to capture both the reference and also the mutation, and the way we can do this is with a regex. So this is going to be a capture group. We'll capture a single word. And then we can also use the sign right here, the larger than sign, that lets us know that we have the reference and then the alternative. So I'll go ahead and do this, and then put it in another capture group, say capture another word, and then end the regex. And then from this captured ref and alternative, we can go ahead and extract them. So I'll say if ref alt match and ref alt match dot length is equal to 3. Then we can say variant details is equal to a new dictionary where we have the position, we have the reference, which will be ref alt match 1, and the alternative will be ref alt match 2. So that is what we capture inside these capture groups right here, defined by these braces. So what this regex does is that it just looks for a pattern like this. So a pattern where you have a word, then you have this larger than sign, and then you have another word. And then we'll be able to extract the things that we defined by the capture groups. So now that we have done this, we just want to make sure that we have captured what we need. So I can say if not variant details or not variant details dot position. Let me just copy this. Or not variant position dot reference or not variant position dot alternative. If that is the case, we'll just go ahead and return immediately because we don't really want to move forward with the function if this data is not correct. Then we can update the clinical variant in the parent. So we'll say variant dot clinical variant ID, and we would like to update it to, we'll keep all properties, and then we would like to update the is analyzing to true for this specific clinical variant. This means that in the parent or in the list of clinical variants, for this specific clinical variant which we're analyzing, we'll set the is analyzing property to true, and this lets us update the loading state such as the one over here inside the button. Cool. So under this we can say try. Inside the try, I'll just put in a catch as well. So inside the try, what we'll do is first of all analyze the variant with our API. So we'll need to just go ahead and hop into the Genome API and create the method for doing that. So I'll say export asynchronous function, analyze variant with API. I would like to pass in a position, which is the number, alternative, which is a string. I would like to pass in the genome ID, which is also a string, as well as a chromosome, which is a string. Then I can just take these for properties and destructure them like this, and just use commas instead. Then what I'll do in here is say we are going to return a promise. So this promise is going to be an analysis results type. So I'll just hop up here and create that type, which will return. And that is just the type that is returned from the Modal API, which we created. So I'll say export, interface, analysis, results. And this will then be position, number, reference, which is going to be a string. We'll have the alternative, which will be a string. So these are two nucleotides, and that is the position where the single nucleotide variation happens. Then we'll have the delta score, which will be the number. We'll also have a prediction, that'll be a string 2, and we'll have classification and confidence, and that'll be a number. So I'll just save this and then scroll all the way down, and then we can return that type. So I'll say analysis results, and then inside the method, we can first create the query parameters for using our API. So I'll say const query params is equal to new URL search params, where we pass in the variant position. That is going to be position.toString. We'll pass in the alternative, which will be alternative, we'll pass in a genome, which will be the genome ID, and we'll pass in the chromosome, which will just be chromosome. And then for the base URL of the API endpoint we're calling, I'll get that by hopping into Modal and just copying this base URL right here. So I'll copy this string, hop back, and then I'll go into my env, so my environment variables file, and then I'll say next public analyze single variant base URL, and this will be equal to the string which we just copied. I'll copy this name of the environment variable file, hop into the environment.js, and then I'll define it under client right here. So I'll define the name, and then down here, I'll paste it in right there and right there. This will let us access our environment variables in a type-safe way, and this will also ensure that our environment variables are actually there whenever we build our application. So the URL can now be constructed, so we can say const URL is equal to a formatted string, where we pass in the environment variables, so we can import that from right here, dot next public analyze single variant base URL, and we'll then use the query parameters dot toString and append that to the base URL. Then we can, under that, call the URL, so we'll say const response is equal to awaits fetch. We'll fetch the URL with the method post, like this. Then we can say if not response dot okay. Then we can say const error text is equal to await response dot text, and then we'll throw a new error where we say failed to analyze variant, and this depend on the error text. Otherwise, if the response is okay, we just return awaits response.json, and this will just be returned as this analysis result type. So that is what we can expect to receive in here in known variants. So now we can go ahead and call that. So I'll say const data is equal to awaits analyze variant with API. We'll pass in the position. That'll be the variant details dot position. Let me just see. Then we'll say alternative, which will be variant details dot alternative. We can then say genome ID, which will be genome ID, and we can say chromosome, which will be gene.chromosome. So this should then call our backend in Modal and then do the analysis of the mutation from already known clinical variants. And then after this has been done, we can go ahead and update the variant to remove the loading state again, and then also attach the evo2 result such that we can show the button that says compare results, for example. So I'll say const updated variant. This will be a clinical variant, and that'll be equal to the variant. So this will destructure all the props of the current variant into this dictionary, and then we would like to update is analyzing to false, and we'll add this new property evo2 results, and this will be equal to the data. And now we can just call the update clinical variant with the variant dot clinical variant ID as well as the updated clinical variant right here. So I'll just say updated variant and updated variant, and then we can call the show comparison to show the modal with the updated variant. And then if I just take this update clinical variant and paste this into the catch as well, I'll just take the try statement actually and wrap that around because we didn't really do that yet. So I'll wrap it around so it tries, the try statement contains analyzing the variant with the API. So this involves fetching some external data source. So this means I'll just wrap this in a try just to make sure that we can handle if it fails. Then I'll take this update clinical variant, put it into the catch statement, and then I'll just copy this part where we created the updated variant. And then if the fetching fails, then the evo2 result is not going to be there, and is analyzing will be false, but we will attach the evo2 error. This is going to be if error is instance of an error. Then we'll put in error.message. Otherwise, we'll just say analysis failed. And then we can just call this error instead, and then it should work. So if I now hop over here and just refresh the page, click try BRCA1 example, and then we select a single nucleotide variant. So this one is likely benign. Let's try and click analyze with evo2 and just see what happens. So I think we need to add this analyze variant. It is actually unused to down here. So I'll just do that, comment it in again, and then add these two braces. Under analyze variant, we'll just need to pass in the variant as well. So I'll do this. And then over here, if I now click analyze with evo2, we see the analysis getting started. If I click the network tab, we should be able to see the external API being called too. So let me just click network. Then right now a pending request is being handled. Then under here, we should be able to see something being called. So we see pending right here. So it did call our endpoint. And now just need to wait for it to finish. It does have a cold start and having to load in all of the model file into the memory of the GPU. So it will take a little while for the first time. I will come back whenever this is done. So it just finished, and now we are also able to see, let me just close here, that evo2 classified it as likely pathogenic, and then the single nucleotide variant is actually likely benign. So there's a difference between these two. Right now, if I click compare results, nothing happens, so we can't really see a modal showing the confidence, etc. So that is probably the next thing I would like to implement. If I click analyze with evo2 again, then hopefully it is a little bit faster this time because everything has been loaded into memory, so there are no cold starts. Let me just click the rest of the examples down here. This one, and this one. So evo2 predicts likely pathogenic for all of these. So this makes me think that there is an issue. So hopping into F12, let me just see what the requests actually contained. If I go ahead and just do this again, so I hop into this one and just analyze these with evo2. We can just check what the request contained and see whether this was correct or not. So I'll say payload, and then the genome seems correct, chromosome seems correct, alternative seems correct, but the variant position is not correct. So as you see right here, it says 43092. So this is only the first 5 characters of the actual location. But really it seems like it filtered away this last part, which is obviously also part of the location. So this means that the request probably can't find the correct, the evo2 model, the backend in Modal probably can't find the correct sequence because this location, it doesn't even exist within the chromosome. So we'll just get some random response back like the pathogenic. So if we hop into Modal, for example, and then click one of these requests, what we can see is that it fetched the genome sequence, and because the location is just some random location that doesn't exist, we get n, which is just a sequence of characters that doesn't exist. It should be ATGCs, so this will obviously give a score that is very harmful from evo2 because it has never seen this before. So we just need to fix this. It seems like a pretty easy fix that we need to do. We just need to make sure that this extra 3 numbers that are part of the location is part of what is being sent to evo2. So I looked into this for a couple of minutes, and what I found is that whenever analyze variant is called, this replaces the first comma with nothing. But if you want to replace all commas, and there are 2 commas in the location, if we take a look, so there are 1, 2 commas, we'll need to say replace all. So if we replace this, replace with replace all, and then save, then we can run the analysis again. So I say analyze with evo2, and then if I check the request right here, then we see that the full variant position is sent with the request. Now if I do this for the rest of these examples, this one, then evo2 will say likely benign for most of them, likely benign, probably also right here. Then if I go back to results, we can test some other ones as well. Let's see if we can find some pathogenic mutations. So these ones are likely pathogenic, so we'll just analyze these 4. evo2 predicts likely pathogenic, benign, and benign. So it is surprisingly accurate actually from the results that we have used. So that is very cool. Hopping into variant analysis in Modal right here, we can just check out what it prints out. So as we can see, now it prints out the actual sequence which it found right there, and a different sequence right here, and then a different sequence down here, GEC, different one down here as well. So this means that it is working as it's supposed to do right now, which is great for us. Cool. So let me just close down the terminal, or F12. The last thing I just want to do here is put in an empty state. So right here we see that there are no variants in the gene. So I would like this to be shown a little bit nicer. So down here, this is what checks if the length of clinical variants is more than 0. So if we hop down, we can just put in an empty state. So this is going to be a div. Inside this div, we'll put in a search icon. Inside this search icon, let me just do that. We'll say class. I just need to import it. Then I'll say class name MB4, height of 10, width of 10, and then text gray 300. Inside the div, I can then also, right below this, I'll actually go ahead and say no ClinVar variants found for this gene. And now we can at least see this, and we just need to style it. Then I'll say, I'll write this in a paragraph first of all. This paragraph should have the styling class name, text small, leading relaxed. So text small. And then we can just center things. So I'll say flex. This is kind of annoying. I'll say flex, and then height 48, flex column, items center. Just save that. Now it centers things a little bit. Then we can say justify center as well. We can say text center, and we can say text gray 400. So this will be our empty state. And then if I go into something that has clinical variants, then it'll load exactly like it's supposed to do. Alright, so one more thing I just want to add is just an icon for this refresh button over here. So if I hop in, I'll just say refresh, and then CV as the icon, and then I can say class name, and then do something like MR1, age 3, and width 3 as well. If I save that, now we have a nice looking refresh icon as well. That should be it for the known variants component, and the next step is going to be creating this comparison modal that where the button shows up for when you click analyze with Evo 2. So it says compare results right here. So that is the button you see whenever you run the analysis. This will then call the show comparison function passed down from the parents. So show comparison is not implemented yet. So this should be a modal which we implement right here in the parent, and then this can be shown with the new prediction from Evo 2 whenever it is clicked. So let's go ahead and do that. First, if we hop inside the gene viewer and then scroll all the way up to the states, I would like to create a new state. I would like to call this state comparison variant. So I'll say const comparison variant, and then also set comparison variant. This will be equal to use states, and it'll store a ClinVar variant or null if the comparison has not yet been done. If the modal should not be shown. So the variant stored inside this use state is the one that should be stored within the comparison modal. And this one we can create down here as a component for itself. So I'll hop under the Explorer, File Explorer, then go under Components and create a new file. This file should be called variant comparison modal.csx. Inside this one we can then export a component corresponding to that name. So I'll go ahead and say export function variant comparison modal, and then for the properties I would like to receive, we're going to have comparison variant, and this will then be a ClinVar variant or null if it should not be shown, and we'll also receive the onClose. So what happens when you click the close button, and this should then probably clear the comparison variant in the parent state. So I'll then just say void accept these, destructure these, open up the function, and then first we would like to not show this comparison modal if these are not, if the result is not here in this variable. So I'll say if not comparison variant or not comparison variant.evo2 results, then we'll just return null. So this will just render nothing. Otherwise, we can go ahead and render out the actual variant comparison modal. So this will first of all be a div. I'll say fixed as class name. I'll say inset 0, set 50, flex items center, justify center, bg black 50%, and a padding of 4. Inside this, I'll just put in an h1 that says hello. Then I can take this variant comparison modal, hop back into the gene viewer, and then we can paste it in right here. So we'll just import this component, add the import, as well as the properties. So comparison variant will be comparison variant. And whenever the close button is clicked inside the variant comparison modal, we'll go ahead and set the comparison variant to true. So I'll say set comparison variant, sorry, not to true, set that to null. This means that it'll no longer be rendered because as you see in here, if it is null, we'll just return null. So the comparison variant modal will not be rendered. We can then also just scroll up here and then create the function that we can use to actually show this modal whenever it is clicked right here. So under known variants, if the show comparison variant modal is clicked, so if the button over here is clicked that says compare results, then it'll show that by setting the comparison variant right here to a value, so it'll be shown. So I'll go ahead and say const show comparison. This will be equal to a function that receives variant, which is a ClinVar variant. We'll then say if variant dot equal to result. So if we do have a result, then we can say set comparison variant, set that to the variant, comparison. Then we can paste that in down here. So I'll just paste in the function like this. And then if I hop in here and then just refresh the page, then we can just try out an example. So I'll just try out this example right here, pick the first one, and then we can do an analysis. This one, for example, I'll just wait for this to finish. So it just finished, and then as soon as it finished, I saw this overlay right here. So that is what we just created inside the variant comparison modal. So we see this overlay as well as hello. And then we just need to create an actual overlay, so a white card on top of this grayed out background with a close button, so you can go ahead and close it again if you want to. Now first I'll just replace this hello with a div and close that div as well. I can then say class name, put in a max height of 90 viewport height. So I'll say 90 viewport height like that. Then I can go ahead and say width full. And then I can say max width 3XL, overflow, Y auto, rounded, large, and then BG whites. And then I'll just create a header for this modal which we're creating. So I'll put in a div. I'll then just say modal header, make that a comment. Then I can put in a class name, border bottom, border, hashtag 3C, 4F, 3D, at 10, and then a padding of 5. And I'll just need to spell this correctly. So border bottom and then padding 5 like this. And now we see the white box which we were hoping to see. Inside this, we'll just create the text. So first I'll create a container for the text and the button to close it. This container will have the class names, flex, items, center, and justify between. Inside this, I'll just put in first of all some text. So I'll put in an h3, so a heading tag. This isn't the largest one, but still a pretty large one. I'll say variant, analysis, comparison, put that in. Then we'll just style that a little bit. So I'll say text large, font medium, and text, and just put in this color right here. So it'll look like that. Then I'll put in a button as well to actually close the modal again. I'll just import the button and then say on click. So when you do click it, I'll just call the onClose function that will nullify this, set it to null, and then therefore not show the modal anymore. Then I can say X as an icon, so we can import this X icon from the seed React. So it is going to look like this when we save it. And then we just need to make it a little bit nicer. So first I'll say variant ghost. I'll say size small and save that. Then we can just define a size right here. So I'll say height 5, width 5. And we can put on some classes right here. So I'll say class name, height of 7, width of 7, cursor, pointer. So right now when you hover it, you don't really see the cursor change. When I save and we put on cursor pointer, we do see the cursor change now to indicate that there is an action by clicking this right here. Cool. Then we can say padding 0, and we can say text, and put in the usual text color, so this one, and I'll say at 70%, and we can put in hover. So whenever we hover, we say BG E9EEA at 70%, and we can say hover text, and then just put in the regular text again. So I'll put it in right here. So it'll look like this, and then whenever we hover it, we can see the text change the color a little bit, indicating the action. So that is exactly what I wanted. And then for the modal content, I'll just say modal content. We'll put in another div. In here, we'll say class name, padding 5, and we'll then put in an if statement. So we'll put in a conditional where we check whether comparison variant and comparison variant dot equal to result is there. If it is, we'll render out a div. This div right here should have class name space y6. I'll just save that, and then we can start by putting in some variant information. So I'll put in a div to store this. So I'll then say class name rounded medium. I'll put in a border color. So just this one up here again at 10. I'll also put in a background color E9EEA at 30%, and I'll put in a padding of 4. So it'll look like this. Inside this div, we can go ahead and put in an H4, so kind of a heading like we did with the H3, just smaller. I'll then say variant information, save that, say class name, and then margin bottom 3, text small, font medium, and I'll put in a text color as well. This text color is going to be this one, and I'll save that, and now we have a box to store the variant information in. I'll then go ahead and create a grid with 2 different columns, so I'll say div. Inside this div, we'll put in the class names grid, and then gap 4. On medium, we'll say grid columns 2. Inside this, we'll put in a div for each item in the grid, so for the left and the right column. So this will be for the left one or the first one. Inside this, I'll just copy this div. Inside this, we'll put in another one where we say space y2. We can then put in another div in here, and this should have the class flex. Inside this, we'll put in a span that says position. So when I save that, it'll look like this. We can then just copy this span right here and then put in the location. So I'll go ahead and say comparison variant dot location, save that, and now we have the location as well. Then we can just format the span text extra small, and for the position we'll also give it text extra small. We'll give it width 28, and then also this text color up here. So I'll paste this in and say at 70%. So that is what it is going to look like right now. Apart from the position right here, I'll just copy this one and then create another one for the type. We can then say comparison variant dot variation type, save that, and now we have the type as well. And then what I would like to do is just copy this div right here all the way down to there, and then at this position I'll just put in 3 more end divs. I think 2 is the correct amount. So now we have the second column in the grid, and this one should be called variants. In here, I'll then just show the variant in its correct color. So what I'll do is show something like A and then 2AT. So it's going to look like this. And then with the correct color for both A and T. And then I'll also just style this to be font mono. So it'll look like we saw earlier on. So first I would like to match the reference and also the variant. So what you see right here, the reference and the variant from the title. So we'll hop into compare results again, and then we'll just use the same regex as we used before. So we can go ahead and just do a solution-wide search and say .match, and then we should be able to find the same regex. So this one right here. I'll just copy this one, hop back into variant comparison, and then I would like to do the following. I'll put in curly braces first, then I'll put in a normal set of braces, then I'll define a function inside of this, and this function we can then execute like this. This means that we can return some code inside this statement, which will then be rendered out. So let's say we just say return and then h1. Well, then this h1 will then be rendered out like this. And this then allows us to match the to run the regex inside of the code instead of having to do it all the way up here. So this just makes it a little bit more concise in the code. So we can then say const match, and then we would like to match the comparison variant.title. Then we can just extract the reference and the alternative. So we'll say if match and match.length is equal to 3. Then I'll say const underscore ref alternative is equal to match. Outside of this, we can then say return a React fragment. In here, we'll put in a span, and we'll just need to remove this other return statement and save this. This span should have the ref. So I'll just go ahead and include this return statement in here. And then if the return statement is not, if the if statement is not true, then we'll have to return something else. So I'll just say return comparison variant.title like that. So I think we messed up the braces a little bit. So I'll just make sure we don't do that. So right now we need to take this entire return statement, put it inside the if statement, and then down here we can then say return comparison variant.title like this. So the first span is going to be the ref. The second span is going to be this symbol right here. So it'll be becomes, and then we can also show the alternative right there. So it'll look like this. And then we just need to actually go ahead and color these. So what I'll do is just use the function we created inside utils, the coloring utils, and then we can use that to get the class name. So I'll say class name, call this function, just import fonts, and then also call it with ref like this. And then I'll just force this to be true. And then I'll do the same for the second span down here. So it'll look like this. And this just needs to be old. So now we have the correct coloring as well, A to G. And now that we also show the reference and the variant, I would like to also just create a link that you can click to actually hop into the variant. So down here, I'll put in another div. I'll just need to close that. Then I can say class name flex. The first thing inside of this div will be a span that says ClinVar ID. Then we can just format this a little bit nicer. So I'll just take the same things as we did up here. Right there, paste that down as the classes. Then I can put in an anchor, so a link. So I'll say anchor. And then we can put in comparison variant.ClinVar ID. Then I also want to put in an icon, so external link. And this should have the class name ML1. Inline block h2 width 2, so it'll look like this. And then we just need to format that. So we already have another link that actually looks like this, and that is the orange one up here. So we can just search for what that links to, and it is something like ClinVar, and then slash, perfect. That is the one right here. That is what we'll refer to. So I'll just copy that, and then in the anchor, we can put in the href, and just paste in the link. And this just needs to be a formatted string instead. So we'll need to do that. And let me see, right now we have a comma, we don't need that. And this then needs to be comparison variant.ClinVar ID instead. We'll then also color this a little bit. So I'll say class name and then text, extra small, text, and we'll use the orange color again. So DE8246. I'll save that. So now we have this link. And whenever we hover it, I would like to put in an underline. So when we hover it, we see the underline like this. And lastly, I'll just put in target. That should be underscore blank. This means it'll open up in a new tab if we do click it, like this. Great. So now we can just say item sensor here. Save that. So you can see the difference. If I remove that, then we can see that it is a little bit above the sensor. If I then put in items sensor over here, it is then centered vertically along with the text right here. So that is what we wanted. I'll then just make this a little bit bigger as well. So instead of h2 and width 2, I'll put in a3. And then I'll just put in the same orange color right here, save that. So now it looks way nicer in my opinion. Cool, so with this done, we can go ahead and put in the actual comparison results. So under the variant information, so under this div right here, we can put in another div and call this variant results. And this will just need to be a div as well. So I think we put it one level too high, so it needs to be inside this space Y. So it actually is, so that is perfect as far as I can see. Now for the class name, I'll say, I think we'll just leave that out, we don't really need that. I'll put in an h4, we can just copy that h4 from up here, and then call it analysis comparison. So we'll have another page right below, or sorry, another section right below. Then we'll also have a card and a grid. So we have the same thing right up here. So the grid I'll just copy to my clipboard and then hop down here below the analysis comparison. I'll then just put in a div and then put in the grid right inside of that. And then close down this div. Now the outer div will have rounded MD, border, free C, 4D, sorry, it's going to be free C, 4F, and then 3D at 10, and then we'll just say BG wide and P4, so it'll look like this. And then the first column is going to be the clinical variant assessment, so not the one we're currently doing ourselves with Evo2. This is what we compare against. So first we'll say div, then we can say class name, rounded medium, background, E9, EEA at 50, and then we can say P4. I'll then put in an H5 in here, so heading 5, and then we'll put in ClinVar assessment as the text, and then just style this H5, so I'll say class name, then I'll do margin bottom 2, I'll do flex, items center, I'll put in gap 2, text extra small, font medium, and I'll put in the usual text color. I would like both the text that says ClinVar assessment, but also a circle right next to that in a little bit a unique style. So I'll say span, put in another span into this. The other one, I'll say class name, flex, height 5, width 5, items center, justify center, rounded full, as well as the background. So this one, and then I'll say add 10, and then for the inner circle we can say class name with 3 and height 3. We can then also say rounded full, and then put in a background color again. So it'll look like this. That is the circle I am talking about. Under this, I'll then put it in the classification. So I'll say div. Inside the div, I'll put in another div. Oh, inside this div, I'll then put in another div. And then I'll just save that. The outer div will have class mt3. The inner div will then have some other classes. First, I'll just put in the text though. So I'll say comparison variant dot classification or unknown significance if that is not there. So now we can see the styling while we actually do it. So first I'll say class name right here, make it a conditional, a string that is formatted so we can use conditions within it. I'll then say wFit, so with fit, so it fits the text. Then we can put in rounded medium, px2, py1. We'll put in text, extra small, font normal. And then for the conditional right here, we'll put in get classification color classes and pass in the comparison variant dot classification. When I then save this, we can see likely benign, and it looks like it does over here inside this table. So this is the clinical variant assessment over here from the database, but we also need to put in the Evo2 prediction. So I'll just make this a comment, say div. I'll just need to add a int tag, class name, rounded, medium. We can then also put in bg E9EEEA at 50% opacity with a padding of 4. I'll then just make this a little bit larger over here so they are side by side like we would see it on a proper screen size. I'll then just copy this circle we did up here from the H5 and paste it in right here, and then change this background to DE8246 so it'll be a orange circle instead. Then I'll also just change this to evo2 prediction and save that. Then under this H5 I'll put in another div. Inside this div I'll say class name mt3. I can then put in one more div and close that as well. I think we closed it too many times. Oh, no, we actually did not. And then we can say class name inside this div. First of all, we would like to put in a comparison variant.evo2 results.prediction. That is the first thing, and I would like to also just put in the shield we saw up here. So I'll put in the shield as well as a class name. So I'll say height free, width free and height free, like that. That is going to be class name, not classification. Then I can just import the shield. It seems like we'll need to do the analysis once again because it reloaded over here. So I'll just reload this analysis and then just continue here while we do that. So for coloring this shield and also the prediction, so it looks like the shield that will pop up right here, we can put in this class name. I'll first of all make this a dynamic formatted string, and then we can put in flex with fit items center. We'll just need to remove this quote right there. We can put in gap1, rounded, medium, px2, py1, text, extra small, font normal. We can then also put in this formatted one called get classification color classes. And here we'll pass in the comparison variant.evo2 results.prediction. And now it'll look like it does out here. Then down here, we'll put in the delta score. So this will be another div, also with margin top 3. Inside here, I'll say div and just close this one to class name, margin bottom 1, text extra small, text with the usual color. So let me just see if I can find that. Maybe right there at 70% opacity. Inside here, we'll then put in delta likelihood score like this. And then under this, I'll put in one more div. So I'll say div. I can then say class name, text, small, font, medium. And then I can put in the comparison variant that evo2 results.delta score. And then I'll just say to fixed. And then put in 6, so maximum 6 decimal points. This means that the delta likelihood score is going to look like that. And then I'll just put in some text that annotates what this actually means, so what a negative score means and what a positive score means, just so the user can kind of get a feeling of what this means and how bad it is or good it is. So we'll put in a div. Then I'll say class name, text extra small, text. And then I'll pass in the color and I'll say 60. Then I'll say comparison variant.evo2 results.delta score. If that is less than 0, we'll then print out the text. Negative score indicates loss of function. If it is a positive score, what we'll then print out is positive score indicates gain neutral function, because that is what it means. And then if you remember this confidence which we calculated, we can use that to show a pretty cool bar. So we'll say confidence bar and just comment this in. Then we can say div, give it the class name mt3. Under this we'll put in another div. Let me just copy this class right there. Or let me just copy the entire thing actually. And this one should then be called confidence. I'll then just put in a div below that. So div, I'll say class name empty1 h2 with full, rounded, full, and I'll put in a background color of E9 EEA at 80% opacity. In here, I'll then just create another element. So I'll say div, and then we'll say class name, make that a dynamic formatted string. Then I can say h2 rounded full. I can then say if the comparison variant.evo2 results.prediction.includes. If it includes pathogenic, then we would like to use the color right here. So that's going to be BG red 600. Otherwise, if it is benign, because it can only be pathogenic or benign, then we'll just use BG green 600, like that. And then the actual width of the bar will need to determine based on the style property. So I'll say style, pass in the dictionary of CSS properties. Then we can say width. And then the width will be calculated from math.minimum. So minimum between 100 or comparison variant.evo2 results.classification confidence times 100. And then we'll just pass in a percentage sign after that. So now the confidence will look like this. And we'll also just show the confidence right after. So I'll say div, and then we can put in, let me just see, math.round, and we'll put in comparison variant.evo2 results.classification confidence times 100. After this, we'll put in a percentage sign, and then I'll just format that. So I'll say class name empty1, text right, text extra small, and then the usual text color at 60%. So it'll look like this, the confidence, and also the bar with it. Then below both of these columns right here, I would like to put in one more div. So this box should contain the assessment agreement. So either this should print out something like evo2 prediction agrees with ClinVar classification, or if they are not the same, it should print out something like evo2 prediction differs from ClinVar classification. So I'll hop under right here, and then I'll say assess meant a agreement, make that a comment, put in a div. I'll then say class name empty4, rounded, medium, background is going to be E9 EEA at 20%. I'll say padding free, text extra small, and leading relaxed. Inside this div, we can say another one, of course, along with its closing tag. I'll then call this class name flex items center gap 2. Then we'll create a circle inside of this. So I'll create a span. This span should have the class name, and the circle should be colored based on the classification. So we can say flex h5 with 5 items center, justify center, rounded, full. We'll then also just pass in comparison variant dot classification dot to lowercase. So if the classification from the database, and I'll just say to lowercase to just always match them, if that matches the comparison variant dot evo2 results dot prediction dot to lowercase as well, then what we can use is going to be bg green 100. Otherwise, we'll use bg yellow 100 like this. So this, because they match, will be a green circle right now. Then inside of this, we can then put in if comparison. Let me just take this entire statement and just copy that. We then paste this in, put in the conditional again, and then say if they do match, we'll put in a check mark, so an icon with the class name h3 with free, text green 600. Otherwise, if they do not match, we'll put in a span, so just a span element. Inside the span, we should put in a paragraph that has a exclamation mark like this. So either it'll be a check mark or an exclamation mark. Then for the span, I can say class name, flex, height free, width free, items, center, justify, center, text, yellow, 600. I'll then save that, and now we have a check mark. But you see it isn't quite centered on the x-axis, and that is because we misspelled justify as justify something else. So I'll just save that, and now it should be correctly aligned as it is supposed to be. Then I'll just take this statement once again, and then under this span I'll put in one more. So I'll say span. I'll then put in the statement. So if it is there, then I'll print out evo2 prediction agrees with clinical variant classification. Otherwise, if they do not agree, I'll put in evo2 prediction differs from clinical variant prediction or classification. And then I'll just format this a little bit. So I'll say class name, font, medium, and I'll say text, hashtag 3C, 4F, 3D, and save that. So this is then what it is going to look like. Now down in the bottom, I'll just create a modal footer. So this modal footer will consist of a div with a button inside of it. This button should say close. And then for the class name, I'll say flex, justify end to center it, to justify it at the end. So to position it at the end of the modal on the x-axis. So justify end, border top. Border is going to be hashtag 3C, 4F, 3D at 10% opacity only. Background is going to be hashtag E9EEA at 30% opacity, and I'll put in a padding 4 as well. So padding 4, save that, and now we see this kind of footer with a border and then also a gray background color. And then for the button itself, I'll say variant is going to be outline, on click is going to be on close, and then for the styling, I'll say class name, cursor, pointer. I'll put in a border as well. That is going to be this one at 10% opacity, BG white. The text color is going to be this one again. So this one, and then I can say hover BG E9EA at 70%. So that is what the button is going to look like. Then when I do go ahead and click it, it closes down the modal, so we can close it both from here and from here. And then we can see the results, and they will be shown both on mobile, and this is the way it'll look, or on larger desktops like this, where it'll always be centered in the middle of the screen. And then I've just ran the analysis on all of these different ones just to find one that is that differentiates, where the results from evo2 are different from those from the database. So this one, for example. So if I click compare results, we can then just check out what that looks like. So we see uncertain significance right here. And then we see the exclamation mark as well as evo2 prediction differs from ClinVar classification. So that is just to show you what it look like if they do not match in their predictions. Cool. So that should be it for the variant comparison modal right here. So what we can then do is hop back in, close that, go into the gene viewer. In here we can create the card for actually doing a manual variant analysis by putting in a position along with the mutation that you would like to create. So to do this, first I'll hop into components, then create a new file called variants analysis.tsx. In here we can then say const variant analysis is equal to a new component, and then we'll just return a React fragment for now, and then say export defaults variant analysis like this. Then we can just take this component, hop back into the gene viewer, and then place this at the top. So we'll place it like this, import it, and now we should be all good. And then we can just create the properties that we'll need to pass down to the variant analysis card right here. So I'll just define the properties along with their type definitions. So first we'll have the gene, which is going to be gene from search. We'll then also have the genome ID, which is going to be a string. We'll have the chromosome, which will also be a string. We'll then have a clinical variants like that. It'll be an array of clinical variants like this. Then we'll have a reference sequence, that is going to be string or null. We'll then have the sequence position, that is going to be number or null. And then we'll have the gene bounds, which is going to be gene bounds or null, like this. And let me see if we can import this like that. Then we can just go ahead and destructure these properties. So I'll do this, this, and then for the rest of the properties as well. Reference sequence, sequence position, and lastly the gene bounds as well. Cool. Then inside the component, I'll go ahead and return a card. So hopping down here, we can return a card from UI card. We can put in the card header, like that. We can also put in a card title. Inside the card title, I'll put in variant analysis, like this. Then for the card, I'll say gap 0, border none, BG whites, PY 0, and shadow small. Save that. Then for the card header, we'll put in PT4, padding bottom 2. So you see there's a little bit more spacing now. Then for the card title, I'll put in text small. I'll also put in font normal and text 3C, 4D, sorry, it is 3C, 4F, and then 3D at 70% opacity. So it'll look like this now. And then for the actual card contents under the card header, I'll put in a card content. In here, I'll put in the class name PB4. Inside the card contents, I'll put in a paragraph and say predict the impact of genetic variants using the Evo2 deep learning model, like this. And then we just need to style that to match the rest of the style. So we'll say margin bottom 4, text extra small, and we'll put in the text color as well. And that is just going to be this one again. And I'll say at 80% opacity. So it'll look like that. Then we can put in a div right beneath this. So I'll say div, class name flex, flex wrap, items end, and gap 4. Inside this, I'll put in another div first of all. So I'll put in a div and then a label for the input field. The label should contain position. And then under this label, we'll need a input field. So first, I'll just style the label a little bit, margin bottom 1, to just space this out a little bit from the actual input field. And then I can say block, text extra small, and I can put in the text color as well. So I'll just say at 70%. And then under the label, I'll put in an input from Shadcn. Then I'll just close it like that. Say, wait a second, we'll just need to define a use states for storing the actual position. So right now we have an input field, but we also need to store the state. So up here I'll say use client because we are using states. Then I'll say const, and then variant position, set variant position is equal to use states. And then we'll use a string to represent this position. And then the default value should be gene bounds. And then we can say dot minimum dot toString. And if that is not defined for some reason, because we have the optional chaining right here, then we'll use an empty string, so just an empty input field instead. Cool. With this done, we can then hop down to the input, and then we can define the value, which is going to be the variant position. We can define the on change, that is going to be, and let's create a function called handle position change, and we can just hop up and create that function. So up here, I'll say const handle position change will receive a React event, so React dot change event. This is going to be an HTML input event. In here, I'll just open up the function, and first I'll say set variant position to e.target.value. We can then also quickly create two use states for storing the variant reference and the variant alternative, as well as the variant result, which we'll show within this card. So I'll say const and then variant reference, and then I can say set variant reference. I'll then say is equal to use states. This will just be an empty string. We'll then just copy this line and call this variant alternative. In here we'll say set variant alternative, and then we'll create one more use states. So this is going to be variance result, so the result of the analysis. We can then say set variance result as well, and this will be equal to a use states. And the use state can either contain an analysis results, or it can contain null if the analysis has not been performed yet. And obviously it'll be null per default because the analysis hasn't been performed before the button has been clicked. So hopping down here, whenever the position is changed, I'll just go ahead and set the variant reference to nothing as well. So whenever the position changes, the old variant reference, the variant reference isn't currently matching the variant position. So I'll just go ahead and reset that so we don't show old data for the new position. And now that we're up here, I'll create some more use states. The first one will be isAnalyzing, and then set isAnalyzing, which will be equal to a use state, which will just be a boolean that is false per default. And then also create one for variance error and set variance error. This will also just be a use state that can be a string, or it can be null if there is no error. This will just be null per default. Then we can just hop back down to the input field and then style that a little bit. So I'll say class name h8 with 32 border. The border should be 3C, 4F, 3D again, and then add 10. And I'll then say text extra small like that. So now we have an input field that matches our style a little bit more. I'll then just copy this entire div right here and then paste it down. The inner div right there. And then paste this inner div down and then say alternative. So this will be the second input field. So that is going to be the variant. So the nucleotide that you would like to change the reference to. So the value for this one should be variant alternative. And we can then say the on change. So the on change will receive an event. In here we can then say set variant alternative to e.target.value to uppercase, like this. And then for the input we can also say placeholder. Placeholder can be egat, like this. And we can then also say max length is going to be just one character because the nucleotide is only one character. And the class name can then again, we do have this actually, so we don't need to change the class names. I'll then just hop up to the variant reference and set that to an A, scroll back down, and then we can show a little annotation that shows what you're changing from, so the reference, and what you're changing to, so the alternative. So I'll just put in a conditional here where we check if we have a variant reference. If we have a variant reference, then I'll return the following div. The div should have the class name, margin bottom 2, flex, items center, gap 2, text, extra small, and text. The text will be 3C, 4F, 3D. So I'll save that. Inside this div, I'll put in a span that just says substitution. I'll save this. Then I'll put in a span right below this. This span should contain another span. Also another span. I'll put in the variant reference right here. So it'll be an A. Then I can give this some classes. The first class is going to be font medium. And the second class is going to be the color of the actual nucleotide. So that will then be get nucleotide color class, where we pass in the variant reference. This should then color this red right here because it's an A. Then what I'll do is just hop into the browser and search for right arrow symbol. And then we can copy this symbol right here, hop back in, and then put in a span. So I'll say span, put in the arrow, save that, and now we have a small arrow. Then I can take this span right here, paste it down below, and then we'll just say variant alternative for this one, and variant alternative down here as well. Then when I put in something like AT, we can then see A becomes AT. When I put in an A becomes an A, etc. And then for a D, it is gray because that really doesn't exist. So that is exactly what we wanted. And then another cool thing we'll just do is say if variant alternative is there, then we'll show that. Otherwise, we're going to go ahead and show a question mark like that. So if we do not input anything, we'll see a question mark indicating that the user should actually input something right here, such as a G or an A, etc. And then with this done, we can go ahead and put in a button for doing the actual analysis. So in this button, I'll say if we are analyzing, so is analyzing, then I would like to show a span saying something like analyzing, so analyzing. Otherwise, if we are not currently analyzing, I'll say analyze variance, like this. I'll just import the button, and then we can go ahead and customize this a little more. So first of all, right here, I'll put in a span, and this should be a span with a spinner. Then right outside, I'll say analyzing, like this, ... And if we set is analyzing to true, just to test it out, we can then just see what it looks like while we're designing it. So right now it says analyzing. Then for the text, the span, which will be the spinner, I'm going to say class name, and I'll put in mr2 inline block. I'll put in h4 with 4, animate spin to make this spin. I'll put in rounded full, save that. Then we can say border 2, border whites, border top, transparent, and align middle like this. So this looks pretty garbage, to be honest, and that is because I put in rounded roll instead of rounded full. So now it looks like something that is an actual spinner, so that is exactly what we wanted. Cool, let me just see if I spelled the rest of this correctly. And it seems like I did, so that is great. And then let me just make this go back to is analyzing. So I'll save that, and now we can see the analyze variant button again. Then I'll say class name h8, cursor, pointer, background, 3C, 4D, 4F, 3D, and then I'll say text small, extra small, text whites, hover, and then we'll use the same background, just with a slight color difference, so I'll make it down to 90%. Then we'll also put on some more things. I'll say disabled, and we'll disable this button if we are currently analyzing. So if is analyzing, or not variant alternative, so if this field right here hasn't been populated yet. This means that if I hop over here and then say T, we are able to analyze. If I say A, we are able to analyze, etc. I'll just do one more thing though. I'll make sure that it is disabled whenever the position is there, whenever the position has not been populated yet as well. So I'll say position. Let me just see what it is called up here. Variant position. So I'll scroll down. Then we can only use this button whenever the position and alternative has been filled in. So this just needs to be negated. So if it is not filled in, so if I take a position and then I take a variant, now we will be able to actually click this button. Now we can go ahead and define the function that is being run whenever we click the button. So that is going to call the handle variant submit function. So we'll say const handle variant submit. And this will call our endpoint on Modal and run the inference with equal to actually determine a prediction. So I'll say is equal to an async function where we have a position that is a string, and we also have an alternative which is a string. So this is the mutation and this is the position of the mutation. That corresponds to the two input fields right here. Good. This function will first do some input validation. So I'll say const position is equal to pass int position. So we'll just pass this to an integer just to make sure it is not a string, for example, because then we can't really use it. Then I'll say if is not a number position. If that is the case, we can say set variant error to please enter a valid position number. In here, we can then also say return. Then what I'll do is check whether this alternative nucleotide inputted is one of the 4 nucleotides, so A, C, G, or T. So I'll say const, and then define a regex for validating those. So valid nucleotides is equal to a regex. So I'll do / and then this upper tick right here. Then I'll put in A, T, G, and C, so one of these. And then I'll end the regex. Now I can say if not valid nucleotides.test, and then we test whether the alt is one of these. Then I'll say set variant error to nucleotide must be A, C, G, or T. And then we can just return, because then we won't do any of the logic below this as well. Now I can say set is analyzing is true. So these are the guard statements. I'll just check if our information is correct before doing the analysis. And then if these pass, then we can go ahead and do the actual analysis. So this will be set to true after this point. Then I can set the variant error to null, because now if we do reach this point, there aren't really any errors. So we just remove any current errors that are already there. Then I'll put in the try catch to do the actual analysis. So try catch, inside the catch, I'll catch an error. And we can do a finally, where we set is analyzing to false. Inside the catch, what we'll do is say console.error, and we'll error the error like this. And then also set variant error, just so the user know an error happened. And then we'll show failed to analyze variants. Then inside the actual try, we'll say awaits analyze variant with API. We'll pass in a position along with a alternative, which will be alternative. So alt will pass in a genome ID, which will just be genome ID, and we'll pass in the chromosome. And then I'll just say await correctly, and then we can destructure the output. So this will be const data, and we actually don't really need to destructure this because this is an analysis result. So right after doing this call, we can say set variant results like this, and then we can pass in the data. Now we have the variant result stored in a use data pair, so we can go ahead and show it on the screen. Then we can take this function we just created and call it down when the button is clicked. So scrolling down to where we defined the button, we can put in an onClick. And then when we do click, so I'll just say onClick right here, we'll run this function, and then we'll pass in the variant position.replace all, and we'll replace all the commas with empty strings. So this means that if you put in something like this, for example, this will always also be valid, just because these commas will be removed as well. Then we can also pass in the next parameter, which will be the variant alternative. And then this should do it for being able to analyze a variant. Then under this button down here and this section in general, we can put in a box that appears if you enter a position of a variant that is already known and that we have the information about in our system. So to do this, we can find out if there is a match, and we'll do this down here. So first we'll check if there is a variant position. If there is a variant position, then we'll say and clinical variants.filter. We can then say variants where variants dot variation type dot to lowercase. Then we'll say dot includes, and then single, because we only want to do it for single nucleotide variants, because we won't be able to analyze these other types of variations anyways. So we'll just search for single nucleotide, or not search, we'll check for whether the title of the type includes single nucleotide. If it does, we can then say and then check if the two positions match between the position up here and then the position of the variant that was of one of the variants that we know down here. So we'll say and pass integer, and we would like to first pass the variant dot location, and then we can say dot replace all, and we would like to replace the commas with just nothing. And then we can say that needs to be equal to, and I'll just take the same logic right here. And this needs to be equal to the variant position from the input field up here. And then we can say dot replace all, and then we just do the exact same thing. Now what we can then do is say dot map, and then we'll just take the first one right here. So the reason we're taking the first element of the matches from the filter back here, so this filter will then return us a list of matches, and the reason we're taking the first one that we map is because we might have multiple mutations to the same position just by having different alternatives. So this location is supposed to be a G, but there could be a mutation both for an A, or for a T, or for a C, that the G is then changed to. So this leaves us with 4 different possibilities, and we'll just show the first one. So what I'll do inside this map is then say matched variant, because this is a matched variant. Then I can return some HTML for each of these. So in here, first I'll do some JavaScript, so I'll say const ref alternative match, and then we could just say title.match, and just copy this logic which we have done before. Hop back in here, just paste that in, and now we should have the reference and also the alternative from the title of the variant. This will need to be matched variant, of course, though, so I'll say matched variant the title.match, and then I think I'll just hop back in here and just take this if statement, and then we can do this, and then I'll say let ref be equal to null, and I'll say let alt be equal to null as well. And then in here I would like to say let ref be equal to the ref, and then let the alt be equal to the alt down here instead, so this one. And then to remove all of these red arrows, I'm just going to go ahead and return something. So I'll just return a rag fragment right now, so we don't have to look at that. It is kind of ugly. And now if both are null, then we probably won't have anything to render. So we'll just go ahead and say if not ref or not alt, then we'll just return null. So return nothing and don't render anything. Otherwise we'll return a diff. So in here I'll say return diff. And then because we're inside a map right here, we'll need to put in a key or specify key. The key is going to be matched variant clinical variation ID. And then we can apply some styling. So I'll go ahead and specify the class name as well. MT4, rounded MD, border, a border color as well. That is going to be 3C, for ref, 3D at 10. And the background is going to be E9 EEA at 30%, and we'll specify padding 4 as well. Then we'll just hop in and find one of these we can test with. So I'll just grab the first one and input that one's location, and we're getting an error over here where it can't read this ClinVar variance because it is undefined. So what I'll do is just hop up here to ClinVar variance and just give it a default value of just an empty array. Then we hopefully won't get this error unless it is still undefined, which it shouldn't be. So now putting in the location right there. And I also completely forgot up here at the gene viewer, we forgot to actually pass down the parameters or the properties. I'll just see what this error is right here and then try and fix that. Use type, great, and then we can hop back in here and then just specify the different attributes. So gene is going to be gene. The genome ID is going to be genome ID. The chromosome is going to be gene.chromosome. Clinical variant ID is going to be ClinVar variants. The reference sequence is going to be a state variable which will track up here. That'll keep track of the active sequence and reference nucleotides. So I'll just scroll down and then create a new one. So I'll say const active sequence position and set active sequence position. This will be a use state that can either be a number or null, and we'll just set this to null per default. Then I'll copy this line and then call this active reference nucleotide, and then just a setter for that as well. And this should also be not a number but a string, and then it'll just be null per default. Then going down, we can say that the reference sequence is going to be reference or active reference nucleotide, and the sequence position will be the active sequence position. The gene bounds will just be gene bounds as we know it, like that. And then we should be able to actually have this work whenever we render it out over here. So if I go ahead and try out the example, enter a position of a variant we know, we are now able to see this gray box, which is the only thing we return. If we then say 8 instead, then the box is gone because there isn't really any mutation or any variance matching that position. Then we can also just try and remove the commas, and it still works. We can also remove one of them, and it still works because we are removing the commas up here with replace all. What I'll then do is hop into this div which we created and create another one, say class name, margin bottom 3, flex, items center, and justify between, like this. Inside this div, I'll put in an h4, and I'll say known variance detected, save that. Then we can just style this as well. So I'll say text, small, font, medium, and I'll give it a text color. So 3C, 4F, 3D, and then I'll save that. Now under this h4, we can put in a span that says position, along with a forced spacing right here. Then we can put in the location. So I'll go ahead and say pass, or I don't have to do that actually. I can just say matched variance dot and then location. And now we see the position over here as well. Then for the span, I'll say class name, text, extra small. And then we'll just give it this color, but tone it down to 70%. So text extra small, then we'll tone this down, save that, and now it'll look like this. Then we can go ahead and put in a grid with the information. This will have 2 columns, so I'll say grid, gap 4. Then on medium sizes or above, we'll say grid, columns 2. Inside this, we'll put in an empty div, and then a div more that says variance details. I'll save that and give it some class names. We'll have margin bottom 1, text extra small, font medium, and we'll give it a text color as well. And that is going to be this one. Then we can say 70% opacity to just tone it down. Now under this variant details, I'll put in the matched variant title. So I'll say matched variants dot title, and I can say class name text small, like that. And then under this div, we can put in one more where we say class name, margin top 2, and text small. So when I save that, it'll look like this. And then I can put in a gene.symbol along with the variant position. So variant position right here. Then I can put in a forced spacing, and under that put in a span where we say font mono. Inside this span we can put in one more span, and this one should have the reference, and we'll just style this a little bit. So I'll say class name and then pass in the color class. So we'll use get nucleotide color class, pass in the ref, and then we can copy this and pass in the alt, copy this and remove the class name, and then we can pass in this larger than sign like this. So now we can see what it is changed from and what it is changed to. We just need to call this alt though to make the colors actually work. Then under this div right here, I'll put in another div. Inside this div, I'll say ClinVar classification. And then I'll just make this with some classes. So I'll say mg1 text extra small, and then give it a text color as well. And I'll tone it down to 70%. Copy this one up here and save that. Then under the clinical variant classification, I'll put in a span. Here I'll put in the matched variant dot classification like that, or I'll say unknown if it's not there. Then for the span, I'll give it some classes too. I'll make this dynamic, and then I'll say ml1 rounded sm px1 and py of 0.5. And then for the dynamic part, I'll insert a variable which contains the CSS classes. This comes from get classification color classes, which we created from matched variant.classification. So now we can have this color around the box as well, just like we do down here. And I think I'll just make this 2 instead of 1, just so it looks like it does down here, just a little more. Then what I would like to do is to create a button over here that looks like these buttons that says analyze with evo2, but this button should say something like analyze this variant instead. This means that you can actually analyze the specific variant. So this will just insert the A into the alternative box and then press analyze for you automatically, so you don't have to do that. So it basically just makes it easier for you to analyze variants that already exists if you do enter them into the position field. So first I'll hop down to right here. I'll then put in a div In this div, we'll have the class names flex, items center, justify, end, and then we'll scroll up to where we created this button, and then just copy the entire button, hop down into the div, and paste it in, and press save, and then I'll say analyze this variant instead. So I'll say, or actually I'll change it a little bit. So I'll put in a rag fragment, so we can have multiple items inside this conditional. Then I'll say sap, so put in a sap icon from Lucid React with the class names mr1, inline, block, age free with free, and then next to the sap I'll put in analyze this variant, and just make this variant uppercase like this, analyze this variant. Then I'll make everything a little bit smaller, so I'll say h3 with free mr1 instead. This should be fine. Then hopping up to the button styling itself, I'll make this height 7. I'll make the variant outline, so I'll say variant outline, save that. I'll make the size small. And then I would like to change the background to E9 EEA, and I would like to change the background when you hover it to at 10%, but keep this one still. And then for the text, I would like to change that to the color we always use, that one. So I'll just use that for the text and save. And now we have this button that says analyze this variant. It is disabled though, which it should not be. So I would like to only disable it if is analyzing is currently true, because you don't really need to enter anything to click this button. And then I'll just apply a border styling as well. So I'll say border, and then I'll tone this down to 20. And then we have this button right here, which we can hover and then also click analyze this variant with. If we do then click it, I would like to just change this a little bit. So put in some curly braces so we can have multiple lines inside this on click, and then paste this back in. And then say set variant alternative to alternative. And then we'll just call handle variant submit again, but we'll say alt down here, and then just pass the value from the alternative, so this map function, that what this map function returns, directly into the handle variant submit. Because if we call this variant alternative, this set variant alternative, that is a use state setter, so this might take a while to update, and this means that this might be called before it updates, and then it'll use an old variant alternative value for this actual submit. But we want to use the newest one, but still set this, so we'll just call it with the value we have directly, and then set this in the meantime in the background. So that is why we set it with this, and then set this before. If I then just hop over here and click analyze this variant, we see that it is analyzing, and this alternative was automatically set to A, which is what comes from the variant down here. So that seems to be working. Now, the next thing I would like to do is just put in the error state. So I'll say if variant error, and then I'll put in a div. Here we'll put in the variant error, and then I'll say class name mt4, rounded md, background red 50. I would like to put in a padding of 3, text extra small, text red is 600, and save that. And now if we do have an error, let me just check. If I do true, and then I make this a string, that is what the error is going to look like and where it is going to be. So that was just for demonstration. Anyways, right now we just ran this analysis, but we didn't see any results. And that is simply because we didn't put in anywhere to show the results. So down here we can say variant results, because remember, we have this use state up here for storing the result. So that is automatically stored, and now we just need to show it. So if the variant results, then we'll put in a div with the class names mt6, rounded medium, border, and then border 3c, 4f, 3d, toned down to 10%. We'll put in a background too of E9 EEA at 30%, and we'll put in a padding of 4. Then inside this div, because we do have a result right now, so that is why we see it rendering out like we do right there. Inside this, I'll put in an H4 that says analysis results, and we'll just style this a little bit. So I'll say MB3, text, small, font, medium, and then I'll put in a text color as well. This one. Save that. Under this h4, we'll put in a div with a grid of 2 columns. So we'll have a grid, gap of 6. On larger devices, we'll have grid, columns 2. Inside this, I'll put in an empty div. Inside this div, I'll put in a div with the class name, margin bottom 2. Then I'll put in a text inside of this. So I'll just make this a div for now and call it variants. With the class names, text, extra small, font, medium, text, and then FreeC, 4F, 3D at 70%. Then we can, and this needs to be a FreeC, not an EC. So this right here. Then under this variant, I would like to put in one more div, say class name, text, small. Then I can put in the gene.symbol, and I can put in the variant results.position, a forced spacing right here. Under this, I'll put in a span. Inside the span, we'll have the variant results.reference. We'll then have the larger than sign, and then we'll have the variance results, and then .alternative. If I save that, we can now see the variant up here. Then what I would like to do is just style this a little bit. So I'll say class name, font, mono, and now it is in the correct format that we know. Oh, let me just hop back to where we were. So all the way down to font mono here. Then under the span, I'd like to create a new div. So a div right here. So span, div, and then we create a new div under this. This div should be empty with one inside of it saying delta, likelihood, score. This needs to be delta, not delta. Then we can say class name, text, extra small, font, medium, text. And then we can say FreeC, 4F, 3D at 70%, like this. Then we can just put in the delta score. I'll just say div, pass in the variant results dot, like this, dot delta score, and then just dot to fixed to only keep 6 decimals. Then I can say class name, text, small. Under this div, I'll put in one more, and I'll say negative, score indicates loss of function. Then I'll give it a class name and say text, extra small, put in the usual text color. So this one right here, and then tone it down a little bit. So that is what you see right there. We have the header, we have the score, and then we have the annotation explaining what the score even means. So negative score indicates loss of function. Then we can put in the prediction. So div, under 3 divs, you can put in one more. That is going to be an empty div with another one inside of it. I'll just copy this one right here, paste that in, and then I'll say confidence and save that, and then just spell it correctly, confidence, like that. And then I can put in one more div, say class name, empty1, h2 with full, rounded, full, bg, e9, eea, and save that. So now we have this confidence bar, which we have also created earlier. Inside of this, I'll put in a div. Here we have class name. This is going to be one we can change with the format string. I'll then say h2, rounded, full, and then I'll put in the variant results dot prediction dot includes. So if we have pathogenic in the word, then we color it red. So I'll do this. Then we say bg red 600. Otherwise we color the confidence bar green, bg green 600. And then the length of the bar will be determined based on this style property. So I'll pass in this CSS dictionary, then say width, pass in a format string, which will calculate from math.minimum 100, so it can maximum be 100, or variant results dot classification confidence, and then times 100. and then I'll append a percentage sign to that and save it. And then we can just show the actual confidence number under that as well. So I'll say diff, and then just put in math.round. And I would like to put in variant results dot classification confidence times 100, and then append a percentage sign to that as well. So that's going to be 100 right here. On the class name, I'll say MC1. I'll say text writes. Then I would like to say text extra small. And we then also just put on a text color like this, tone it down to 60. So it is going to look like this. I then want to be able to click a nucleotide down here. So click this one, for example, then you should scroll up and focus into this position input, and then also fill out the position for me. So to do this, we need to do a couple of things, both in the variant analysis and also in the gene viewer. So first we need to scroll all the way up, and then we can export an interface called variant analysis handle. This will contain a focus alternative input, which is going to be a function that just returns void. Then we can wrap this entire variant analysis component in a forward ref. So I'll say forward ref, and then say variant analysis handle, and then we just need to define the props as a type instead. So hopping up, I'll say interface variant analysis props. Just define this, and then this is going to be the type both here and also defined here in the forward ref. Then we have to wrap the entire thing, so I'll add one more curly brace, hop all the way down and do the same down here, like this, press save, and then right there we can also destructure the ref, so we can also get this ref parameter. Then we can define a ref locally here, so I'll hop down to our use states, then say const alternative input ref will be equal to use ref HTML input element, and then we'll just set this to null. This will then be a reference to the position field up here. So scrolling down to our input variant position, we can then say ref is equal to alternative input ref, and then we just need to define or implement this interface and define what happens when this focus alternative input is called. So we can do that down here as well. So what we'll do is use a hook. So we'll say use imperative handle, then we can pass in the ref as well as a function. Then we can say focus alternative input, and then if this function is called, we'll say if alternative input ref dot current, then we'll say alternative input ref dot current dot focus. So this means we'll focus in on the input field that we saw over here, so that this is focused right here and this is not focused, so it'll do this for us basically just automatically. Then hopping back into the gene viewer, I'll just save this by the way, we can scroll all the way up to our use states, and then I would like to create a new one. So const variant analysis ref is equal to use ref, and we'll then pass in the variant analysis handle, set this to null. If we then take this, scroll all the way down to our known variants right here, we can then pass down the ref. Oh, sorry, it's going to be not the known variants, but the variant analysis right there. We can then pass down the ref. And let me just see what it says right here. This probably needs to be type or something. Yep, it does. And then if we scroll down to our sequence component, we have this on sequence click. So I'll scroll up to somewhere where we have other sequence logic right here. Then I'll create a function handles sequence click. This will be a use callback, and then the function which we'll define is going to take a position, which is a number, a nucleotide, which is a string, and we'll then say set active sequence nucleotide, position. This will then just be the position. Let me just import use callback if it hasn't. I think we need to just define the dependency array. Yep. And then we also need to set the active reference nucleotide, which will be the nucleotide right here. Then we can say window.scroll to. So I'll scroll to the top, and then I'll set this to 0 to scroll to the top. Then I'll say behavior I think we need to pass this in as a dictionary, and then behavior is going to be smooth, and this needs to not have a U. Good. So it should scroll to the top and focus on this input. So to focus on this input, we'll say if variant analysis ref dot current, then we'll say variant analysis ref dot current dot focus alternative input, which will then call this function implementation from in here, and then focus on the actual input field. So now we just need to take this handle sequence click, hop down to on sequence click, and call that instead. And then if we hop into the variant analysis, we can just put in a use effect. So this use effect, I'll just pass in a dependency array. So whenever sequence position or reference sequence updates, so this is the position and this is then the reference nucleotide, whenever these updates, we'll then update them locally and store them right here in these two use states. So I'll say if sequence position and reference sequence, we'll then say set variance position, and we'll set this to a string of sequence position, and we'll say set variant reference, and we'll set this to the reference sequence up here. So I'll press save, and then if I hop over here and click one of these, it'll focus in on the position. So I'll just try and scroll down and click it again. So it scrolls up for us, and then also inputs the position that is correct. So if I click an A right here, we can see that the reference changes to an A, then I can input a T, and then we can see what substitution is taking place. I have forgotten one thing when I look back, and that is to put in the actual prediction inside this analysis result. So what I'll do is just create a div here, and then wrap this entire confidence section in that, give it a class name of mt3, and then in here I'll put in one more div. This one will say prediction, and it'll have the styling like we do right here as well for confidence. So I'll just put that in and save that. And then under this, we'll put in one more div. We'll then say variant results dot prediction, and I'll just save that. And now we can see the prediction, and we just need to go ahead and style that. So I'll put in some class names. Then I would like to say inline block, rounded, large, px3, py1, text, extra small, and then we can put in the get classification color classes, and then we can put in the variant results dot prediction and save that. And now we're able to see the actual prediction just like we are down here. So if I do take one of these locations, insert them in here, and then I put this to a G, for example, and then say analyze variant, we should be able to see this in just a second. And it just finished, and now we see the result down here. So it all works end to end now. I also just want to fix the spelling error up here. It says deep learning model. It should be deep learning model. So let me just find learning. So deep learning with an R. I'll save that to just make it spelled correctly. Then the last thing I would like to do inside the variant analysis component is the following. So whenever I click a nucleotide down here, it highlights the position input field, but it should actually just highlight the alternative instead, because when you do pick one down here, you have actually decided on the position, so it makes more sense that you want to enter the alternative. So therefore, this should be the one that is being focused and not this one over here. So this is pretty simple. Down here, we'll just need to change this to the input instead. And then when I save and click down here, now it focuses on the alternative input and not the position. And that is then it for the variant analysis card. So I'll close this down along with these other ones and only leave GeneViewer open. Inside the GeneViewer, I would like to first create a loading state, because right now if we scroll all the way up, we can see that is loading right here is not being used. So let's go ahead and scroll down and implement that. So if is loading is true, then instead of returning what we return right here, we'll return something else. We'll return a div with a div inside of that. The outer div should have the class names flex, h64, items sensor, justify, sensor, and then the inner div should have some class names as well. These should be h8 with 8 as well, animate spin. It should also have rounded full, border 2, border gray 300, border T gray 800. And then if I go ahead and set this to true, we should be able to see a loading state. If I hop back in here and just reload, so then I'll just press right there, pick one, and I can see that I forgot to put in a return statement right here. So that is why. So this is going to be how the loading state will look like. So I'll then say is loading and only render it if this loading state is currently active. One more thing we need to look at is the following inside variant analysis.tsx. So let's say we hop into a given gene, like this one for example. We can see that it starts out with A as the reference nucleotide. That is obviously wrong, and that is because we have set the default value for the variant reference to A. So we'll just go ahead and remove that and press save. This means we'll start out with a clean slate every time we press one of these, and then we can press a position, then put in a nucleotide, and then we'll be able to see what it is supposed to show. So I've created this section called EndNotes in the Excalidraw, and first we're going to talk about what we can do better next time. So first, as you might have noticed, we have entered in the same colors a lot of times. That is this specific color, for example, but also this specific background color. So instead of hard-coding these colors every single time, what you can do is in Tailwind, define a theme. So you can define a theme such as a color that you call primary text color or a primary background color. And then you define this color once, and then you will then be able to say background and then the name you defined. So then you can just say background primary, for example, or text primary, background text primary. And this means you'll avoid having to remember all of these different color codes and then also enter them every single time. And if you then decide to change some of these colors, instead of you having to go into all different files and change them all, you'll then only need to change the theme file, and then the colors of the entire application will then be updated as well. So that is definitely something that should have been done from the start. Next, you can also use pre-made components. So instead of defining this entire component from scratch and then also coding it, because it is a pretty complex component, it is a range slider. You might be able to find some of these components online from NPM. So this is React range slider input. So we might have been able to use this instead to avoid all of the logic behind creating this ourselves. It is pretty cool to know how it works though, but you can avoid a lot of work if you do use NPM packages instead. And then for the exercises down here, I've divided them into 3 different categories. Green are easy, these are medium, and these take the longest, so the red ones are the hardest ones. The first one is pretty simple. You just deploy the frontend to the cell. You might have to make sure that the linter will actually pass before you can build it and then deploy it to the cell, but this should be a pretty easy task. The next one that is a little bit harder is building auth around the application, so you can make this into a SaaS product by building a login system, and maybe you would also like to store the user's history, so history of generated predictions in a database. And then if you would like to make it into a SaaS product where anybody can just abuse your endpoints, you might also want to authenticate your Modal FastAPI endpoint. Luckily, there's already logic for doing this with Modal, and they have some pretty good documentation on how to secure your endpoints, so that shouldn't be too hard to do, but it's still a little bit harder than the green ones. The next one down here of supporting other mutations is by far the coolest exercise and the one you'll learn the most from. So right now we only support single nucleotide variants. So right here we are able to analyze single nucleotide variants, but not deletions, duplications, insertions, and other mutations. So let's say we would like to support deletions as well. So right now in the Modal backend we have a reference sequence and we have a sequence with a variant, so a mutated nucleotide. Then the scoring of both of those sequences is done, and then they are compared, so we take the difference between the scores. If you would like to support deletions, for the variant sequence you just remove the nucleotide that is deleted down here, and then you score both sequences, so the scoring logic remains the same. So that's why I say for deletion, the variation sequence should just delete the reference nucleotide, so nucleotide, but the scoring logic should remain the same. So then you would also need to change the frontend to also allow deletions and not just single nucleotide variants like we do right now. You will probably also need some way to differentiate between what kind of request you're making to the backend in Modal. So there's a difference between whether you're doing a single nucleotide variant and whether you're doing a deletion, for example. So this is something that the request to the Modal backend should also include. But you can start out by doing a simple one like deletion and then move on to other ones and then try out all of these if you would like to. So these were the exercises I have for you, and this is then the end of the video. So thank you so much for listening along, and I hope I see you in the next video as well.